म्हटले की मेघा कविराज.
Good morning, everybody. Dr. Manak, are you there? Dr. Manak?
Hello. Yes, sir.
Yes, are you there? Good morning.
Yeah, morning, sir. Morning. Very good morning. Good morning to all.
So you have come to office or are working from home?
Right now, I'm working from home, sir.
Okay. Yes. So let's wait for some more time, then we'll start.
Exactly. We need to wait for some time, sir. People are joining.
The chairman for today's session is Dr. M.S. Hadda. He's already
Yes,
logged
sir.
in.
Mhm.
And co-chair will be Dr. Sushama Sudhishri with Dr. Vivek Maria and Dr. Amit Mishra as conveners and Dr. Dev Kumar and Dr. S.K. Gupta as co-conveners. So, Dr. Manak, can you please just call them and then ask them to come online? Just check out with them.
Okay, sir.
Dr. Hadda is already there. Good morning, sir.
So shall I start?
Yeah, good morning, sir. How are you?
Yeah, fine, fine.
Yes, you can start. Maybe we can wait for 5 more minutes because
Okay,
the participants
sure.
are not yet in and 9:30 is also not there.
Sure,
But
sure.
sir, you have the program with you?
Yeah. I received the program. Even Sanjay also sent your program. Also, I received. I had on my mobile, and there
Sir,
are
in
approximately.
that among the posters listed for this session,
Yeah.
first of all, we like to call those for which the posters have been received by us.
Okay.
Then we can ask them to upload their own posters if other candidates are there.
Okay.
So we have received, you can tick it in your copy.
No. How? Okay. You can tell me which are
You can
available.
note down, sir. Yeah, we have poster number 4 by Ayushi Jha.
Just a minute. Arushi Jha. This is soil ministry. Okay, just a minute. Just. Yeah, Arushi Jha. Yes. Available.
We have poster number 7 by Geeta Kumari.
Number 4, number 7 by Geeta Kumari. Yes.
Number 8 by Dr. M.L. Gaur.
M.L. Goharia. Okay.
9 by Megha Kaviraj.
Okay. Yeah.
12 by Shekhar Singh.
Okay.
13 by Sonal Rani.
Okay.
17. These might not be listed because these
Yeah,
are
these are not listed.
17th is by Rehana Javed and 18th by Sanket Sahu. This you can note down.
What the name of 17? Name of
17 is
17.
by Rehana Javed.
Rehana. Okay. And
Rehana
18 by
Javed.
Javed. Javed.
REHANA JAVID.
JVI.
And 18th by Sanket Sahu.
Sanket
Sanket Sahu.
Sahu. Okay.
Ajit, please mute
So these are
yourself.
available. These are available.
Yeah, these have already sent our posters.
Okay.
Okay, so we can upload them, but others have not sent, they might present themselves.
Okay. Shall we wait for some time more? Shall we wait or shall we start?
Sir, we should wait. Still, it is not 9:30. We'll wait for 5 minutes and we'll start.
Okay.
I request all of you to keep yourself muted until unless you are speaking.
Sir, we have another presentation that is Bhagasri R. Jalgaonkar. So we received yesterday, late night.
Okay.
So we can include this one as a 19.
Okay. Yes, sir, you can please note it down also. After these presentations by Bhagyashri R. Jalgaonkar. So, Mainak, you have his poster, her poster?
Yeah, I have her poster, sir. I'm uploading this one.
So, do not upload it now, only when called for. Okay?
Okay.
You will be uploading the posters now? All the
No,
posters?
sir, I am uploading that poster in Google Drive, sir. No problem for that.
No, but it will not be possible for me to download it in the midst of the session.
Okay. No problem. Then we can ask the author. He can, sir, share that one.
You just send it to me by mail or WhatsApp.
Okay, done.
तुरंत नजदीकी रेल कर्मचारी से संपर्क करें, जिससे आपको तुरंत सहायता प्रदान की जा सके। आइए मिलकर अपने आप को और देश को सुरक्षित बनाएं। भारत
उमेश
Dr. Kohli. Dr. Kohli. Hello.
We can start now.
Okay. Good morning, all the delegates and participants in the e-poster session 4 pertaining to the aspect water conservation, management and improving water productivity. So this session, as we are starting approximately 15 minutes late, and listed time is up to 11:30. I think before that we are to finish by 11 a.m. So we are left with 1 hour 45 minutes only. So safely we can have 1 hour and 5 minutes. And through the participants, those who have uploaded their presentations, there are approximately 9 presentations which are to be made in this session. And I expect that nobody should take more than 5 to 7 minutes. So for 2 minutes, we can have the discussion and 5 minutes for your presentation only. So we are constrained by the time, though we are starting 15 minutes late also. So we have to compensate for the same. So please brief only to the point and refer pertaining to the most important aspect so that we are able to deliberate and finish the session in time. And along with me, there are co-chairperson, Dr. P.K. Rai. May I know, is he available? Dr. P.K. Rai, is he available? The conveners, Dr. V.M. Arya, Dr. Amit Mishra.
Good morning, sir. I'm available, sir.
Okay,
Good morning, sir. I'm Amit Mishra available.
Amit Mishra also available, okay.
Yes, sir.
And co-conveners, Dr. Dev Kumar and Dr. S.K. Gupta. Co-conveners, Dr. Dev Kumar and Dr. Okay. Anyhow, so with this is going to be an important aspect on which we'll be deliberating is the water conservation management and improving water productivity. So all of you know well the importance of this aspect. In this regard, already since I belong to PAU, the state government of Punjab has already taken initiative by making a water resource development and regulatory authority in the state of Punjab, and they have made the appointments also of the 2 experts and 1 person which is of the chairman level from the IAS cadre. So seeing the importance of the water, this part has been initiated by the state government of Punjab. I think to my mind, it appears that the government of Punjab is the first to take a lead in this aspect. And following the government of Haryana has also put a system to look into the regulatory system of mechanism with respect to the water use and its management. So first of all, I'll be inviting the poster number 4 by Ayushi Jha. Is she available? Ayushi Jha. Ayushi Jha.
Good morning, sir.
Good morning. Good morning.
Good morning, sir. Dr. Sushma Sudhishri wants to join. She is the co-chair of this session.
Okay.
But I think Dr. Mainak Ghosh or Dr. Kohli should let her in so that she can.
No, actually, this is not required now. Ask her to log on. This is open now.
No, there is problem. She is calling me that someone lets you in is a message coming. So you please address it.
I have disabled this waiting room. So there is no option for that. Okay, I'll try to enable it and do it once again. Okay.
Okay. Okay.
Ask her to log off and then log in again. It will be possible, I think.
Okay. Okay. With same.
Yeah, on the same link.
Okay. Okay.
So may I once again request Ayushi Jha, is she available? Ayushi Jha. Okay. If she is not available, then we can go to the next. Number 7, Geeta Kumari and Navneet Kumar, Geeta Kumari.
Yes, sir.
Yeah. So Dr. Geeta Kumari, you are to make your presentation. Have you sent your
Yes, sir.
Yes,
I
I
have
will load
already
her
sent.
presentation. Sir.
PPTs? Okay. So you can then start with your. We have a time constraint. So please manage within that time of 5 minutes, and 2 minutes after that we can have a discussion. In total,
Okay.
7 minutes. Yeah.
Okay.
Dr. Geeta, you can start presentation.
Please do in full screen mode. Hello.
Yeah.
Yes, you are audible, Dr.
Can I
Geeta.
start?
Please start.
But I am not seeing her on the screen totally. I am not able to see on total screen.
Yes, sir. I am also not able to see.
Some part is blackened.
Yeah.
Maybe
some
that's
platform.
I can see both of you as well as the presentation. I think, can you see the presentation, sir?
No, no, I am seeing only 1/4 part not visible. That is blackened.
अच्छा अरे कैसे हैं मैंने कहा ठीक है तैयारी चल
Last
रही
part is a
Maybe there is
black.
some
है इसमें
bandwidth
चल रहा
problem.
है ना अच्छा इसमें
It
कोकन
should
में
take
नहीं
some more time.
Okay, you can start, no problem if whatever way is
Can I
visible.
can I share the screen, sir?
बत्तीस की कर
Yeah, sure, sure.
So please.
I have shared your poster, Dr.
Yes,
Geeta.
already.
You need not share it.
इसको
Yeah.
It should be visible to all
Yeah.
in a few moments. Otherwise, I do it again. Just a minute.
Is it okay, sir? Is it visible?
वो कर रहा
Dr. Devakumar, please mute yourself.
Dr. Devakumar, please mute yourself.
Everybody except the speaker should mute.
Hello. So now
Yeah,
it is visible.
Yeah.
Dr. Geeta, please unmute yourself and start.
Okay, now I'm audible, sir.
Yes.
Good morning to all of you. Respected Chairman, sir, Co-Chairman, sir. Today I am presenting a poster on topic effect of integrated nutrient management on carbon sequestration under maize wheat cropping system in Alfisol. As we know that soil organic carbon is an important index of soil fertility because of its relationship to crop productivity. Declining soil organic carbon level often leads to decreased crop productivity. Thus, maintaining soil organic carbon level is essential for long-term sustainable agriculture. Carbon sequestration in general increases under practices of balanced fertilization, organic amendments, conservation tillage, and resulting in higher crop yield and higher accumulation of soil organic carbon as well as mitigate greenhouse effect. Thus, continuous application of 100% NPK along with lime, FYM, and integrated nutrient management practices had beneficial impact on restoration of soil organic carbon, maintaining soil fertility, and ill sustainability of maize wheat cropping system in Alfisol. Therefore, present investigation was undertaken to observe that the long-term effect of manure, fertilizer, and lime application on soil organic carbon status, soil quality, and ill sustainability under maize wheat cropping system. In materials and method, this is a permanent manual trial which was in progress since 1956 under maize wheat cropping system at BAU Ranchi on acidic red soil. And the design of experiment was RBT replications 4. Number of treatments which I was selected was 7. And these are the treatment details which was given in on the screen. And as we, in general, we know that the soil organic carbon level increases under practices of balanced fertilization, organic amendments, conservation, tillage, etc. And all these practices lead to increased crop residue return into soil, which promotes soil organic carbon accumulation. And this figure reflects the trend of soil organic carbon under permanent manual trial. That it reflects that the increase in organic carbon level was observed from the inception of this experiment. And FYM application increased organic carbon considerably in the first 20 years, but integrated use of this fertilizer and manure increased organic carbon significantly after 20 years. And so between these treatments, there was a decline in yield for FYM treatment as comparison to integrated use of FYM and chemical fertilizer. So for sustaining higher yield increase in soil fertility status, integrated nutrient management is desirable. This table shows the long-term effect of continuous cropping, fertilization, manuring, and liming on some biological properties, that is carbon dioxide evolution, soil microbial biomass, carbon of soil under permanent manual trial. And the table revealed that there was a significantly higher evolution of carbon dioxide and microbial biomass carbon, that is 54.7 to 62 mg per 100 gram soil of carbon dioxide and 105 to 105 to 0.4 to 100 and 121.3 mg per kg of microbial biomass carbon in soil receiving FYM alone or integrated use of this fertilizer and manure. And higher carbon dioxide in FYM treated plot indicate that when we use FYM, then it improved microbial population, which may have resulted in increased metabolic activity by utilizing substrate in the form of root exudates, whereas FYM may have helped in evolving more carbon dioxide. COT evolution and microbial biomass carbon also increased with the addition of phosphorus, potassium, lime with nitrogen, but only in nitrogen treatment. The carbon dioxide evolution and microbial biomass carbon were lower than control. Higher CO2 in FIM treated soil is indicative of nutrient turnover at higher expenses met through added organic carbon. And these are the different practices which we sequester carbon in the soil, that is cropland, reduced tillage, rotation, cover crops, fertility management, erosion control, and irrigation management. Then finally, we conclude that this continuous application of 100% NPK along with integrated nutrient management practices had beneficial impact on restoration of soil organic carbon, maintaining soil fertility and ill sustainability of maize wheat cropping system in Alfi soil. Hence, soil carbon sequestration is a natural, cost-effective, and environmentally friendly process. Thank you.
Okay, Geeta Kumari. So from the audience, anybody would like to interact? My co-chairperson or the conveners? Anybody?
Dr. Geeta, your treatments notation is not clear. A and B, A plus B, C. Can you explain what is it actually?
Sure, sir.
Sure.
So my screen is not visible. Host disabled the screen.
Huh. Okay, okay. You can tell me. I have seen that, na?
Yeah.
Yeah.
So, sir, in treatment A and B stand for full dose of phosphorus and potassium. That is 90 kg P2O5 and 70 kg K2O per hectare respectively. Whereas X and Y represent the amount of phosphorus and potassium present in full dose of FYM applied on the nitrogen basis to meet 110 kg nitrogen per hectare.
Okay. Okay, so the now what is the yield trend in respect to the carbon? Carbon is improving. So how much yield is improving over the T3 treatment? That is 100% NPK. What is the effect presently on the yield?
Sir, yield is actually soil organic carbon is an important index of soil fertility
Yeah.
because it's relation to the crop productivity. So when we add FIM and integrated use of FIM with chemical fertilizer, the soil organic carbon increases, and the grain yield of maize wheat is also improved, means also increased. And if the yield and productivity increase, the load of carbon is also decreased in atmosphere. The yield was not mentioned here, but
Yes.
we
Indian
calculate the
Car-
sustainable yield
Indian
index,
Guy
which is derived from the actual yield over the year.
Yeah, that is why I just want from my
हाँ
curiosity,
गुंजन
is there is sustainability?
हेलो हाँ गुंजन
Yeah,
Is there,
sustainability is
हाँ,
there, sir. We derive the
अगर
sustainable
काल
yield
पे
yield
index from actual
are
पार्ट
yield
अब
over
in,
नहीं
the
था वो
year.
था ब्रह्मानंद
And
का
if the
बहुत पार्ट दिया
sustainable yield index
ब्रह्मानंद को
is
जानती हो
higher, it indicates more progressive management
अरे
capable of
भरोसा नहीं यार
producing
हम पे
higher yield over the years.
अरे अपनी कसम नहीं तो पागल हो गया
Ivanshu, please unmute yourself. Anybody from the audience?
I request the chair and the speaker to unmute themselves, please.
Yeah, so I was referring that you made a presentation on the integrated nutrient management on carbon sequestration under maize wheat cropping in unfree soils by imposing a 7 treatments, T1 to T7, and you inferred that approximately 100% NPK plus integrated nutrient management improved the soil organic carbon sequestration to a larger extent. And you have referred probably it takes 20 years to build up. How much time you infer that it took for building up of the soil organic carbon in the soils? How much time does it take for building up of the soil active organic carbon pools or the carbon sequestration? Is there any way of inferring the time period from our observations? Dr. Geeta Kumari, are you listening to me?
Dr. Geeta, please unmute yourself and reply to the question.
Oh, sorry, sir. Sir, actually, this experiment was progressing since 1956 under maize wheat cropping system, sir. And this was my PhD work. I 2 years I work on this experiment, and this was started from 1956. So the organic carbon content they estimate from year by year. So I have through this graph I showed that how the soil organic carbon was accumulated, the trend of soil organic carbon, how it was increased.
No, trend is
I
okay.
also observed the active and passive pools. Passive pool I also observed. In passive pool, humic acid, fulvic acid I also observed. In active pool, carbon dioxide evolution, soil microbial biomass carbon, and water soluble carbon, water soluble carbohydrate. And in the slow pool, particulate organic matter. These things I observed in my studies.
Okay, so there is one observation from your studies. I think you can fit in the kinetics or the other kinds of the modeling approaches, which will help you in probably to how to interpolate in between the years what was the status of the soil organic carbon, or other. I think you can seek the help of your colleagues or the other friends. That will help you in more polishing of your research work. Thank you very much, Dr. Geet.
Okay,
Now we'll move on to the next presentation, the presentation number 8. So the presentation number 8 will be by Ayushi Jha, Dr. Murari Lal.
Yes, sir. Yes,
Is
I am
he
available.
available?
Yes, I am available, sir.
Okay, so
Are you able to listen me?
yeah, just a minute. So he'll be making a presentation on enlightening livestock water productivity under water stress situation, a way forward. So this you are going to make a presentation about your research work or
No,
your
it's not a research work, sir. It's a kind of a policy framework. It's a
Okay.
reviewed food of thought.
Okay.
Sir, will the screen will be shared by them only because it is showing
Yeah.
host disabled the
Yeah.
participant screen sharing.
Yeah, sure. Dr. Kohli, please share her screen, his screen, please.
Because I'm getting
This is
message.
by Dr. M.L. God.
Yeah, Emil got.
I'm getting a message that host disabled the participant screen sharing.
No, I didn't. They'll be going it. No problem.
Okay.
Dr. Geeta, please unmute. Please mute yourself.
So are you sharing my screen, sir?
Yeah, I'm sharing your screen, Dr. Gaurav.
Okay, so please make it full view. Okay. Please make it full view. Yes, okay. Thank you, friends. Good morning, friends. By this e-poster, this is actually not a research project or some kind of data. It is a kind of some food for thought for policy, because we are talking right now on the water a lot, water conservation, water management. Then now let us talk is water productivity. But even the water productivity is very confusing. Whole world is fighting what is actual water productivity. It is not the number of grams per liter of water, rather it includes some virtual water also. So my intention is here to incorporate livestock water productivity, because we are the country which are having highest number of cattle in the world. We are country having highest milk, meat, egg. But do you know how much water is being utilized by this sector, livestock sector? And the overall agricultural water productivity is largely depend upon the livestock water productivity. So my presentation will include just some general aspects and information, because global animal production, the latest estimate requires about 2422 gigameter cube of water per year. And this includes mostly the green water and 6.2% blue water. But problem is that when we manage our water resource systems, do we consider the concept of treating animals? Because the livestock water productivity is not only viable for the conservation, it is viable for the livelihood as well as environmental resilience also. So we can have three kind of broader interventions to improve the livestock water productivity. First intervention depends upon the feed management, because livestock gets his water mostly from the drinking water. 80% of the water required by the livestock is by drinking water, but we are not providing them sufficient amount of drinking water. The animal has to walk away kilometers, so it loses energy and it creates soil erosion problem. It creates many, so the most of the water to the animal is provided by feed, and feed is generally either green feed or it is a concentrated feed or grain or some kind of pasture of grasses. But our grassland and pastures are deteriorating, so the amount of the water that is provided through feed is very less and very, very unscientifically managed. So the concept of livestock water productivity revolves around the managing feed, how to manage the feed. So how to improve the quality of the green water from our grasslands and rangelands, how to manage it. So in agricultural water policy, livestock water policy is not being included right now. So there is a need to give food for thought that it should be an integral part of the agriculture water policy, because agriculture and livestock production are intrinsically linked, and they are equally crucial for the security of food, water, and energy. So this agricultural water productivity must consider livestock water productivity as well, because we don't know information that what kind of animals requires how much water at what time in what form and what feed could improve the animal water productivity, because feed we are giving randomly and the feed should be improved as per the quality of the cattle's. Every cattle has its own specific requirement that is given in the table. I'm not going to read it because time limitation is there. The final conclusion is that we require some modest interventions for improving the livestock water productivity. It could be done by providing adequate livestock drinking water facility. Number two, ample high quality feed. Number three, land and water improvements at the catchment scale, where the watering points could be near to the animal sets. Animals are not supposed to bark up to long distance, because if they bark, they take water, they lose energy, and product quality will be down. Then we have to promote some of the agro-silvipastoral systems in a catchment scale. Some portion of the catchment must be reserved for the animals' point of view, where the good quality fodder or pasture, legume-based pasture, they could be created so that they can get good water via feed. And there must be this concept should be integrated with the national water policy, because national water policy doesn't include the livestock water policy component. So this is the required, and we have to discourage the high number of animals in a small farmers holding, because animals are the interventions which gives more income. We are talking farmers doubling the farmers' income, but the farmers' income is mostly dependent upon the animal products, and that is given very less importance in current days. So the final line is that the barter productivity must be seen from the angle of livestock barter productivity as well. Thank you very much.
Okay, thank you, Dr. Murari Lal. You have enlightened the livestock water productivity under water stress situation away forward. So anyhow, you are talking separately with respect to the livestock water productivity under water stress situations. But if you go through the earlier systems of the farmer, they have the integrated system of farming. Okay, so they are raising the animals also, they are growing the crops also, they are meeting the local kitchen garden requirements also. So in together they were working. It is not that they are, through the tubal source of water or the other water resources, they are only using for animal purposes. So it's an integrated system. Naturally, as per some kinds of papers available through the literature, the people are referring that the consumption of water per unit of the body weight of the animal is higher in the livestock compared to the agricultural crops or the other arable systems of the area. So, you have to have a system of data operations, how you can gather the more information on this, and how you can gather the more data. Nowadays, the more concept of the artificial intelligence in agriculture is coming up.
Yeah.
So I think with reference to that, you can have the more data availability, and you can compile it and see that you can infer more in a better way uh, through the numerical simulations or the other kinds of simple to easily understandable modeling approaches. So where you can have the more uh, and certainly you can involve the policy peoples also. Some there are policy level institutions also. Maybe at the IARI level, they have the agriculture economics and other policy institutions. So you can strengthen more your research aspect with this area. So that will be more fruitful.
Sir, one small example I would like to quote here. See, if we are seeing the dairy industry. In dairy, we are using lot of water to clean the sheds, to clean the animal waste. And a good water is being wasted by cleaning the dairy farm and dairy products. Such kind of smaller interventions are not getting place in research and in policy framework. So this is one
Yeah, that's
example.
what I am saying. You need to characterize more.
Yes, sir.
You need to place the more emphasis
Yes, sir.
by putting your idea that this is what is lacking and this is what we need to put into the some kind of system or some kind of the columns or the which you need to incorporate more when you have to have the augmentation of the water resources for the livestock purpose.
Correct, sir. Yeah.
So that way that will be more beneficial with respect to that. So anybody from the audience would like to interrupt? My co-chairman, my conveners, or anybody else? Okay, thank you, Dr. Murari Lal. I think there is nobody else who would like to interact. Now we have the next presentation, the poster number 8. Poster number 8. Okay, 8 is done. Now we have the poster number 9. Sorry, 8 was by Dr. Murari Lal.
Yes.
Megha Kaviraj.
Yes, sir.
Pindra Kumar. Megha Kumari Lal.
Yes, sir. I am here.
Okay. So you
Please
can
share my
start.
screen. Yes, sir.
So she'll be deliberating on the coupling effects of vitamin C and Azotobacter cocum AVI2 on plant growth promotion under water deficit stress in rice. So she'll be talking with respect to the rice crop, how to have the plant growth promotion through the Azotobacter cocum.
Yes,
So
sir. May I
again,
start, sir?
yeah, sure.
Yeah, good morning, one and all presenting here. I am Megha Kaviraj, SRF and PhD scholar from ICR National Rice Research Institute, Cuttack, Odisha. And I would like to say thank you to the whole organizing committee who giving me a chance to present my research work in front of you. So let's start. So today my topic is coupling effects of vitamin C or ascorbic acid and Azotobacter coccoculum, that is strain AVI2, on plant growth promotions under water deficit stress in rice. So as we all know that rice cultivations requires a higher amount of water and in fertilizers. So drought is one of the major factor which decline its yield. So basically, drought creates an oxidative environment which produces reactive oxygen species or ROS, which damages the cell. And another aspect is excessive use of chemical fertilizer not only hampered but it deteriorates the soil health. Thereby biofertilizer is the best options, but most of the cases biofertilizers are not working, as when they are exposed to environmental stresses or factors, they eventually died. So before I coming to my hypothesis and objective, I would like to briefly explain my previous study. That is, I have taken that Azotobacter coccoculum AVI2, which have multi traits of plant growth promotions, and also it is endophytic diazotrophs, that it fix the nitrogen by itself, and it have many traits of plant growth promoting like ammonia productions, HCN, CEDRO4, and so on. We have taken that organisms as a model organisms, and we given different doses of H2O2 ranges from 0 to 5.5, and we found out, and we given the stress of H2O2 to creating the oxidative stress, and we found that more than 2.5 millimolar of H2O2 killed that organism. But once we used that 1 ppm of ascorbic acid, not only it enhances its survivability, but it also enhances its plant growth promotions. So then we observed its nitrogen fixing capability under those dosages, and check out that plant growth promotion traits, and we finally gathered the data and published just previously in Plant Physiology and Biochemistry journal. And from this, keeping those things in our mind, and finding after reviewing many literatures, and we found that there is no literature about combinatorial effect of that bacteria and vitamin C, that too under water deficit conditions. There are many literature on solo applications of bacteria, solo applications of ascorbic acid, but not the combinatorial effect. So for this experiment, we postulates this hypothesis, and we design an experiment, and you can see in my result table that we have taken the treatments of 5 treatments, that is flooded control, moisture stress conditions with bacteria, moisture stress conditions with that vitamin C, and the combinatorial applications of bacteria and vitamin C under stress conditions. And we have taken that 4 rice cultivars, 2 from drought susceptible, that is IR64 and Naveen, and 2 from drought tolerant cultivars, that is Ankit and Satyabhama. And from my result, you can see that we sir all know that is the stress indicator enzymes are the proline, and they are in surge or higher content when they are imposed with stress conditions. But you can see in combinatorial effect that proline surge is decline, and the antioxidants enzymes are also in good conditions. So we can see that combinatorial effect have some good impacts. And we did the we analysis chlorophyll content and the fluorescence chlorophyll imaging technique, the new technique we also did that. From that we can know that photosynthetic efficiency. And my slides gone, please back.
Dr. Kohli, please. Yeah,
please make the full screen. Yeah, back to my point. Yeah, thank you. And I did the chlorophyll fluorescence imaging. From that we can know that what is the photosynthetic efficiency of that cells, and photochemical quenching, non-photochemical quenching, maximum fluorescence. And actually basically the technique is we given the different pulses from 0 to 710 pulses, and from that electron transport chain through PS1 to PS2, we can say that yes, PS2 conditions, actually PS2 is very sensitive to the stress conditions. So you can see in my graph in figure 2 that in combinatorial effect the graph is showing the fluorescence emissions is much more, that depicted that the photosystem efficiency is much good in that conditions. But in that red line showing that moisture stress conditions, where the fluorescence emissions is little bit less. So, and this is in figure 2, you can see my agronomical data, grain yield and height data, and grain yield increased 34% and 44% in IR64 and Nabin, and 39% and 36% increased in Ankit and Satyabhama rice cultivars compared to the moisture stress conditions. And I also did that NEFA gene targeted quantifications. I found that inside the root, as I already told, that is this bacteria is endophytic in nature. That endophytic nature is already published in Planta journal through freight analysis, and we did the NEFA gene quantifications, and we found that in combinatorial effect, that NEFA gene copy number is much more in that treatment inside the root. And finally, after our measuring, after taking our measured variables in putting this in this PCA, you can see that in green circles or eclipses clubbed in a single group, which are the drought susceptible cultivars, and drought tolerant to cultivars are grouped in a single group. So from this I can conclude that based on the chlorophyll content, based on the chlorophyll fluorescence imaging data, NEFA gene quantifications, antioxidant enzyme assay, oxidative stress enzyme assay, and agronomical data, we can conclude that that bacteria and vitamin C formulations in future may be used as the novel formulations to alleviate drought stress, as well as enhances nutrient use efficiency in rice. And I must say that today we are doing some popular techniques are there: gene editing, gene mining, for enhancing or modifications of the capability of the functional traits of that gene. But if we do such easy and simple technique to alleviate or accentuate the functions of the gene, then this will be much better. Actually, those techniques, that CRISPR-Cas9 talent techniques, are very expensive, but if we got this promising result, then we can go for this one also. Thank you so much.
Okay, Dr. Megha Kaviraj, the presentation is on coupling effects of vitamin C and azotobacter under water deficit stress in rice. So I would like to ask, is it some kind of the laboratory plus field study, or totally field study from the beginning part?
Sir, first of all, as I previously told, that first we did an lab experiment that to giving different traces from H2O2 from 0 to 5.5. After that, we getting that such interesting result that yes, that bacteria can survive under those higher concentrations of H2O2 after additions of ascorbic acid. Then we planned for a pot experiment, and we still found a very good result, and that data we gathered, and it is already communicated in that plant physiology journal too.
So when you have done the pot experimentation,
yes,
like
sir,
since in the
yes,
like we go in the rice crop, so we go for the puddling systems in the field. So are you able to replicate so that kind of things in the pot
yes, sir,
experimentation?
yes, sir, yes, sir.
You were able to replicate same kind of situations
Yes, sir,
in the pot experiments.
Yes, sir. We are thinking for the field experiments too in future, sir.
Yeah, I think you can take it to the field until unless,
yes,
though
sir.
it reflects good that as in the human beings we are taking vitamin C in the pandemic time to build up
Yes,
our
sir.
immunity. Similarly, it responds well to the plant. That is also a living matter to have the more promotion under the water deficit in the rice. Under the stressed condition, the plant
Yes,
is able to survive. So you are talking with respect to the drought stress maybe, or water
yes, sir.
stress.
Yes, sir.
Okay. So ultimately that is the main part on which you have concentrated. Have you
Yes.
analyzed in your area where you have conducted the study that how much is the water deficit percentage, like in the season wise, in the summer monsoon months or the winter months, or what is the rainfall distribution pattern in your area prior to, if you really look from transferring your information from the laboratories to the field condition, from port to the field. So you need to have look at which are the places where you need to intervene and you have to take your way forward.
Yes, thank you, sir, for your great suggestion.
Okay. Anybody else would like to interact? from the audience? Okay, thank you, Dr. Megha Kavira. We have the next presentation as per that will be serial number 12. So this is by Shekhar Singh
Yes, sir.
and Sonali Kumra.
Yes, sir. I am available, sir.
You are Shekhar Singh is available. Okay.
Yes, sir.
So you have to make a presentation on analysis of groundwater quality of Haridwar region by application of Memoro Pollution Index method.
Yes, sir. Yes,
Okay.
sir.
Yeah. Okay. You can start.
Sir, please share my e-poster.
Yeah.
Your presentation is on the screen.
Yeah.
Okay, sir.
Please start.
Sir, I am audible, sir.
Yes, you are audible. Please start.
Audible.
Okay, sir. A very good morning to all chairmen and co-chairmen and all the dignitaries and participants. My name is Shekhar Singh and I did my MTech from GV Pant University of Agriculture, Pantnagar, Uttarakhand. I am going to deliver a e-poster presentation entitled with analysis of groundwater quality of Haridwar district by the application of Nimero pollution index method. Now, as we know, water is very essential to live on the earth, and groundwater resources are alternatively source of water in agriculture, industry, and domestic use. Since the groundwater is easily accessible for use uh, in India, this resource has been overexploited since past few decades. Hence, it is very important to monitor the groundwater quality. Now, uh, the objective of my research is to study the pollution contributing parameter of groundwater of Haridwar district using NPI method. Now the study area has been selected in the Haridwar district, consisting of 6 blocks covering the 4 tehsils, and the sample was 38 groundwater sample have been collected from the 6 block of Haridwar district. And in order to find out the pollution in groundwater sample, NPI method has been used, and QGIS technique has been used to make the map of this area. Now, in the table 1, as you can see that the pollution of the pollution of categorized water like NPI value more than 80% were comes in the slightly polluted area, and 10% is the water which is clean, and 5.26% are moderately polluted, just because of NPI value increase 0.25 to 4.25. And if NPI value is larger, more than 1, then our contamination or pollution is more in that area, as we can refer in this study. And figure 2 shows the pollution contributing parameter in groundwater, and the parameter was collected um, uh, on the basis of ICMR and WHO organization. And the in figure 2, we can see that 2 parameter pollution pollution contribute 3% of water, and 5 parameter contributing pollution around 62%, and sorry, 5 parameter pollution contribute 3%, and 3 parameter pollution contribute 62%. And the conclusion of this study, the pollution was just because of industry and their form, which should not percolate indirectly to the groundwater as it contains high amount of nitrate. So, thank you, sir.
Thank you, Dr. Shekhar Singh. As we have gone for the groundwater quality analysis, you have used the NMI index, the numero pollution index. Why you have taken only one pollution index method? Are they not available in the literature, the other methods where we classify them as per the analysis of the groundwater quality?
Yes, sir. So
Or
there
there
is
is
lot
some
of
basis of selecting this
yes,
over
sir.
to other methods?
Yes, sir. Sir, this method help to study the effect uh, uh, of any single parameter of water quality. It means, sir, if we want to give the reason for particular parameter like hydrogen, nitrogen, so it gives a brief knowledge about that parameter. That's why I choose the NPI method, sir.
See, since we are talking with respect to agriculture, is
Yes.
this method safe? Like since you are talking of the numerous pollution index method, have you gone for any kind of plant analysis also? You are talking as per the guidelines of your WHO and ICMR.
Yes,
So
sir.
is it more relevant to the human beings or to the agriculture sector also in the plants way also?
Sir, you are asking for the plants.
Yeah, yeah.
Sir,
Have you
but
gone for any kind of analysis or simply you have referred this with respect to the human aspects?
sir, I just refer to the human aspect, and this is the study of water quality. That's why I choose that.
Yeah, yeah. So I think this is better. I think in future also, if you will look into the whether the plant growth, see ultimately this is a life cycle from the plant, from the animal, from the plants to human beings to animals. So it is all integrated system.
Yes, sir.
So
Yes,
I
sir.
think it is better you can, if you, in future you can formulate a project involving the animal means veterinarian also. You can involve the agriculture people also. Though you have already taken care of your human people means ICMR or if some kind of the hospitals in your area where you can get involved with the doctors of the relating to this the pollution aspects. So that will be more beneficial for the society also and for the agriculturists also to come up conclusively that how it is going to help this analysis, or if that you are able to characterize this is how it is affecting the quality. So how to I mean better design it or where it has to be taken care of for the beneficial purposes of the society.
Okay, sir. I will.
Uh,
I will keep in future.
yeah.
I will.
Okay. Next,
Sir, I have a
Amit.
query.
Yeah, yeah, sure, sure. Welcome.
Yes, sir.
I have a query.
Yes, sir.
Uh, you have mentioned the conclusion that nitrate is the problem in the
Yes,
groundwater, but
yes, sir.
nowhere you have analyzed that nitrate. What are the indicators you have used for means for calculating the pollution and NPI? You say what were
Yes.
the means analysis part? Which element was analyzed? You have nitrate. I don't see in your slide.
Yes, sir. Sir, as I told, the NPI method uh, gives the result of single parameters. So, sir, I choose parameter
What was
like
that parameter?
हाँ sir, yes sir. Sir, electric conductivity, alkalinity, total diesel solid TDS, and turbidity, nitrate, and hardness, and sir, pH value. I calculate all the parameters. But sir,
These
in this slide,
these these parameter doesn't relate directly with the nitrate. First thing. Second thing, these cannot be justifiably the indicators for pollution. Okay, salinity, TDS, mm, maybe NPI was somebody who has calculated earlier. You have taken it from there, but they have number of parameters considered number of parameters, but you have only seen for the turbidity and pH and EC. That doesn't means indicate that there was a pollution, and especially with a very small number of samples, say 30 or 36. Hmm? There
yes,
are
sir.
large number of indicators, salinity hazards. There is toxic ion concentrations. So means complete analysis is required. Otherwise, saying on the basis of PATC and these things cannot give an idea that this water is fit or not fit.
Yes, sir.
Anybody else?
So means nitrate you have not analyzed, no?
Sorry, sir.
Nitrate, have you analyzed nitrate in the groundwater?
Yes, sir. I use it, but sir, the
Use means
result.
you have analyzed it,
Yes, sir. I analyze around, sir,
analyzed in
10 to.
laboratory.
Yes, sir. Proper testing. I did, sir, nitrate and which you are saying alkalic calcium and chlorides. All the analysis has been done in the laboratory, sir. But so I did not show the result of all.
Okay, anybody else?
What was the method used for nitrate estimation?
Sorry, sir.
Nitrate estimation method which you used? Hello? Okay, thank you. Thank you.
Okay, if there is nobody other else from the audience. Thank you, Dr. Shekhar Singh, for your presentation. Now, next we have Sonali Rai, serial number 13. So she'll be deliberating on impact of wastewater irrigation on soil and their management. Sonali Rai, please.
Thank you, sir. I am Sonal Rani.
Okay. So
Please
you are
share my
Sonal.
e-poster, sir.
Your name is Sonal or not Sonali? Okay. So
Sonal Rani.
okay. Yes. Yeah. You can start.
Yes, sir. Thank you so much, sir. Respected Chairman, sir, distinguished dignities, and all participating members. Good morning to all. I am Sonal Rani, second year MSc student of Department of Soil Science of Dr. Rajendra Prasad Central Agricultural University, Pusa, Bihar. I am going to present my e-poster on a very trending topic, impact of wastewater on soil and its management. Now let's start with introduction part. We are all living in a country like India, which accounts for 2.45% of land area and 4% of water resources of the world, but represents 16% of world population. And this precious resource is being contaminated by human action, and the prevalent cause is explosive population growth and industrialization along with urbanization. There are various sources which adds wastewater production are domestic, industrial, commercial and agricultural activities. And this wastewater contains many nutrients which have positive and negative impact. Others are pathogenic agents, heavy metal contamination, etc., which causes negative impact on human health as well as environment. Positive impact includes the nutritional security for poor who are living in the country. And in addition to health hazard, it also poses problems on soil like salinity hazard, sodium hazard, heavy metal toxicity, etc. Here is a figure which illustrates the data about status of sewage generation and treatment. We can see the outer circle showing sewage generation, which is 61,754 million liters per day. Out of that, there is great proportion of untreated water accounting 38,791 million liter per day. It means we have only treatment capacity of 22,963 million liter daily. And rest are either used directly for irrigation purpose or it meets the mainstream polluting the river. This picture is taken from CPCB website, Central Pollution Control Board Bulletin. Now switching to the next topic, that is objective. Our objective includes extracting pollutants and toxicant by various methods like editor titration method for calcium and magnesium, which is called total hardness, turbidity method for turbidity, and other parameters of soil, which includes pH, EC, organic carbon, available nitrogen, phosphorus, potassium, and sulfur. We are also aiming at reduction of BOD, that is biological oxygen demand, COD, eutrophication, etc., and increasing DO, dissolved oxygen. Now talking about materials and methods, many samples of water and soil has been taken from different part of the country, usually in the interval of 50 meter, starting from the point of disposal of industrial waste, and in case of soil, the sample depth is up to 15 centimeter, that is root zone of soil. And the various parameters like pH, EC, available nitrogen, phosphorus, potassium, turbidity, total dissolved solid, etc. are calculated. And the methods used are pH meter for pH calculation, electrical conductivity method for electrical conductivity, for organic carbon rapid titration method, available nitrogen by alkaline permanganate method, available phosphorus by Olsen method, TDS by conductivity method, chloride concentration by Moore titration method, etc. And salt index is also calculated for analyzing the suitability of wastewater to be used in irrigation. Now, I want to conclude my e-poster. There should be treatment of wastewater before disposal as per the pollutant contamination. The treatment may be up to primary level, secondary level, or tertiary level. There should be analysis of parameters like pH, BOD, COD, DO, TDS, and heavy metal contamination before irrigation. And in case of no treatment plant available, there is alternative of using wastewater for irrigation is to dilute the wastewater so that the concentration of pollutant gets under the permissible limit. And at last, I want to add one point. Wastewater can be managed to individual level too by using 3 R's, that is reduce, reuse, and recover. Thank you for your time and thank you for your time. Thank you, sir.
Okay, Sonal Rani, I have very basic and fundamental question. You have written in your methodology appropriate, several samples have been analyzed from regions across the country where wastewater is used as irrigation.
Yes, yes.
So, I mean, from which areas of the countries, which regions you have taken, and what is the basis of, have you taken with respect to some benchmark? Means, if you find as the purest form, then you are able to compare the impact, or you have not shown what are the areas from which we have taken the samples.
Sir, actually, I am studying in RPCIU Bihar, and there is one industrial region here that is in Muzaffarpur district. And previously, our seniors worked on that, and I collected information from that, sir.
No, you have taken only
I have
from
also
Muzaffarpur.
reviewed some literature, previous literature, sir.
Then you have not properly mentioned you have taken this data from the literature. It appears as if you have taken the written several samples have been analyzed from regions across the country where water is used. So
Yes.
you need to improve your methodology.
Yes, sir. Yes, sir.
And what another question was, have you used any benchmark index? I mean, did you find as the best area from the irrigation point of view? Then you are able to compare your results that with respect to their treatment and
Sorry,
they are
sir,
better
I
in.
haven't worked on this topic, sir. I'm sorry.
Then how can you compare the results? How far you are ever sure that whatever you are doing samples and when you have to draw the inferences of your study, how you will be able to really come to the conclusive part?
Sir, actually,
To my point, your hypothesis appears to be weak.
Yes, sir.
The hypothesis of your study, the objectives to extract pollutants, remove toxicants, kill pathogens, aims at reduction of BOD, COD eutrophication. So that you need to redefine.
Yes, sir. Yes, sir. Sure,
Yeah,
sir.
that will be much better. Only then you can have the better indicators of the analysis of BOD, COD, DO, TDS, whatever the parameters are required to the wastewater, and then see how far it is useful.
Yes,
Any
sir.
other from the audience, please?
Yes, sir.
Yes, sir.
This is Anshuman. Well, I actually really appreciate the efforts of students these days when they need to, when they think of participating in online conferences, etc., and then they try to review some topic and then present their work. It's really appreciable, but at the same time, you need to learn some things. There are some ethics that you should actually put those references. You should actually give credit to the of that work to the person who has actually done that. And then in addition, you see, it's nice that you have tried to do that and your other colleagues have also put up several posters like this. But then how to write a good review and how to make conclusions. There are set procedures. There is a process of meta-analysis which you can learn if you want. And then also even without that, if you want to conclude based on several papers, you have to analyze that data. You see, the very word review of literature, what does it mean? Review, if you are conversant in Hindi, you will understand the word review in translated into Hindi means samiksha. It's not just that you put on those references in one place. That is not a review, but you analyze those references and then draw your conclusions. There can be different methods of analyzing. But I think this is really appreciable. And you see, reviewing is one good way of learning about anything. If you make a literature review of any topic, you learn that topic very well. Okay, so best of luck, but there are many things that you should learn.
Thank you, sir. Thank you for your suggestion, sir.
Okay, anybody else would like to interact? Okay, thank you, Dr. Sonal Rani, for your presentation. And you have to take care of the observations by Dr. Kohli, and whatever I suggested, maybe that is also suitable in relevance to Dr. Kohli, which has suggested for improving your further work from the research point of view.
Yes, sir, sure, sir.
So we also have to take care of the time. Please be quick.
Yeah, okay. I think we are left with 2-3 only.
Yeah.
We'll be finishing in time, I think. Okay, so the next is by Rehan. I do not have the name. Rehana Javed. Javedkar? Okay, Rehana. So please have your presentation. Rehana, please. Rehana Javed.
Rehana, are you there?
Is Rehana available? Hello. Rehana Javed, yes.
Rehana, are you available?
Please unmute yourself and say whether you are presenting.
Yes, sir.
Okay.
So should I start?
Yeah. You can start.
Good morning, everyone. Myself, Dr. Rehana Javed. I have done my PhD from SCOST, Kashmir, and here I'm going to present a part of my research that's on the water use efficiency of high density sweet cherry under different irrigation regimes using the drip system of irrigation. Basically, the problem was taken into account because sweet cherry in Kashmir Valley is grown on carivas, which are totally rain-fed with poor water holding capacity, and farmers are not in a position to provide or to supply the crop the specific amount of water, and that too during the critical stages of the tree growth and development. So the problem was taken into account in order to calculate the total water requirement of sweet cherry, and also the water use efficiency to calculate the water use efficiency under the different irrigation scheduling that was used during the research, so that the farmer could give that specific amount of water at a particular stage, and also he will manage the water during the periods of drought, etc. The methodology that was used, I will briefly discuss here. the two this treatments were used. First was the irrigation levels, in which the four levels of irrigation were used. That is, when the crop, that is I1, I2, I3, I0, I1, I2, and I3. That's when the crop was supplied with no irrigation. That's during the I0, the crop was allowed to grow under the rain-fed conditions. And during the second level, that's the I1 level, 50, the crop was supplied with 50% crop evapotranspiration level of irrigation. Crop evapotranspiration was calculated from the data that was taken from the meteorological station, and also from the daily crop evapotranspiration that was taken into account. And for that, pan evaporimeter data was taken into consideration. And during the I2 levels, the crop was irrigated as per the 75% of crop evapotranspiration. During I3, it was supplied with 100% level of irrigation as per the crop evapotranspiration. And the stage that were used, that is T1, T2, T3, that is after the fruit set stage, at pit hardening stage of the fruit. And here the dates are mentioned, that is when the irrigation was supplied. And during T3 stage, that's when there is a fruit growth stage in the cherry. This is the method, these are the treatments that were used. And it was found that this yield, productivity, and quality, it was found better when 75% ETC level of irrigation was used, followed by the 50% ETC level of irrigation. As far as the water use efficiency was concerned, it was found maximum by using the 50% ETC level of irrigation, indicating that the crop uses efficiently, more efficiently the water when lesser amount of water is available to the crop. And also the total water requirement that was calculated for the years 2017 and 2018, it came out to be approximately 92 and 97 liters per tree. Based on this, the amount of water that is required by the cherry in liters per tree per day, it came out to be approximately 14 liters. And it was also found that the fruit growth and fruit set stages are most critical stages of irrigation for this crop, that's the sweet cherry. And also this water use efficiency, that is yield per unit water, showed a decreasing trend with increase in the amount of water applied in this case. And also the maximum value of water use efficiency came out to be 5.75 kgs per meter cube, which was recorded with when 50% level of irrigation was supplied to the crop. Lower water use efficiency at higher evaporation replenishment was due to the greater transpiration of the tree, thereby concluding that the efficient use of water by the tree during the periods of less water availability to the tree. Thank you.
Okay, Rehana Javed. So with respect to the relevance of the fruit also and relevance of the session, the presentation is very good. And water use, I mean, I have a query. I mean, for how long you conducted the study in the field?
It was conducted throughout the year. That is from
No, only for 1 year or 2 years?
2 years. 2 years.
For 2 years you have taken the data.
Yes.
And how much time does this crop take to mature?
It takes about it takes about 3.5 months to come into the fruiting. But after the harvesting, we have given another, we then started another level of irrigation after the harvest, that is post harvest period पे हमने एक और irrigation level start किया था, so that it will increase the yield for the next year. That is, and it was found that
जब आप प्रोजेक्ट में इफको का प्रोजेक्ट है,
Kumar, please unmute yourself.
दे रहे हैं तो इनके
Kumar, please unmute yourself.
ये ऐसा तो है नहीं, किसानों के यहाँ घर जाना पड़ता है लेने, एक बस भेजते हैं।
And it was found that the irrigation level that was given after the harvest of the crop, it came out to be fruitful for the fruit bud differentiation of the sweet cherry for the next year. That also increased the quality as well as production for the next year.
Hello.
Okay. So, I mean, have you gone for any cost benefit analysis also?
No, sir.
I mean, if you are really calculating per unit of water use efficiency for the high density plantation, since you have taken the irrigation timing 3, age of the crop 4 years, irrigation level 4, irrigation treatment 3, and you have determined with respect to the FAO method. So I think it is better you can have the critical appraisal at the first stage, second stage, third stage, if you are keeping up to that level, or since after that you are applying the one more irrigation, so you can look upon more critically that how you are more efficiently using the water. So what kind of system you installed in the area for this? Is it through the
It is drip.
drip system of irrigation? Okay. So have you recorded the yield also?
Yes, sir.
Because I was not able to clearly see your conclusion. That was written in a very small letters.
I have recorded the yield also.
Yeah. So will you please repeat your conclusions? Just you can read it. I could not see on the screen thoroughly. The conclusion part, can you see and repeat? I cannot see thoroughly.
Sir, I'm not able to see the poster.
Yeah, that's what I was also not able to see. But if you have available with you in writing, you can spell out.
Dr. Rihanna.
Yes, sir. Yes, sir.
Hello. I would like to congratulate you for your such a nice presentation. I just want to know about what is the actual water requirement of this crop, fruit tree.
It's about 16 liters per tree under the traditional system of irrigation, but here we have used the drip system of irrigation, which uses the every drop of water more efficiently. So it came out to be around 14 liters per tree per day.
That means 2 liter saving per day.
40 liter per day. This is the annual requirement or this is one time or
40 liters per tree per day requirement, and the annual requirement
per day.
around 92 to 95 liters.
Okay. Tree needs per day water.
Per day.
I'm not. Yes.
So per day is 40 liters.
Okay. Thank you.
Thank you, sir.
Anybody else would like to intervene?
What is the yield of sweet cherry, Ms. Rehana, per plant? Hello?
Sir?
What is the yield of sweet cherry per plant?
Sir, under high density plantation, it's around 7 kgs per tree.
Per tree?
Per tree.
Only one picking it takes place, or more than 2?
Yes, sir. I'm not you're not audible.
What is the number of picking in sweet cherry? It is harvested in a single picking, or more than 2, 3?
Sir, it's picked in one go actually, because it's yes, sir. Its storage is very less. That's why.
Okay. And what is the productivity per hectare of sweet cherry in Kashmir, in your area? Hello?
Sir?
What is the productivity of sweet cherry per hectare?
Sir, it's about 3.30 metric tons per hectare.
Okay. Thank you.
Okay, thank you, Dr. Rihanna, for your presentation. As I have already spoken, you have to work out about the benefit cost analysis so that it is really worthwhile to go to the farmers level. Ultimately, your fruitful research can carry the message forward.
I will incorporate.
Okay, thank you. Now we have the next presentation by 18 number. Number 18, I do not have the name of the person with me.
Sanket Sahu.
Who is the person?
Sanket Sahu.
Sanket Sahu. Okay, Sanket Sahu, you can make a presentation.
Sanket, are you there? Sanket Sahu, are you there?
Sanket Sahu.
I think he's not there.
Okay. Then we can have the next number 19, or I think the Sanket Sahu was 19. Or there is some Bhagyashri
B.R.
R.
Jalgaonkar.
Pardon?
Number 19, B.R. Jalgaonkar.
Yeah, B.R. Jalgaonkar, yeah.
Yes, sir.
Yes, you can make your presentation.
Bhagyashri, yes, you can.
Yes, sir.
You can start presenting. It's on the screen.
Am I audible, sir?
Yeah.
Good morning to all of you. Myself, Ms. Bhagyashri Ramesh Jalgaonkar, PhD Research Scholar in Department of Soil and Water Engineering, CTA MPT Udaipur. Today I will conduct my PhD work, e-poster presentation on delineation and hydrological response unit, that is HRUs, analysis of West Manas River Basin using soil and water assessment tool, that is SWAT model, under the guidance of Dr. Mahesh Kothari. SWAT is a conceptual model, semi-distributed and continuous in time, and it was developed to predict effects of different uses, covers, and soil management on water and sediment production, as well as water quality. Efficient planning and management of water resources and quantitative understanding of hydrological parameters in the river basin should be seen as important. The research focuses on SWAT model of residential scale to forecast the effect over a period of land, soil, and water changes. The figure first shows the location of basin was selected for this study. This included delineation of river basin, landmark, soil and slope definition. The data required was a stream dam for delineation of river map and a slope map for HRU generation. Figure second shows the delineation of river basin generated by the SWAT model with stream, sub-basin, inlet, figure and its 10 land use classes. Most of the area comes under agriculture area class and less area under wetland non-forest land. Figure 3 shows the soil classes which divided into two major classes, that is loam and silt loam. Most of the area was under silt loam. Percentage, it was divided into 5 classes. Majority of the area was under gentle very steep slope class. HRU analysis, was 1,596 HRUs were found for this in the classes. The conclusion of my study was delineation was accompanied tuning sub-basin parameters. The sub-basin parameters premeditated where basin the agriculture practices and forest land. Hence, any adverse effect due to climate change will have greater impact on these. Proper planning may be devised to sustain the agricultural practices in the basin and preserving the forest cover. Thank you.
Thank you, Dr. Jalgaonkar, BR.
Thank
So
you, sir.
you have used the SWAT model.
Yes,
See,
sir.
this model is already, I think, at large scale. I mean, the basic framework was done by the, I think, US people. So they have done this model. United States of America. I think they are the people.
Yes, sir. Texas.
Yeah,
Yes,
yeah,
sir.
Yeah, soil and water assessment tool.
Tool. Yes.
So your objective was to research focuses on SWAT model for residential scale to forecast the effect over a period of land scale, land, soil, and water changes. So you
Yes,
have
sir.
only analyzed from the residential point of view.
Yes, sir.
So since we are more concerned as per the agriculture aspects also, so how can you reutilize that information from the residents to the area in the agriculture sector? I mean, what benefits will accrue of your research for our agriculture sector?
Yes, sir. The model used for soil and water assessment tool, ना? That's
Yeah.
why the बेसिकली अपन जब फील्ड पे जाते हैं तो कौन सा एरिया किस क्लास में डिवाइड होता है वो पता नहीं चलता. तो इस टूल से क्या है सर कि जितना अपन को एरिया चाहिए, जैसे कि ये है 2152 स्क्वायर किलोमीटर एरिया है. इसमें कौन कौन सी सॉइल आ रही है, कौन से लैंड यूज हो रहा है और स्लोप कितना है, एलिवेशन कितना है. इससे अपन सर एक आइडिया लगा सकते हैं और कौन से एरिया में कौन सी क्रॉप ग्रो हो रही है, कैसी हो रही है, या फिर वाटर पैटर्न क्या है. इसी से सर अपन वाटर प्लानिंग भी कर सकते हैं.
वो तो कर सकते हैं. आपने ऑब्जेक्टिव में लिखा ना मॉडल ऑफ रेजिडेंशियल स्केल, तब मैं पूछ रहा हूँ. तो इसलिए आप इसको ऑब्जेक्टिव को थोड़ा रीडिफाइन कीजिए थॉरली, सो दैट वी कैन रियली कम टू द aspects on which you are looking for its utility for our otherwise I know that how to have the soil and water assessment since we are looking with respect to the system of soils and with respect to water also naturally the latitude of the area, the sloping slopes or what are the other geomorphologic or other kind of parameters that can be utilized to have. I mean, once you have started with this is your some doctorate work or you are under?
Yes, sir. PhD work, sir.
How many years of data you have taken?
20, sir. 1999 to 2018.
You took this data from literature or it was available at your place?
Available on SWAT site, sir.
On the?
SWAT site. Soil and water assessment side है ना sir, Texas की. जिसे लिया sir.
No, the area for which you are. So this study was basically you are undertaking at which part of the country?
Rajasthan, state of Rajasthan.
State of Rajasthan.
Yes,
sir.
It
means you have the information with respect to the Rajasthan.
Yes, sir.
Available with you, and those you are able to digitize the data of the Rajasthan.
Yes,
I mean, have you used any kind of aerial photographs or it was directly available the data, or you have to use something?
Yes, sir. From all data from the website, sir.
Okay.
Yes, sir.
Hello, Jalgaonkar.
Yes.
Hello.
Yes, there can be any interaction from the audience, please.
Yes. हम ये जानना चाह रहे जलगांवकर जी, ये हमें बताइए कि वो अर्बन सेटलमेंट, न्यू अर्बन सेटलमेंट को एस्टेब्लिश करने के लिए ये स्टडी बहुत बढ़िया है कि कौन से एरिया में अर्बन सेटलमेंट के लिए हो, और सरकार इस पर अर्बनाइजेशन पर सबसे ज्यादा जोर दे रही है कि वेरिएबल्स एग्रीकल्चरल फील्ड को ना यूज करके ऐसे एरिया को यूज करें जो हमारे लिए बेनिफिशियल है अर्बन सेटलमेंट के लिए. क्या इसको राजस्थान में इंप्लीमेंट किया जा रहा है अर्बन सेटलमेंट को एस्टेब्लिश करने के लिए, इस टेक्निक को या इस फाइंडिंग्स के बेस पर बना प्लानिंग किया जा रहा है? हेलो.
I think Dr. Devan Kumar, probably she may not be conversant since she's at the doctorate level. Of the state level, she may not be much conversant that how much that state government is utilizing. So she is at a research phase of the university at MPUAT. I think it will take time to really percolate to that level.
Sir, there is a growing need to save our precious agriculture.
No, I agree. The need is there. That's why she is taking a study area. And how much part is benefited, or how the government is going to ultimately, once the research benefits will be reaching the level of the policy planner or to the management people or the other areas, only then it will be slowly, I think, percolate.
Okay, thank you. Thank you, sir.
Okay. Thank you, Dr. Jalgaonkar, for your presentation. Okay. Dr. Kohli, do we have any other presentation listed now? Hello? Dr. Kohli? Hello? Dr. Kohli, do we have any other more presentation in this session?
Yeah, Ayushi Jha is waiting. You can call her.
Okay. Arushi, you can make your presentation. Arushi Jha.
Yes, sir. Yes, sir.
Yeah.
Sir, I can't share my screen.
Can't share your screen. Okay. You'll be able to share. Sir, your was listed number 4 as per the program adaptation of subsurface drip irrigation. Okay. Okay.
Yes.
So you are from PAU. Hello.
Yes, sir.
You are.
Yes, sir.
Okay. You are pursuing your master's or doctorate program? Or you are
Masters. I've just completed. Yes, sir.
MTech you have done. This is a part of MTech.
Yes.
Okay.
Yes, sir.
Okay, you can tell me. Okay, you can start. Yes, start please.
Very good morning to all. I'm here to present my topic, that is adaptation of subsurface drip irrigation for efficient utilization of fertilizer and water in wheat crop under Punjab condition. So we all are well aware that wheat is a widely consumed crop, and with the increasing population, its demand is also increasing. But the cropped area, which is under wheat production, is not increasing. So the other way to increase the productivity is by efficiently utilizing the water as well as fertilizer. And micro irrigation system have already proved its superiority over other traditional methods for efficiently utilizing the water as well as fertilizer. And under micro irrigation, subsurface drip irrigation is most efficient one, as it provides a concise and timely application of water as well as fertilizer. Under this study, the objectives were to study the fertilizer uptake efficiency of under the wheat crop, by the wheat crop, and to develop an fertigation and irrigation optimum schedule. There were 4 level of irrigation as well as 3 level of fertigation. Under 4 level of irrigation, 3 were at subsurface level and 1 was under the surface level. The first level was at 100% ETC, the second one was at 80% ETC, the third level was at 60% ETC, and the fourth, that was the surface irrigation, was again under 80% ETC. There were 3 level of fertigation. First one was at 100% RDF, that is recommended dose of fertilizer. Second was at 80%. Third one was at 60%. The fourth one, F4, it was denoted by there was no fertilizer applied at that level, and that was a control treatment for the sake of comparison. Then all this experiment was laid out in a split plot design. The nitrogen were applied in split doses as well, and the P and K were applied as a basal dose. Then statistical analysis was done using the CPCSM software. The plot was per plot is about 21.6 meter square. There were 3
Dr. Hari
replicas.
Satyanaran, please unmute yourself. Yes.
Then there were many parameters that I have observed, but I can't show here all. Basically, I have shown the yield. Under the straw yield, it was observed that the highest yield was under the I1 F1 level treatment, which was about 40% higher. And there was no significant difference between the I2 and I4 level of irrigation. Under the grain yield, the higher yield was observed under the surface level, which shows the conformity with the PAU recommendation from the package of practice. Then under the grain yield, there was no significant difference between I1, I2 level of irrigation and F1, F2 level of fertigation. Means either of the combination can be used. That result in 20% saving of water as well as 20% saving of fertilizer.
Then concentration of nitrogen was observed under the grain as well as the straw. The table I can't show here, but the result was the nitrogen concentration was higher in grain as compared to straw, 1.5 times higher, and with increasing level of with decreasing level of fertigation and irrigation, the concentration was declining significantly. And then at the this graph which is shown here, that represents the additional yield. Additional yield means whatever the yield was there of the grain as well as straw, subtracting it with the control, that is the F4 level of fertigation. Whatever was obtained, that was additional yield. So basically, it was observed that the grain yield was increasing with increasing level of fertigation. The grain yield was, the straw yield was increasing with increasing level of fertigation. The grain yield was also increasing with increasing level of fertigation, but not much increase in yield was observed when increasing from F2 to F1. My statement is actually not, this graph is not justifying that, it's not proper, sorry for that, but that was observed. So ultimately, coming on to the conclusion, it was that after all this work, we can recommend that using 80% irrigation level and 80% fertigation level, the result was similar to that using the I1F1 level, that is 100% one. So this level can be recommended for growing the subsurface drip irrigated wheat, that is I2F2 level was recommended after all my work. Thank you.
Ayushi, I mean, the table which you have shown under the material methods, what does this F1, F2, F3, F4 indicates? What does this
So these are the fertigation level at 100.
fertigation levels? Okay, you have not somewhere referred in the poster. It is not.
Yes, sir.
And similarly, I1, I2, I3, I4 are your irrigation level. Irrigation level. And what is this? Is it the figures under this? We are getting, what is this? This is your yield level of the crop. Hello. Like for F1,
Yes,
F1, I1, you are getting 6,570. What is this parameter? You have not defined
Sir, actually, I have changed.
What are the units
no,
of
sir,
this?
actually, I have.
Anyhow, what are the units of this? Is it crop yield, kg per hectare, or is it something else? Hello?
Probably.
Ayushi, are you listening to me?
Yes, sir. Sorry, sir. Sir, actually, I have changed my presentation, but I am not able to share that, sir. I have changed this
That's
table.
okay. I am asking what are the units of your this figures?
The kg per hectare, sir. Kg per hectare.
Grain yield.
This is a grain yield, okay.
No, sir. This is a straw yield. This is not grain yield. I have not shown grain.
Yeah, that's why. That's why aap mat boliye. Let her elaborate so that that is her work. Let her elaborate the things. So that gets a confusion in others' minds.
Yeah, actually, I would just like to ask one thing.
Yes, sir.
When you install a subsurface drip irrigation for a particular crop, you must customize it so that it is applicable to the whole system.
Yes, sir.
So, with wheat, how do you do that? What other crops are customized? It's not possible that you grow only one crop in Punjab.
Sir, sir, only one crop was not grown, sir. It was in rotation with rice. Sir, it was in rotation with direct seeded rice.
Okay.
You see? Hello.
Yes, sir.
Actually, that is more important in this.
Yes.
Now, when you do it for rice, you see, water use efficiency increasing, that is more important in rice than in wheat.
Yes, sir.
That should have been.
Ayushi, can you, have the harvest index value from your data with yourself? Do you know what is harvest index? If you can take together the straw yield plus the grain yield.
Yes, sir, I can have that.
Yeah. So you can make a total and then you see that is it really coming up? And then you can further deduce a conclusion also. You can add one more conclusion that how it is working with respect to your treatments or the interaction of the irrigation and the treatment levels. So that will be more conclusive with respect to your study. And what was the subsurface depth where you put up your irrigation system?
Yes, sir.
What was the surface depth?
Sir, it was 15. So 15 centimeter.
15 centimeter. Up to 15 centimeter we consider surface.
Yes.
Uske baad hai. So below, more than that.
Yes, sir.
So means
Sir,
that
20.
is the,
Yes, sir. 20 centimeter subsurface.
so that's what I was looking that at what depth you are putting it so that it could be. And have you worked out what was the cost of installing per unit area you have of the system this which
No,
you are?
sir. I have not. No, sir.
So go for that so that at least again one thing harvest index and another is if you have the cost of this thing, then again since we are having ultimately we look at how the permutation combination of surface drip irrigation to have the more efficient utilization of fertilizer and water. So we have to look on the cost benefit analysis also.
Yes,
Or
sir.
if there could be some more better index that you can take, that will be more worthwhile to check it. Anybody would like to interact more from the audience?
You see,
Yes, sir. Yes.
you have just said that the nitrogen was splitted. So I want to know two things, simple things it is. What are the dose of nitrogen in case of your experiment, that is for wheat, and what was the splitting? It was 50, 20, 20 or 25, 25, 25, 25?
The dose of nitrogen was 123 kg per hectare. It was applied in this
So what
rate.
is the splitting proportion?
So there were 16 splits applied, sir.
16 split.
Yes, sir.
Okay.
Yes, Dr. Devan, actually this is through drip, so this can be done. And that is why it will increase the nitrogen usage.
Okay, just why you want. Thank you, sir. Thank you, Mr.
Thank you, sir.
Anybody else? Okay, thank you, Ayushi Jha, for your presentation. And you can look into the suggestions made by Dr. Kohli or the Devan Kumar or to me also. So that will improve your more presentation and your work can be. Have you published your work anyway?
Not yet, sir.
Okay, you have just concluded with the study. You have
Yes,
completed
sir.
or still you are in the process of?
Sir, just completed, sir. I have to collect my PDC now. Yes, sir.
Okay, okay, your Viva and everything.
Yes, sir. Yes.
Good.
Thank you, sir.
Yes, Dr. Kohli, do we have another presentation left in the session?
I don't think, sir, it's time also. So it's better that you conclude.
Yeah, okay. Okay, thank you, Dr. Kohli. And I place on record the organizers of the Soil Conservation Society of India from the top patron of the society, Dr. Sanjay Arora, Dr. Kohli, and other chief patron of Dr. Suraj Bhanji, under whose dynamic leadership this program has been organized. And I have been invited to chair this session of the session number 4. So we had a large number of presentations in this. I mean, though as per listed we could not have, but we have in total, I think there are 9 to 10 presentations which took place in this session. The most of the presentations were very nice, and they refer to the theme of the area, and somewhere we have already suggested where they need to look for more under the water conservation management and improving the water productivity. They have to infer, since we are working for the agriculture sector, it's no doubt if we can integrate it, the system with the livestock also, or if we can integrate the system with respect to the society from their soil health, the human health, or the livestock health part. So that will be more beneficial. So ultimately, we need to sensitize onto the water conservation management and improving the water productivity. In this relevance, already the presentations have been made by most of the doctorate students and the master students, maybe in the engineering, maybe in the agriculture sector, or maybe into the my mind the plant physiology, effects of vitamin C and azotobacter type. So this is permutation combinations of the soil microbiology or the other aspects. So with this, I can say on the whole, we need to have the sensitize the state government also the recommendations we can go of this to the areas from where we have the presentations at the central level also we can have the more sensitization part to the government really onto the water conservation management, improving water productivity. So this is a area of concern on which in future the people have predicted wars over water. So we have to see how we can economically utilize per unit of grain which we are getting per unit of water. So already the stressed ecosystems with respect to the water usage in the state of Punjab and in the other parts of the country also, we are feeling a real problem of this water resource and its management in the country vis-a-vis in the states of the India. So thank you very much all for inviting me to have asked me to chair the session on the water conservation management and improving water productivity. The suggestions of this session will go more in improving further the study part and refining their more technological aspects or refining their more the future aspects of research domain to continue with this kind of work more on an elaborative basis. Thank you one and all for joining with me. Thank you, Dr. Kori and Dr. Arora and other organizers of the conference for this.
Thank you very much, sir. I take this opportunity to thank the convener, co-conveners, along with the chair and co-chair who very effectively managed this session. Although we started a bit late, but sir, with your leadership, we have been able to complete it in the time allotted, but obviously we have crossed this time because we just started late. And all our speakers for the next session are in the waiting. So there's not much to say. Wishing all of you a very warm good morning and a happy World Soil Day. And thank you very much. I request the conveners and co-conveners to please share their notes for development of the proceedings through email. Thank you very much.
Thank you, sir. Thank you very much.
Yes. I would just like to invite the chair, co-chair, and the speakers of the subsequent session. This session is the plenary session with World Soil Day thematic lectures by Dr. Dilip Kumar Swain, a professor from IIT Kharagpur, and Dr. Upendra Kumar, who is scientist with ICR NRRI Cuttack. So I request Dr. M.J. Singh to chair this session.
Thank you, Dr. Sir.
Yeah, welcome, sir. Good morning. Happy World Soil Day. Dr. N.K. Parekh, sir, you are requested to please be the co-chair. The conveners for this session are Dr. Bishamdev Prasad and Mr. Rajiv Padbhushan, and the co-conveners are Dr. Kasturika Sen Beura and Dr. Rajkishore Kumar. So please take good notes for developing the proceedings. Thank you very much, sir. Invite Dr. Swain and Dr. Upendra Kumar to also please join. They are already there. And I hand over the mic now to Dr. M.J. Singh, sir. Please, you can unmute yourself and carry on with the proceedings.
Thank you, Dr. Anshuman Kohli. We are going to start a very important this session. I am audible, first of all. Am I audible?
Yes, sir, you are audible, sir. Perfectly audible.
Okay, thank you. So we are going to start a very important World Soil Day thematic lectures. I welcome my co-chairman, Dr. N.K. Parekh, conveners of this session, Dr. Vishnu Deo Prasad, Mr. Rajiv, co-conveners, Dr. Kasturika Sen Beura, Dr. Raj Kishore Kumar. So we have two speakers, Dr. Dilip Kumar Swain, who is the professor of agronomy at IIT Kharagpur. Then Dr. Upendra Kumar, who is ICR National Rice Research Institute. He is microbiologist there, soil scientist there. So we have total 1.5 hour, and there are 2 lectures, very important lectures related to our World Soil Day celebrations. So first of all, I request Dr. Dilip Kumar Swain to start his presentation on climate change adaptations for food security. So Dr. Dilip Kumar Swain, he is available. Okay,
Yes,
sir.
yes, sir.
So you may share your presentation and start your this presentation. Sir, how much time it will take approximately?
Yeah, maybe 40 minutes, fine.
Okay. Thank you. You may start now, sir.
So, is my slide visible? Hello?
Yeah, your slide is visible, sir.
Okay, thank you.
If you can do it on the
Yes,
full.
so good morning, all. So, thank you, the organizers, for inviting for these presentations. My topic is climate change adaptations for food security. You know, this is one of the burning issues and emerging issues as we are facing the rising temperature, and it's affecting the food and productions and other sectors as well. Not the climate change, the climate variability is the main threat for the agricultural productions. You know, you can find in many places the rainfall may remain same, especially the rainfall variability that causes a serious problem for the agricultural production. Either we have the flood or you may have the drought. So in IIT Kharagpur, that only IIT at Mother IIT can say, we have the agricultural and food engineering department, and in this also we have as agricultural science, we have agronomy, soil science, and also we have the bioscience divisions in our department. So you have the farm for the experiment, and also we will be involved in the outreach activities. We have the farmers field demonstrations for the technology we have. We need to take to farmers field for their for adaptation purposes. So with this content, introduction to climate change and the global food security, climate change impact analysis on agriculture, then ensemble of experiment and decision support system, agricultural production technologies, climate change adaptations, mitigation. Finally, I can go for the conclusions. See the climate change, you can see the rising temperature over the years. So there is a temperature expected to increase by 1.1 to 6.4 degrees Celsius by 2130 based on the range of emission scenarios. So you have the different scenarios. Now you have the RCP also. The moderate growth is now A1B, that's current RCP 4.5. So where we have the sustainable development, not the low growth or not the very high growth RCP 8.5, we are looking for the RCP 4.5 as moderate growth. And with that, with sustainable development, in effect, from year 2000, the carbon dioxide will increase by 45% to 110% by 2030, with the warming around 0.2 degrees Celsius per decade. And also increasing number of heavy precipitation events along with prevalence of droughts. So what I can see, so as per the RCM, the temperature with as compared to 1961 to 90 by 2020, we have almost faced the rising temperature around 0.8 degrees Celsius. And it's expected to increase around close to 3 degree by 2050, 2 to 3 degree. So that becomes highly alarming. But as per the IPCC, we want to restrict the rising temperature to 1.5 degrees Celsius, and it's not impossible. So that can be possible by having the sustainable technology, like agriculture also one of a victim of climate change. At the same time, agriculture is also cause of climate change. So you have the climate resilient technology, so that we can minimize the emission of greenhouse gases to atmosphere at the same time. So we can have the adaptive technology how to minimize the adverse effect of climate change on food and production. So what is climate change and food security? So food security has the 4 major components. One is as a food availability through production and trade, then stability of food supply, access to food and food utilizations. That means no, we need the the adequate productions so that it can be available. And every person should have the proper access. There should be economic well-being to purchase the foods, and finally actual food utilizations. Actual food utilization means the bioavailability of nutrients in human body. Not only you have to, that means it indicates we have to take balanced diet, not only cereals or the carbohydrate or proteins. We need to have the carbohydrate, proteins, minerals, and vitamins as a balanced nutrition. That indicates the food security, not only the carbohydrate or not only the protein. So projections of undernourishment depends on climate impacts and also on economic development, technical conditions, and population growth. What we see at the beginning of the millennium, between 800 and 900 million people were at a risk of hunger, and most of them lived in Asia and sub-Saharan Africa. So what is there? The economic growth and slowing population growth can significantly reduce the number of people at risk of hunger. But in a pessimistic scenario, strong global warming, high population growth, and no carbon dioxide fertilization effects, the number of additional people at risk of hunger may be as high as 500 to 600 million by 2080. So that becomes a serious concern for us because we have to keep on going, keep on have technology to improve the food grain productions. At the same time, we have that is the balanced diet. So every children, every family should have the proper foods, not only the carbohydrate, proper the nutritional security that is more important. So the healthy diet is not all. So for the daily diet, you should have the more of the cereal grains. So if you see the triangle, the daily exercise and the weight control, that keeps a human being healthy. So nowadays we are facing a lot of the problem because of disease, so several diseases like cancer, diabetes, or the cardiovascular diseases. That is because the food we are taking. So we are what we eat. If you take a good food, the food controls the health is mostly regulated by the food we intake. Besides other factors, the work pressure, tension, so the different things are there. So how can you have a regulated food? So food that we should have the total fat should not exceed 30% total energy. Take a less of the saturated fats. Also trans fats should be as minimum as possible, less than 1%, and shift in the consumption from saturated fat to unsaturated fats. Limited intake of free sugars, salts, and the diet should include mainly fruits, vegetables, legumes, nuts, whole grains. So, so as you see, if you go to top of the triangle, so you need the fruits like whole grains, the fruits, vegetables as a fresh products. And if you go to top of triangles, we need to minimize the use of red meats or processed foods to be as minimum as possible for a better health, for a healthy human being, for a healthy child. We should take minimum amount of the processed food or the red meats. So the positive impact of climate change on food production, we can find that in case of tropical and subtropical countries, any rise in temperature is likely to affect the yield adversely. Even 1 degree in temperature, the yield is going to decline. But in case of the temperate climates, especially, there may be the rising yield if rising temperature only up to 1.5 or maximum 2 degrees Celsius. But the yield of those countries will be severely affected if the rising temperature is more than 2 degrees Celsius. Similarly, there is a water because there is a sea level rise that that causes the salinization of the many coastal lands and the extensive damage to coral reefs, rising number of the species extinctions, and large scale shift in the climate systems. As you see, in case of the climate change, what is climate variabilities, we are getting the more number of high intensity precipitation events. Though there is that becomes dangerous for the agricultural productions because the distribution of rainfall is not uniform throughout the growing season. So you see, because of climate change, global food productions, as you are discussing, many of the tropical, you see the increase in the CO2 concentration by 2 times. It has seen that the yield of many countries is likely to decline in the world. But there are few places, temperate climates, their yield may be increasing. But most of the countries will suffering due to climate change or rising temperature. The global hunger index, if you see, the positions around very serious because among the 107 countries that have participated in Global Hunger Index, India ranks 94. So we need to take the major steps as a technological development. We have to increase the productions. At the same time, the food should be accessible to every and each human, every person in the country. So see the 1 billion population by 1930. So there is a rising population, and the population will touch around more than 9 billion by 2050. And we have to feed the rising populations. And there are several challenges before the farmers. The input availability is a challenge, like farmers should get the good quality of seeds or the inputs on time. That should be available, which is a constraint for the farmers. Climate change, as we discussed, the shrinking farmland is getting converted to other purposes. Soil erosions and land degradation because of the climate change. Pest and disease is a serious problem for the farmers. Drought and the floods, there is also a problem. And the farmers are most under debt, loan, and mortgage. Many farmers, they do commit suicide because of the not able to repay the loans. Then because of the climate change, unpredictable rainfalls, unnatural calamities, flood, and the drought, that causes serious problem for the agricultural productions. So, what is the positive impact of climate change of high CO2? Because of rising CO2, there is some benefits. The benefits are there is a increasing photosynthesis because of CO2, increasing longer growing seasons in areas like temperate climates where we are expecting to have the higher yield. There may have the longer growing seasons, and the higher yield may be expected on the temperate climates, but increasing precipitation also in some semi locations. But there are several drawbacks of the high CO2, that the drought and floods, that is very frequent, heat stress, and because of as many pest and disease. And in case of tropical climate, there may be shortening of growing periods that causes yield reductions due to the low growing durations as well as high temperatures and increasing flooding and salinization coastal zone that becomes threat. And as a phase experiment, you can see as a PAR carbon dioxide emission experiment. For every 75 ppm increase in carbon dioxide concentration, rice yield increases around 0.5 ton per hectare. At the same time, for every 1 degree Celsius increase in temperature, yield is reduced by 0.6 ton per hectare. So the positive effect of high CO2 that is nullified with the rising temperature. So ultimately, the climate change, the rising temperature, the high CO2 is likely to affect the food grain production of the country. So similarly, I discussed the pest and disease. So there is an increase in pest, disease, and weed population due to high CO2 temperature. And soil quality. What will happen to soil quality? Soil quality, if you say, organic matter, organic carbon is one of the important parameters of the soil quality. And there are two schools of thoughts how the high CO2 or will influence the soil quality, soil organic carbon content. The first school of thought says that because of the rising CO2 and the rainfall variability, because of high intensity precipitations, there is soil erosions. Because of soil erosions, the topsoils are mostly fertile and it may degrade the soil quality. There may be loss of soil carbon. But second school of thought says if the high CO2, there is higher biomass accumulations, higher yield, and the better root proliferations, root growth, it may add the organic carbon to soil. So it depends upon how the carbon changes. It depends upon the location specific and the management practices, how we can minimize the effect of rainfall variability or the high CO2 temperature on soil quality. So if you see this, the effect, how we can tackle this one using the experiment and decision support systems. Suppose if you see the rice crop, as per the crop productions, we need the four major components. One is the crop itself, crop genotype, and the environment, the atmosphere, the soil itself, and the management. How do we manage the crop? In case of rice crop, if you see, suppose you have to apply fertilizers. Rice is grown under standing water. At which zone we should apply fertilizer? Whether it is either flood water, or whether it is a top few centimeters of oxidized zones, or the reduced layers. Usually, we do apply in case of rice fertilizer in case of reduced layers. In the reduced layers, so there will be no urea converted to ammonium that remains in ammonium solution. Because of lack of oxygens, the ammonium remains as ammonium, and ammonium is a form of nutrient that is also taken up by rice. But if you apply other layers, you can see either there are the flood water, there will be more of runoff loss. If you apply oxidized zones, urea gets converted to ammonium because of oxygens, gets nitrate. Nitrate because the leaching comes to the bottom layers, or diffusion gradient that comes to bottom layers. And because bottom layer, there is no oxygens, it gets lost to atmosphere as a denitrification loss. And that happens because of that conditions. Rice contributes also the global warming, climate change by emission of nitrous oxides, yield management, and poor management of fertilizers. So as you do an experiment, if you apply fertilizer at the reduced layers, you get the higher efficiency. Similarly, take care of the crop modeling simulations where these informations are taken into account, develop a model which can give, we can replicate, we can mimic the experimental behavior to have a better simulations. In view of this is a crop model. What we do, taking at this experimental outcomes, we go for the modeling exercise, the decision support system, the knowledge in engineering and domain relationship. The people from the domain area, like scientists or engineers from the agricultural sides, they took joint hands with knowledge engineering, means the person from computer science or mathematicians. So after their integrations, we develop a crop model. And this process is a time taking. It may take longer periods, maybe 5 years, 10 years. And we have to do the several experiments, control experiments for developing a crop model. Once we have a model available, then we go for the simulation. Simulation nothing but solving the model. That's what if you say that y is equal to a plus bx, you are developing models, but the process to how to make y is equal to a plus bx by making the programming, logic, flowchart, that brings simulations. And simulation is not a time taking. It's a very easy process. Once you have developed models and once you put the x value, you can get the value of y. And if you x and b, you can get the value of y within no time. That means the simulation is we can get in fraction in a second only. So through simulations, you can get information, data, knowledge. The time-taking process is the model development. Simulation is a ready-made process. Once you have the model ready, you can get information, you can get data, you can get the knowledge. But for that, these all models, you have to apply for particular conditions. You have to go for the well calibrations and validation of the model for local conditions. Model is built for the generalized conditions. When you apply for local conditions, we need to go for the calibration, testing of the model parameters. Then you can go for the simulations where you can gain information, data, and knowledge. So once you have the information has to percolate to the end users. The end users, it may be the decision makers, policy makers, and maybe farmers who are beneficiary. That is what is a decision-making systems. So this one is the decision-making system. The information systems is a scientific goal where scientists, engineers, they are involved to develop a model. Once you have the model, you get the output, then you get this data should be populated to the end users. That is what decision-making process that comes in model. But where we are lacking, so this is the simulation output we are getting when the population to the end users, maybe farmers or policymakers, there is a huge gap. So yeah, there is a need. The technologists, the engineers, the scientists, they have to work how the information that generated from the laboratory or from your experiment that can well percolate to the end users for whom the informations have been generated. That is what challenges for the farmers and scientists and the engineers. So in view of this, as you're looking for the climate change and the development, as not at it separately, you have to go for the integration of climate change and development. That is a sustainable development, climate change and sustainable development. If you see the sustainable agriculture means what is sustainable agriculture? It's known to all. So there are 5 indicators of sustainable agriculture. So those are the water. That means earlier I have seen some presentations. You have to minimize the use of the water for agricultural production. I see in the future climate, water availability is going to be limited. But agriculture consumes large amount of fresh water, but not going to be available in future periods. So you have to minimize the use of water by having some limited irrigations like micro irrigations. And soil health, as you discussed, soil health because of rainfall variability, intensity of rainfalls, that is soil erosions. So we have to minimize soil erosions by taking some conservation practices because you have to protect the soil health, having the soil conservation practices. Land conversion, so you have to stop deforestation, and so that you have to go for the afforestation to minimize the effect of climate change. So land conversion must be changed from other sources to agricultural lands. Pollutions like application of fertilizers, application of pesticides. So that must be checked. We should go for slowly convert from the chemical farming towards the integrated nutrient management, like organic farming. Not possible completely organic. We have to go for integrations. We have to minimize use of chemical fertilizers and chemical pesticides. They harm not only soil, but also environment, water quality, everything are integrated. As you see, many people are suffering from the cancer disease, that because of food we are taking, the food are the rich in the residual pesticides, high amount of pesticides. The water we are drinking, that is also contaminated due to high use of fertilizers and pesticides. And finally, climate change, because agriculture also contributes to climate change. You have to minimize the use of chemical fertilizers, and you have to show the technique how you can apply chemical fertilizers so that you can minimize the emission of greenhouse gases to the atmosphere. So there are some approaches: crop selections, crop diversification as a climate change adaptation mitigations, or mechanizations. The mechanism, because why mechanism? Because to have better adaptations, mechanisms can ensure that the better application of the fertilizer at proper zone to minimize the emission of greenhouse gases to atmosphere. At the same time, the operation can be completed within a certain time so that we can adapt the climate change. Water management, as we discussed, also nutrient management, weather-based agri information. Farmers need information as per daily weather-based information that help farmers in the regular operations, and it's highly essential for the capacity building of our farmers. So these are some variety as a genetic as a breeding technology. We can bring some variety. We can sustain under the flood conditions and like sub-1, sub-1. The variety we can sustain under drought conditions like stake dentite that is introduced for the sorghum, and also crop diversification. That is also important for now because you have to grow for the different crops, not only as a adaptive to climate change, but also to minimize the pest and disease pressure. And there are some information we have generated from our experiment. There are the rice cultivar, the effect of high CO2 and temperature on rice cultivar. We have seen IR30 Swarna, Swarna Sab1, Basabog. These three are the high yielding variety, IR30 Swarna, Swarna Sab1, and Basabog is centered rice. You can see these three cultivars, they are adversely affected because the yield is declined with the rising carbon dioxide temperature in case of the controlled environmental experiment and simulation analysis. Whereas the Basabog, that responds better. If there is a high CO2 temperature, Bassa variety that gives a higher yield. And what we have seen through genetic level expression, there is a sucrose transporter gene that was SUT1 gene. Some research they have worked, and we have found that this gene expression is there in case of Bassa under high CO2 conditions, but that gene expression is very poor in case of high yielding variety when the variety grown under high CO2 and temperature. And we have seen some hybrid rice, but hybrid rice we have we did through simulation analysis. We have seen the weather parameters that are very significant at different stages for hybrid rice. So some for like Tmax, Tmin from during the active learning panicle system, and T minimum temperature, T minimum during panicle season to heading, and the like solar radiation from heading to maturity. These are the parameters they influence significantly the final yield. But what I've seen, these parameters are the Tmax, Tmin. The threshold levels are slightly higher as compared to the normal high yielding variety. And we have seen the rice yield is decreasing with high CO2, most of the high yielding variety. But we have seen wheat yield is increasing with the high CO2 because we have in controlled experiment as compared to ambient conditions with rising CO2, there is an increasing yield in case of the wheat crops. But that's a small increase is noticed with the high CO2 and temperature in case of the wheat. But the soil condition, soil fertility with the elevated CO2, the available nitrogen, both ammonium and nitrate nitrogen, that declined with high CO2 environment. But interestingly, the phosphorus content is a reverse trend. With the high CO2, there is an increase in phosphorus content in soil. And that may be because of the high CO2 conditions and the carbon there is a higher population of the microbial activity under high CO2 environment that probably contributed to higher phosphorus ability under high CO2 and temperature environment. And we go for nitrogen management. So usually you should look for the site-specific nitrogen management. When you go for the top dressing, we need to apply the fertilizer based on the need based of the crops, like a green seeker that gives NDVI value. Through NDVI value, we can know the status of the crop based on the soil health. Accordingly, you can go for the fertilizer management. So that gives the NDVI different values, but we to consider as a same N content for all cases. This need to be taken care having the the soil of low, medium, and high fertility. Based on that, best NDVI value, we can go for the nutrient recommendation. And similarly, for the dryland farming, or where is the rainfall variability, go for the water harvesting structure that should be created in the farm pond. So the water can be stored, which can utilize for live stream irrigations for the same crops, and for the irrigation for the subsequent crops to this storing water. And the drip irrigation, that's for rice. We use the drip irrigation for the rice crop. What you have seen, we could save around 40% water in case of the drip irrigations, and yield penalty was only 10%. There is a 10% yield reduction in case of the drip irrigated rice as compared to conventional water transplanted rice, but we could save huge amount of water in case of the drip rice. In case of the climate change adaptations, if you go for the drip irrigations, that will help to save the rice crop. And there is suggestions from the students of our institute, because can you have soil analyzer machine like ATM model? As we go in ATM model, similar model, can you have a SAM machine, that's a soil analyzer machine in a village panchayat, where the villager can insert the soil samples, and through spectral signature that will analyze the soil, the quality can be analyzed, and the farmer can give the information about the mobile number and the locations of the place, and we can get the reports like the soil fertility, soil pH, and soil moisture content can be determined, and it can be sent to farmers through his mobile phone. And then, based upon that information, the farmer also can be given suggestion about the type of crop he has to grow and the management practices he has to follow using this. These suggestions can given preferred crop to be planted, soil nourishment plan, and irrigation schedule can be given. So the other one, climate change adaptation, you can see the vertical farming, that is also become very popular because close to cities, in city area or very close to city, you can have the vertical farming systems where especially the high value vegetables can be grown without soil, like soilless culture, hydroponic systems. So we are successful growing lettuce has come very well in case of hydroponic systems. So this type of system, because many developed countries, they have the vertical farming, and because having the vertical farming close to cities, we can minimize the transport of vegetables from the far places from village area to cities. In the transportation also, the the food quality gets spoiled, and also there is a emission of pollutions of the atmosphere. To minimize that one, you can have like vertical farming very close to city that can meet at least partially the food security of the people living in cities. And the agriculture, as you say, the emission of greenhouse gases from different sectors, agriculture around 14%, and among the agriculture sectors, the soil emissions and the entry fermentation is the maximum contributing to greenhouse gases. So you can reduce the emission of greenhouse gases from agriculture sector by including the leguminous crops like nitrogen fixing crops. They can so that you can minimize the use of the fertilizers and also avoid the flooding. So you can see by having the organic farming, what you have seen, you can minimize the emission of greenhouse gases to the atmosphere. From our experiment, as you can see, by having integrated nutrient management, the yield loss is only 4%. If you apply the vermicompost like chemical fertilizers, yield loss only 4%. But at the same time, we can reduce the global warming potential close to 29%. What I have seen from our experiment, the amount of carbon dioxide released per hectare around close to 3,000 from different average from different nutrient management, and from the rice cultivations around 500 gram of carbon dioxide released for each kg of rice we do produce. And another aspect of home organic farming, this is organic farming concept. As you do the home as Agnihotra, we can say, that can be done in a large area, around 10 kilometers dia area. In a cottage, we can have a pyramid copper pot. Inside the copper pot, we can have the raw rice and dried cow dung cake and a ghee, the desi cow ghee that can be put, and you can put fire. And its slogan is Sanskrit slogan is there. We can chant the slogan, and that should be done the sunrise and sunset, the particular time. If you done in a particular farm, and the fumes comes from cow dung cake, the dried cow dung cake, raw rice, and the ghee. That fumes does spread around the crop field, and there is also because of the fume, so spread over the crop field, that increases the solar radiation interception capacity by the crops. At the same time, crop is protected from many pest and is no pest, and less or no pest and is attack. So in that way, that can increase the productions as per this Homa organic farming. This concept came from this, I can say this is a Indian traditions we do, but now the German scientists, they are claiming that patenting this one as a Homa farming. And now German scientists, they are coming to India and they are trying to propagate and expand for this idea of Homa organic farming. And also you have to go for the changing consumer diets. If you see, you can change, being a vegetarians or non-vegetarians. Usually the being a non-vegetarians, we are the more emitter of the greenhouse gases as compared to being a vegetarians. So highest greenhouse gas emission is the beef productions, around 10 kg for CO2 released per kg of meat production in case of beef. And so poultry around 2 to 3 kg carbon dioxide released per kg productions. Milk around 1 kg CO2. And in case of plant-based products like vegetables or the food products, plant-based products, only release of 500 gram of carbon dioxide per kg of the products. So that's what changing consumer diet somehow it may decrease the emission of greenhouse gases to the atmosphere. So most of the cases, organic farming is superior to the others, climate change adaptations and the water storage. Only one case the yield, but yield can be maintained if you are using the proper technology for the organic farming. So these are some of the practices we have developed in our farmers field. We have implemented. So we do water technology. We go for the farmers field very close to our institute. We have around 15 acres land. Most of the technologies are implemented in that farmers field. And also we have introduced the mechanical planting and the soil water maintenance systems in the field itself. And there are some practices. So with this, we also have the lab classes we do conduct in the demonstration site in the farmers field. So with this, I conclude that understanding the nature and degree of vulnerability is essential to build location-specific adaptation strategies to climate change. With understanding the ground level adoption of the adaptation is very important. Adoption of modern technology like decision support system, information communication technologies, drones, etc. provide the many useful data and analytical solutions for developing scalable and actionable technologies and evaluate the path we are targeting to improve livelihood of farmers and food security of the nations. Besides adaptations, agricultural production management offers a scope for reducing greenhouse gas emissions as climate change mitigations. Organic farming is the way forward to minimize the greenhouse gas emissions and decrease the global warming potential of agricultural productions with quality food products. With this, I thank you all and I'm looking for this. Thank you.
Thank you very much, Dr. Saab. You have given a very good presentation, right from the climate change, how it is affecting our earth, and right to the farmers field from global level. That's very important. And thank you also for completing your presentation even before time. So we have about 10 minutes for some questions. I will request the participants, anybody who want to ask questions or want to some discussion or some clarification, we have about 10 minutes for that.
Hello.
Yes.
Hello.
Yes. Please, whoever is asking, please give his name first.
I'm Dr. D.K. Chaudhary from RRS Agwanpur, Saharsa.
Okay, thank you. You may ask, sir.
Sir, thank you for covering and encompassing every aspect of the agricultural problem, which is climate change, organic farming, water management, soil erosion, soil management for food security. You don't think that our 85% farmers are marginal farmers or a small farmer rather, and for whom how we can address all these problems for securing better production system in this small holdings area? What is your view? And another point, in my view, it is not faulty developmental plan that aid in climate change, that aid in erosion, that aid in taking away the fertile agricultural land, and that is shrinking our holdings. Thank you, sir.
Yeah, good question. You see, we all our farmers' conditions, the small farmer, marginal farmers, and especially when you look for the increasing productions in a small and small holdings, we need to think of the as a community farming, which is not there, and there is to sensitize the farmers in a group. Not only is a small farmer, okay, there is a problem of using machine. Our engineers are trying to develop machines which can fit the small farming But however, the small farmer cannot have the access to buy the machines individually. So mechanization is one of the aspects which to be looked into. The future agricultural production depends upon mechanizations. If you are not going for mechanizations, we will be lagging behind. And our mechanization must feed to the farmers. So to do that one, for the better mechanizations, we need to have the group of farmers, the farmers group, and there should be a custom hiring systems because small farmers are not accessible, cannot buy. This is not worthy of having a machine for a small farmer of 1 hectare less than 1 hectare land. So custom hiring and the maintenance of the custom hiring systems, and you can say every clinics, if you have every clinics at every block levels to solve the farmer's problem. And they should be well equipped, well sensitized, and well knowledgeable to meet the farmers and to have all access to the farmers, especially the seed quality. Now farmers are facing, we are not getting a proper seed, good quality seeds, and on time also. That should be available. And that can be, if you can, the farmers, what are farmers, they can be good quality seed and the implements. The implements for the operation. Now labors are very expensive. Farmers cannot use labors for their farm operations. That becomes very expensive and the output becomes very low. And integrated concept, this is one thing what policy level decision has to be taken. The other thing what is say, let's say earlier you had the integrated farming systems. Every farmer used to have a pond, used to have, as a cow. Now cows are vanished because we have no cows. It's good that we are going for, cultivation using the machine, but at the same time, cows should be there because cow dung, the cow urine, they are valuable materials for the agricultural purpose. Every farmer must have cow. He should have cow because they meet the milk requirement of the family. At the same time, the cow dung is a beautiful resource for manures, for the composting or the vermicomposting. We should try to use more and more organic sources and to minimize the use of chemical fertilizers. And of course, I do agree. At the same time, you should have technology so that you have the biofertilizer, organic fertilizers that should give same level of productions as of conventional chemical fertilizers. There are approaches. It's possible. I can say not impossible. It's possible to have a biofertilizer, organic manure that can provide same amount of nutrients as the crop needs and can protect the crop. So it needs a integration aspect. It's like sensitizing our farmers, community-based farming, and decision-making processes, and having the cows and integrated farming systems. You should have the ponds. Some ponds should be there, and crop diversification also. That is essential. I cannot say if you can go for the more number of crops. I am from Eastern India. You can see the farmers of Eastern India, they don't want to replace rice crop. Okay, whatever in the cases, they want to grow only rice, but rice is not a profitable crop. Suppose I'm a family with 5 members, I should grow rice for my family, not for sale. I'm not getting profit. If I have to increase my income, the rest area of rice should be diverted for some other high value crops so that I can increase income generation of my family. That's what my point.
I understand there are many issues, particularly for the marginal farmers. It's very important so that our technologies are adapted at mass scale. So thank you. Any other question?
Yes, sir. Good morning, sir. Sir, good afternoon. Myself, Kalyani Divedi, sir.
Yes, please.
Sir, in your presentation, I saw a word which is integrated CCSD strategy. Sir, I'm a fresh student in MSc Agriculture, so I have no idea about that strategy. I just want some idea about it.
Yeah, that's for climate change and sustainable development.
Sir, can you repeat, sir?
Climate change and sustainable development.
Okay, thank you, sir.
This is integrated climate change, sustainable development. I discussed. We have to go for the input management, like less input and more, so resource use efficiency. How we can increase the resource use efficiency by minimizing the use of water, by minimizing the use of nitrogen, by the proper application technique, and as per the need-based applications. By the way, you can minimize use of inputs, less emission of greenhouse gas to atmosphere, less climate change.
So any other?
Thank you, sir.
Any other question?
Sir,
Yes.
just tell me about Homa organic farming.
Yeah, Homa organic farming, yeah, like you do as a traditional home. If you see, you should do any puja in home. We do Agnihotra Homa, and because of this, because the occasion we do follow doing Homa. So we do also in their Homa. You put dried cow dung is most dried cow dung. We do a chandan cut with food, and the fume does come. No, when you make Homa in your home itself, it purifies your home, because people in the home, they are protected from many the outside diseases, and also that once you inhale the home, that fume, that purifies the whole body systems. And similar concept, if you do a homo in a farm area, open farm area, and that is your particular time, the sunrise and sunset particular time is there, and the fumes comes out, and fumes, because ingredients are, it should be copper pot, pyramid copper pot, particular size, and ingredients are the dried cow dung cake, the raw rice, and the cow ghee. These are three ingredients. You put the burn it. Cow dung is a tremendous source of energy, because you are using cow dung for every purpose here, and in the ghee you are putting cow ghee, and the fume comes, that fume spread over the crop field. It protects the crop from any pest and diseases. At the same time, there is a scientific evidence to say that it increases the radiation interception capacity of the crops. That way it can increase the productions as well as it's just one type of organic farming, less use of pesticides or no use of pesticides.
Thank you, Dr. Sir. I
Thank
understand.
you, sir.
I understand there are different type of natural farming systems, and it is one of them. We may put some question mark here until it is we are able to prove, and there is more scientific studies are there. So in the meantime, we can go ahead with the different type of experiments. We are the scientists. So next question, please.
Sir, I'm Vishwandev Prasad, working as assistant professor in Bihar Agriculture University, Sabha. Sir, very nice presentation. I have a small question. You said that if there is an increase in carbon dioxide concentration, rice yield is decreasing. So, but rice is C3 plant. If there is an increase in carbon dioxide concentration, yield should increase. That's why, like, people are trying to convert this C3 to C4.
Yeah.
So,
yeah, from my presentation to see, I don't say with CO2 the rice yield decreases. With high CO2 there is an increase in rice yield. But what happens with the CO2, there is a rise in temperature. Because of high temperature, that causes the yield reduction, not carbon dioxide. And what we have seen, because of high CO2 and temperature, there is a particular gene that's called sucrose transporter gene, OS-SUT. That gene expression becomes very poor in case of the high yielding variety. But this doesn't happen for the local variety like Basavo. It is not affected with the high CO2 and temperature.
Okay, thank you, sir. Thank you very much.
Thank you very much, Dr. Dilip. We cannot take more questions now. So thank you for your presentation also. You may please unshare your presentation.
Thank you very much.
Now I request Dr. Upendra Kumar, who is a scientist, soil science at Indian National Rice Research Institute. Dr. Upendra Kumar, he is available. Dr. Upendra.
Yeah. Yes, sir. I am available.
Dr. Upendra,
thank you very much.
I request you, how much time you will take, sir?
40 minutes.
It will be better if you complete within 30 minutes so that we have some time for discussion also.
Okay, I will try.
Okay, thank you. You may start your presentation.
So is it visible, sir?
Yeah, it's visible, yes.
And I am clearly audible?
Yes, you are audible also, yes.
Yeah. So good afternoon, one and all. At the outset, I am thankful to the Soil Conservation Society who is given an opportunity to present on the occasion of Soil Health Day today. Our previous speaker, Dr. D.K. Swine, sir, he has presented the effect of climate change and what is its role on food security. Here, I am going to emphasize the role of microbial diversity in rice-based system and its influence of crop and soil health. We already know the uniformity is not a nature way. It is a diversity. That's why this diversity is actually building the civilization based on the biological resources. That's why in 2010, International Biodiversity Year of Biodiversity has been celebrated. Louis Pasteur, father of microbiology, once said that the microbe will have the last word. And we know already we have 3 million species of bacteria, 1 million species of algae, and 1.5 million of fungi. That's why we are celebrating today World Soil Day. And the theme is keep soil alive, protect our soil biodiversity. Rachel Carson, you know, she wrote the book Silent Spring. And why she wrote? Because she has shown the extinct of bald eagle, which is due to DDT, one kind of pesticide. Once the application of DDT is there, then what happened? The biomagnification exists in the bald eagle and the premature hatching of the egg is there. So DDT was going to ban in USA and now all over the world. And she wrote the book Silent Spring. So she wrote, in nature, nothing exists alone. See the diversity. So all species are interlinked to each other. In nature, the circular frame is there. If Yugo is there, so this one is become triangular and it is going to be altered. See, in green revolutions, we are earlier in begging ball, but since 1960 and 70s. So what happened? The green revolution came in picture in India, but with the cost of environmental pollutions, and that is due to application of chemical fertilizers and pesticides. And these are actually necessary evil because just like a transpiration, okay, this application of pesticide and chemical fertilizer is necessary, but unnecessary evil to the non-target microbes which is present in soil, and that maintains the sustainability of the soil. But this application of a fertilizer and pesticide having three kinds of effect, either positive or negative or neutral. Negative means some toxic effect either. Positive means the microbes also utilize some carbon sources. Neutral means they will not take any kind of carbon sources from the applications. Even in India, the fertilizer applications and pesticide applications are skewed. Somewhere more application are there, somewhere less. As for example, if you see 90 to 90-20, and then we only concentrate the fertilizer in consumptions. You know, in 1961, once the government of India has introduced urea in the market, the farmer outrightly rejected that one. They said, no, this is the chemical nature and this is actually deteriorate the soil. That time they are aware. But once the government official proved that after application of this urea, actually showing the more yield compared to where they have not applied the urea. So farmer convinced. Now what is the scenario? Farmer over convinced. They are thinking if you apply urea, then their yield will be increased. So what happened in this scenario? 11 times increase of fertilizer in consumption is there, but there is limitation of the crop, particularly for rice and other crops. Their uptake of only 3 times. And what happens to the remaining part? They are going to be lost. If you see in rice, the overall nitrogen use efficiency is 35%. If maximum also is there, then 50%. So what happened? It is going to alter the soil health. How? You can see the nitrogen cycle here. 3 most important cycle is there: nitrogen fixation, nitrification, and denitrification. These nitrogen fixations occur in soil due to certain kind of microbes, prokaryotes. Other microbes will not do because they don't have nitrogenous enzyme. Even plant and animals have no capability to fixing the atmospheric nitrogen because they lack the nitrogenous enzyme that needs a gene which puts the nitrogenous enzyme. So this is the things. Atmospheric nitrogen, though it is present in 28%, it is unutilized by the plant. So that's why exogenous application of urea or other nitrogen fertilizers are required. So how once the nitrogen is there, this is the complete cycle where nitrogen fixation is there, this is coming to ammonium, then nitrification is there, it is forming nitrate, these two formulas available for the plant, and then again it is converting into nitrogen. So this is the loss process. This is the available form of the nitrogen. If you see molecular aspects, so if you see here atmospheric nitrogen to ammonia, which is only done by the certain kind of microbes, and that they contain the Neff chain enzymes. And this is again nitrifications. And after that, this nitrate is converted into nitrate. So this you can see during nitrifications, the N2O, which is actually causing the global nitrous oxide. This nitrogen fixing bacteria, uh, even farmers are also knowingly or unknowingly, because in legume you can see the urea consumption is very less because they know the potentiality of the rhizobia. But in case of cereal crop like rice, they don't know. But so, because they are not forming the nodule, and then these nodules actually having this nodule is having rhizobia in case of legume, but in rice some certain kind of endophytes are there, which is also harming. So if we harbor these endophytes, then definitely farmers will make the aware. Then farmers must aware about the endophytes, and they will certainly reduce the consumption of nitrogenous fertilizer. Now coming to the effect, because our topic is related to diversity, how it is going to alter. So once the application of nitrogen fertilizer there, everybody is knowing if available nitrogen is there, then nitrogen fixing capability is going to be ceased. So in long-term aromatic rice cultivations, where the application of nitrogen dose is slightly more compared to that one, there what we have seen, the frequency and diversity of nitrogen in rice rhizosphere is going to be reduced, because in rice what happened, certain anaerobic condition is there, slightly microaerobic condition is there, microaerobic condition there, azoospermillum brasilensis. So this high bacteria is not related to this one. Some diversity is there, though it is known that if slightly aerobic condition is there, azoospermillum is abundant. So anyhow, the message of this is there is going to alteration of the microbial community if you apply continuous apply of nitrogen fertilizer in case of aromatic rice cultivations. We have long-term fertilizer experiment, and long-term fertilizer experiment is always a treasure. That one is initiated in 1969. So we did the functional diversity of microbial community and structural diversity as well. What you found that where the continuous application of nitrogen alone is there, their functional microbial community is going to be altered. But if you combine the FYM plus N, then their soil is healthy and their functional microbial community is also healthy. In case of structural diversity, what happened if you continuous application of nitrogen is there, you see here there is alteration of the microbial community, and some of the community is likely to extinct in future. If you see here in this graphic, and that is directly linked with the grain yield and straw biomass. Then other management practices also there, which is actually altering the microbial community. Even urea briquette application is there, that is also related to the greenhouse gas, and their soil enzyme activity, nitrogen loss, and all this is also affected due to continuous application of the nitrogen. Coming to the pesticides. So we have one beautiful experiment, that is long-term pesticide, and long-term pesticide experiment is found in rice is very less, I think. So here we use 2 insecticide chlorpyrifos, kata, 1 fungicide carbendazim, and 1 herbicide pretilachlor. What you found that continuous application of the pesticide, particularly chlorpyrifos, it is also going to alter the nitrogen fixture, nitrifier, denitrifier, like this. But we didn't say long-term effect of that. It's actually altering, but it is slight altering of that one. Even the nematode population is there. That one is, slightly under this chlorpyrifos treatment. You can see these two kind of nematode, which is going to be altered. And the half-life is actually vary. But in this case, the half-life is there only 4 days for that purpose. Then pretilachlor. So here pretilachlor is we have done the experiment. This particular you already know this is the herbicide, and we have done the experiment and we found that the recommended dose we can use, but more than that we can not use, because it is also again altering the soil enzyme activity under tropical soils. Imidacloprid is also widely used insecticide in rice, and it is showing the negative effect of on soil microbe and soil enzymes. Then then here in this experiment, this bisparibak, everybody knows bisparibak sodium, it is a widely used herbicide. Here also we did the experiment and we found that yeah, it is also altering the soil microbial population, enzyme activity, and functional microbial diversity in paddy soil. Our previous speaker already elaborated very nicely, summarized the effect of climate change on food security. Here I am going to emphasize how this microbial community is going to change under elevated carbon dioxide. And this is really, already, if this microbes are diverse. What microbe you are getting, that is 1%, and that is the only media biased. So that's why we must explore the microbial community under this climate change and other management practices. See, long-term exposure of elevated carbon dioxide on herbivore mycorrhiza community. You already know importance of herbivore mycorrhiza. So what happened under aluminum IC platform, the drastic reduction is there, like Cladrioglomys and Glomus species. But this particular species is going to be dominant under elevated carbon dioxide. So this study actually concludes that anticipated increase in elevated carbon dioxide consumption in future may severely alter the structural community of herbivore mycorrhiza in sub-humid tropical paddy soil. Similarly, here also one article published, and there also we emphasize that there is also the interactions effect of native glomerular species. Then recent article published in Agriculture Ecosystem Environment, there the target is there. We have done the functional soil microbes from tropical dryland exposed to elevated carbon dioxide, and there we found that this direct effect of nitrifier and denitrifier population is there on nitrous oxide emissions. One interesting study also we did, combined effect. What happened if three factors is going to be combined? If elevated carbon dioxide, nitrogen fertilizer, and water deficit stress is there, so what happened? The diazeotropic community, nitrogen fixing community. So what we found, this under elevated carbonation and higher dose of nitrogen is going to be subsided. If high dose of nitrogen is there, so there is always a decrease of nitrogen fixing bacteria. But under elevated carbon dioxide, that is going to be normalized. Similar case we found in two species of Azolla. Same thing, effect of higher dose of nitrogen on diazeotropic efficacy of both Azolla species. In Azolla cyanobacteria is there, so that one is going to minimize under influence of elevated carbon dioxide. Even elevated carbon dioxide, the degradation of chloropyrifos is going to be fast. So what happened? This negative influence on persistence, it is less, and the positive effect of soil enzyme activity is there on the chloropyrifos. Then some research options must be there to manage this. So why not? We must think about the microbiome, because we must think about the microbiome. Earlier, the people only focusing on the media-based bacterial culture. Now we must think the whole microbiome, and that is a part of only 1%. But here we must think about the whole. So this is the microbial manipulation we will do, and what is the diversity there we must see. So even drought-tolerant bacteria, thermotolerant bacteria, we have to adapt, and that we will use for the sustainable production. Here, methanotrophs. We know carbon cycle. If ratio of methanotroph and methanogens, methanogen and methanotroph ratio is what if methanotroph is more, then definitely methane emission is going to be less. So here, if proper incorporation is there, manipulation is there, methanotroph, definitely we are going to reduce the methane. And endophytic diazotroph, I explained earlier, because just like a rhizobia, some endophytes are there in rice, and we have to explore the percentage of that one. And the DNRA, disseminated nitrate reduction to ammonia. This is the constructive process. I will explain in later slide. And the another process is there, anamox, anaerobic ammonia oxidation, which is a destructive process. So most export some bacteria, which is actually reconverting nitrate into ammonia, and that we utilize by the plant. So this is the short circuit cycle in biological nitrogen to conserve nitrogen in terrestrial ecosystem, which we published in Science of Total Environment, and there the full review is there and meta-analysis study also there. So DNRA is disability nitrate reduction to ammonia is the novel and shortest biological pathway of recycling in terrestrial ecosystem. Here, actually, nitrate is reduced to ammonia via respiratory or fermenting. This pathway has potential to reduce nitrous oxide emission because it is reconverting nitrate into ammonia. So meta-analysis also revealed that DNRA is mainly regulated CN ratio, and these are the factors. So therefore, DNRA may be treated as a tool to reduce groundwater nitrate pollution, enhance soil health, and improve environmental quality. And this work we have taken, and we are working on this direction. Second thing is, everyone knowing about the azolla and importance of azolla, but it is not popular in farming community because certain limitation is there. Because this is working as a biofertilizer due to presence of certain kind of cyanobacteria. But we did the Illumina My6 sequencing, and we found that there Anabina nostoc is not there. People believe in that, yeah, azolla, if cyanobacteria is there, that is, of course, Anabina is there. And they are also naming also Anabina azolla. But we proved for the first time that no, Cylindrospormopsis is more abundant, and that is also coming under Nostocaceae family. But if you do the cross inoculation of cyanobacteria among azolla species, because in legume, host specificity is there. But in azolla, host specificity is there or not, we have to work. So we have to isolate and then cross inoculation the potential BGA in azolla. So definitely one kind of problem is going to be solved in future. And our team with ERI, even Sabor, we are working on meta-analysis aspect. Because whatever the published data is there, we are going to make a trend to whether it is good or bad. So based on that analysis, we found that integrated nutrient management in rice feed system is important. Because if you continuous application of fertilizer is there, that is not good. We have to make the combination approach. Then coming to biofertilizer. So earlier slides, I already explained that there is, alteration of microbial community if you use fertilizer. And this is the news article came in Science, Indian Science Wire, and down to up based on our publications. Then government is actually really framing the policy to reduce the usage of chemical fertilizer, especially urea, because urea is the poisonous in it is going here and there, just like a siva, and then it is causing the cancer and all those things. And if it is going to mix with the water, then it is causing the blue baby syndrome. But what is the status of biofertilizer in rice or overall? If you see the Indian scenario, biofertilizer production in different regions of India, then eastern states only 2%, only 2 to 4% biofertilizer production is there, and rare rice specific biofertilizer is available. But we must think about biofertilizer production for rice. Why? Rice is the most cultivated area, particularly in eastern states. Legume farmer is knowing, but rice, whether it is there or not, because you must think about that. So we have developed certain kind of product here. So based on the nitrogen requirement, we have developed the Azotobacter procum, and we just not developed this endophytic Azotobacter. We did the extraneous work to do that one. So what we did, first we isolated from the rice root, then we have to see the track, whether it is endophyte or not, and for that fluorescence energy transfer technique we used, and then we have done this tube experiment. In tube experiment we found that yes, the maximum is there, this Azo AVI2, and then the pot experiment done, and then we did the 3 times field experiment, and we found that yes, it is working under field conditions. Then we made the formulation, and the shelf life of the formulation more than 1 year, and then I started experiment with the formulations with certain variety, and then we found that yes, it is going to save 12 to 25% of nitrogen without compromising the yield. So these are the, and we, the team of ERI actually developed the site-specific nutrient manager rice crop manager. But there the consumption of nitrogen is more. So why not we have used the endophytic nitrogen fixed bacteria, that is a kind of integrated nutrient management approach. Azolla you must know, and actually Azolla is also having, different things. We think Azolla is not having characteristic. There is different types of Azolla are there. So we did that one based on shape, imbrication, and all those things. Some biomarker we want to identify it. And we are actually conserving 102 strains of Azolla, which is highest in, I think, ERI Philippines is actually preserving 500, but they are working or not at presently, I don't know. But we are working on Azolla, and what we are doing that one, we are going to isolate the morphological marker, physiological marker, and even the important thing is that sporocarp based, because that is the major problem, because 10 to 15 tons Azolla is required per hectare for the farming. So what we did, we have done the simple formulations and we developed the sporocarp based, because simple, only half kg is enough for the per hectare. So this one is the some glimpses of the picture how this Azolla is going to be germinated, because here also we got new methodology, because germination is also very difficult, particularly for Azolla, because it is this sporocarp production only takes place in few months. In November, now it is going to initiate November to March only. After that, it is not there. That's why the formulation is required, because the rice is cultivated in Kharif season. So that time this is required. This farmer is always approaching. They are asking, okay, Azolla is, because it is going to die, because it is water form. Once they divide of water, it is going to decompose. So that's why this Azolla sporocarp technology definitely give a good impact in future. We are working on that one. Some value added, because Azolla itself only not acting as a biofertilizer. We all know the livestock feed is also there. So we are working on that direction also, because we are going to enhance the shelf life, and we have to give the proper amount, just like a tablet. Because if we are sick, we need certain amount, 50 mg, 500 mg of tablet. So for animal feeding also, we must see the proper to provide known amount of nutrient, and of course the wastage. So one machine also we have developed, and we are going to make the Azolla pellet. And other things we did, because whatever the microbial growth media is there, that is highly costly, and other thing is there, that is animal waste, like beef extract and all those things. So this Azolla is very cheap and very fast. It doubled within 2 days. So we have tried to make a NRI growth media, and we have validated also this fungus and bacteria also, and it is showing the at par result with the whatever the commercial media is there. We compare also that this is a tentative price and it is very cheap. We are also conserving 42 strains of cyanobacteria strains along with the 4 commercial strains, and we are also working on that direction. So this phosphorus also we have some formulations. Drought alleviation also we have formulations. So this one my student already presented in poster sessions, that here very good concept is there. One thing is there, if some stress is there, so oxidative stress, their reactive oxygen species, it is going to make the dye of the tissue. So what happened? Some antioxidant always come in our mind, but testing the antioxidant in plant and animal, it is quite time consuming. So we thought Acetobacter glucocum, that is a fastly growing 2 or 3 days. So we have taken this H2O2 as a oxidative stress, biological component Acetobacter glucocum, and then what we ascorbic acid as a antioxidant. What once H2O2 we have given, that after 2.5 millimolar, so bacteria is going to die. Once we added the antioxidant, 1 ppm only, not only bacteria die, but enhance the survivability. Second thing is biomass increase. Third thing, the functional characteristic of this nitrogen fixation, it is going to be double. So People will think always the mutation approach. Once mutation is there, they are going to enhance. So mutation approach will go, then this is also comes under genetically modified microbes. The people are accepting or not, this is also a question mark. So simple formulation if you do, then definitely the function will be increased. So we did a similar thing in the rice under drought stress, and this is the actually mechanism under moisture stress. A reactive oxygen species is coming. This one is actually after this is actually once AVI2 is there and addition of the ascorbic acid there, it is working in a two-way. One is it is activated once it is oxidized state. Once ascorbic acid you are going, it is forming the dehydroascorbic acid. In this activated state, it is going to the cell and then it is reduced back to the ascorbic acid. So ascorbic acid is having dual function. One is actually it detoxify the effect of reactive oxygen and convert into dehydroascorbic acid, and the another way it is in this oxidized state it is going to the cell and then reduce back to ascorbic acid. So this is a simple formulation we did and we got the very good result, and we have published in plant physiology biochemistry, and this is also under review under second paper also it is there on plant physiology and biochemistry. The most important is their field work. So we are also constructing the 4 biofertilizer units at NRY. One is liquid biofertilizer unit, and that one initiated. Another is mass production of fajula, mass production of blue-green algae, and of course, arbuscular mycorrhiza. So once the production is started, we are going to sell for specific for the rice crop. Similarly, microbial pesticides. We have already filed patent for the 4 BT, 1 Beauveria bassiana and meta-rhizobium for leaf folder. So these are the things, and that is going to be published as a technical bulletin from ICR. It is available soon. And the one more thing, we have reported for the first time Escarmella species. So far, as a biocontrol, Escarmella species has been not reported. So this is the first time we have, and this is also having the similar potential as a BT. The overall take-home message is, if you apply fertilizer or pesticide, then it is definitely altering the soil microbial community. So must think about this and must think about the soil health. In once I went the Adelaide, there we found that people here also, they are rarely focusing on the below ground. They are thinking about yield only, above ground plant. But soil is life. You must think about that. That's why we started work on soil. And this previous night only, we got the this from the agronomy where we have published 2 articles. And lastly, I am really acknowledging my all staff, and then of course Director NRI, Head Crop Production Division, and the Soil Conservation Society of India given opportunity to talk before you. Thank you all.
Thank you very much, Dr. Upendra Kumar, for completing your presentation in exactly 30 minutes. That's really appreciable. And it was very informative. It is very close to the theme of this year's, our theme is Keep Soil Alive, Protect Soil Biodiversity. So you have worked on very deep information you have given. So we can take 1 or 2 simple questions, small questions from our audience. Anybody who want to ask any?
Sir,
Yes.
thank you, sir. Thank you for giving very in-depth analysis of this biodiversity. Sir, my simple question is that at the in 1960s we are using pesticides only 0.1 million tons. Now by the turn of 2020 we are going to use about 30 million tons pesticides as per Government of India's data. So without decreasing the use of these deadly pesticides, how can we expect hope that the biodiversity will increase? Is it possible?
Yeah. It is possible because government is also more concerned about that. That's why they are framing policy. For fertilizer, for instance, you see the fertilizer aspect. Government has recently only you also read the news that the subsidy you will not get until unless you will not purchase the bio. So government is more conscious about it. And pesticide, we just see so much work is there, non-targeting. And definitely microbes are there, if filtering is there, all nutrient cycling, everything is going to be. And pesticide is really a poison. So government is more concerned about that, and they are also framing policy day by day, and they are promoting the microbial pesticide and of course bio.
And also, I would like to add that the type of pesticides, now the green pesticides, which are having a low residual effect or their life is less, these are being promoted. There is change in policies, and hopefully, it will improve our biodiversity. Any other question, please? Any other question? If there is no question, thank you, Dr. Upendra Kumar, for a very nice presentation. Now I will ask my co-chairman, Dr. N.K. Parikh, to give his comments about these presentations. Yeah, Dr. N.K. Parikh.
Thank you, sir. Thank you. I think need not to mention anything because Dr. Kumar have presented nicely about the biofertilizer and their roles and their utility. So nothing from my side. Okay, thank you, sir.
Sir, unmute yourself, sir. Unmute yourself, sir. Sir.
Manmojit Singh ji, unmute please.
Yeah, sorry. Thank you, Dr. N.K. Parikh. If anybody from our convener or co-convener want to give some comments or something like that. Any convener or co-convener? So, thank you. Now I will give some, I would say, concluding remarks on these presentations. These are very, very important presentation related to our this World Soil Day. As I already discussed, our first presentation was regarding the climate change. There are location-specific influences. We cannot generalize at global level that carbon dioxide is increasing, temperature is increasing, but the effect is at localized level. At some places, the effect is positive, somewhere negative, somewhere there is no effect. So we have to see the localized situation. And moreover, for the adaptation measures, he has very nicely presented. Dr. Dilip has given very good presentation how we can adapt to the various situations. For regarding the selection of various crops, these crop varieties and water management and many other issues like hydroponics, which are these things are very common in China and in other many developed countries. These hydroponics, particularly vertical farming, because of lack of land and other issues, it is coming up. He has given much more emphasis on organic farming. We agree that organic farming or our traditional farming in India, it is very resilient to this climate change because we were having integrated farming system was there. We were having different components. It was highly diversified. So the diversified agriculture will definitely survive to the climatic vagaries. In addition to that, we need to give emphasis on this custom hiring center. He has mentioned that also because the mechanization, it's not possible for our small and marginal farmers to purchase each and every machine. So custom hiring centers have to come up, and it will lead to our youth will get the employment opportunities also in that. We can think of that. Then we need to go for some contingency planning also, contingency planning. In case of climate change, particularly in our Indian condition, it's monsoon based, this climate is there. Sometimes there is a late onset of monsoon, sometimes early withdrawal of monsoon, in between dry spells are there. For that, we need to develop various contingent plans. Some contingent plan have already been developed by different centers. Within that contingency plans, we need to go for preparatory as well as real-time contingency plans. For example, some delay in this monsoon is there. Immediately, what the farmer can do, what alternate crops he need to grow, what he can do like that. If early withdrawal of monsoon is there, the farmer is having crop in his field, what he can do. So these type of real type situations, for these real problems, we will have to give the real time contingency measures. We need to strengthen that also. Integrated farming system is very important for climate change adaptation. It's very, we need to give more emphasis on that. In the second lecture about Dr. Upendra Kumar, he has given very good information, very novel research methods, I would say. Azolla, its practical implications, practical issues, and various products which are made from the Azolla. It's very important. And biofertilizers for rice. Normally, rice crop is ignored as far as these biofertilizers are concerned. So he has rightly pointed out, and rice is the major crop of India. So the biofertilizers and other issues, we need to take care of that. And he has presented very good amount of research on that. So these issues, these two speakers have described very nicely, and viewers has also shared many concerns, and they have given very good answers to their queries also. At the end, I wish all of you World Soil Day, a very happy World Soil Day. We need to be more concerned about our soil. We say Pavan Guru, Pani Pita, Mata Tartamahat. So it's our mother nature, mother soil, and we should be more attached to it. Our next generation, it's our duty also. Our next generation, our children are more attached to the soil. So this is the final, I would say. But at the last, one more comment is there. Our son of the soil, we call to the farmer. We need to address his issues also. If there are any issues, he is on the roads or something like that. So we need to address the issues of the son of the soil also. So this is the last comment. Now I hand over the stage to the organizers, and I am very thankful to the organizers, Soil Conservation Society of India, for giving me an opportunity to share my views. Thank you very much. So Dr. Anshu.
Yeah. Thank you, Dr. Manmohanjit Singh. So I take this opportunity on behalf of the Soil Conservation Society of India to thank our guest speakers, Dr. Swain and Dr. Upendra Kumar. Sir, we are very grateful to you to having conceded to our request to speak on World Soil Day. Probably you might be busy because today every institute is celebrating World Soil Day, and you being a part of the soils, so you definitely might be engaged, but we are grateful to you that you acceded to our request and gave us time and enlightened the gathering. We are fortunate to have you here on this forum. At the same time, I am also grateful to Dr. Manmohanjit Singh and Dr. N.K. Parikh for having spent quite some time and very nicely chairing and co-chairing this session. Thank you very much, sir. I am also grateful to Dr. Bishundev Prasad, Mr. Rajiv Padbhushan, Dr. Kasturi, and Dr. Rajkishore for having taken the notes for the proceedings as conveners and co-conveners. Thank you very much, all of you. And I'm also thankful to all of you because you have completed this session in time. We are now ready for the Olympiad finale. I'd like to tell you that we have had several poster presentations, several e-poster presentations over the yesterday and today morning. And the select posters will be presented in a final round today from 1 p.m. to 3 p.m. to select the Olympiad winners. So probably this is the first kind of exercise for an Olympiad in poster presentations, in scientific poster presentations in the country. I welcome you all and invite to be a part of that grand finale also. Thank you very much. We'll just be breaking for 5 to 10 minutes and gathering again on this very link for the finale. Thank you very much, everybody.
So, good afternoon, everybody. We are in the final round of e-posters. During the previous sessions, yesterday and today, we have identified or screened some of the good posters presented during the session. So in that, we have selected from each session as per the judges, which include the chairman, co-chairman of the sessions and conveners, and on their views, we have screened several posters for the final round so that we can come out with the best poster during the 2 days program celebrating the World Soil Day. So we will be, we have asked the authors to come to the platform during this finale session. The selected posters will be presented. I request Dr. Anshuman Kohli to please enlist or give the idea from different sessions which we have screened out the poster presenters, so that one by one we will be taking. During this session, I am accompanied by Dr. Sanjay Swami. I think he is here. Dr. Sanjay Swami. And conveners of the session, I think they must have joined, because your critical analysis for these posters is required. So you should be present, and we have a jury for that to, as per the scorecard, we will be providing marks for each things, say the poster presentations, the title, the legibility, as well as the content, the clarity, and there are many aspects. Yesterday, Dr. Vikas Sharma has emphasized how a good poster is to be prepared, and on that, not only the presentation but also the content, the quality of the work, which will be considered during this. So we have judges. I will not disclose at present the name of the judges, but after the session we will be disclosing. So I think the judges have also joined. So we will be starting. I request Dr. Kohli to please give the idea and give the names of the poster presenters enlisted or screened for this present. Dr. Kohli.
Dr. Arora. A very happy soil day to all of you. This session, which we call the Olympiad finale, this actually is a session in which the poster presentations from various four different sessions held yesterday as well as today morning. We have selected some good posters based on their poster as well as their presentation to present in a finale, so that a comparison of all the posters can be made in the session. This is sort of a final poster presentation for this Olympiad. Probably you'll like to know that this probably is the first time when Olympiad for e-posters is being organized in the country. I thank the Soil Conservation Society of India for providing us this opportunity. I invite Dr. Sanjay Arora to please chair this session, and Dr. Sanjay Swami to be the co-chair for this session. Dr. Priyanka Suryavanshi and Dr. Swaraj Kumar Datta, you are also requested to please join the session as conveners, and please take good notes of the proceedings, because they will be used for making our final proceedings. I'll just call out the names of the finalists who will be presenting in this session. There are total 9 posters which have been selected for presentation in the finale. First one will be by Dr. Anjani Kumar. The second one will be by Dr. Bishumdev Prasad. Third by Dr. Namita Das Saha. Fourth by Dr. Ranjan Paul. Fifth by Dr. Shubham Singh. Sixth by Dr. Anand Kumar Navaram. Seventh by Dr. U. Bhagavati Amman. Eighth by Ayushi Jha. And ninth by Megha Kaviraj. I request all the 9 presenters to be available online. So as and when the chair calls your name, your posters will be displayed and you can start presenting it. For the rest of the proceedings, I invite Dr. Sanjay Arora to take over the stage and carry over with the proceedings. Thank you.
Thank you, Dr. Kohli. Dr. Kohli, you will be there during this complete session, so that if one presentation is over, then we can you can call the next one as you have the list. For this session, I will suggest that the presenter should take 5 minutes only to describe about the study and the details, along with the methodology adopted, the results obtained, and the conclusion, and what inference your study has drawn in respect to management of soil or agriculture crop production. And after that, there will be 2 or 3 questions, say for 2 minutes. Total 7 to 8 minutes for each presenter. So you have to manage your time. Time is critical in the for judging. And then we have a question answer session from different speakers. So the first poster, as Dr. Kohli will ask the first presenter to start the presentation. Dr. Kohli.
Thank you, Dr. Aruna. The first presentation is by Dr. Anjani Kumar. Dr. Anjani, are you there?
Yes, sir. Good afternoon, sir. Yes, sir.
Yeah.
Shall I share my
screen? You will share your own screen, okay?
Okay, sir.
Yeah, you can. Please go ahead. You can share your screen. I
sir,
just unshare.
Please permit me, sir.
Yes. You can upload.
Sir, shall I start?
Yeah, please.
Okay, sir. Good afternoon to chairman, co-chairman, and the presenters present in this e-poster Olympiad. Myself, Anjani Kumar from ICR NRRA Cuttack, and the topic of my presentation is modeling approach for exploring relationship between nitrous oxide emission and functional soil microbes from rice soil exposed to elevated carbon dioxide. We all know that under elevated CO2 condition, there is an increase in photosynthesis, which results in higher root exudates. And it also results in the changes in functional and structural diversity of soil microorganisms. With this view, we hypothesize that that soil nitrogen dynamics and N2O emission is influenced by combined effect of elevated CO2 and water deficit stress. The objective of our work was to evaluate the interactive effect of elevated CO2 and water deficit stress on soil nitrogen dynamics, nitrogen fraction, microbial population dynamics responsible for nitrogen transformation, and N2O emission during different rice growth periods. We did this experiment under under OTC, that is open top chambers, with facility of creating elevated CO2 up to 550 ppm and 700 ppm. The methodology was that we quantitatively analyzed the real-time PCR of MOA, NIRK, and NOS-Z genes, and path analysis using PLS-PM approach was used to discriminate the direct and indirect factors influencing nitrous oxide emission, and the software R package was used. Coming to the results and discussion, here we can see that under elevated CO2 condition, there was a increase in the denitrifier population, whereas there was a decrease in the nitrifier population. There was a significant increase in for labile nitrogen fractions like microbial biomass nitrogen, whereas there was a significant decrease for ammonical nitrogen, nitrate nitrogen under elevated CO2 condition. And we found that significant higher cumulative emission of nitrous oxide emission was observed under elevated CO2 as compared to ambient CO2 at both the rice growth stages, that is the maximum tilting and panicle initiation stage. We also did the PCA analysis, and we found that the PCA biplot constructed using the scale data set explained the PC1 explained around 66%, and the PC2 explained around 12% of variance for ambient CO2 condition, whereas under elevated CO2 condition, the PC1 explained around 61%, while the PC2 explained around 14% of variance. We also did the path modeling, that is the PLS-PM approach, and we found that if there was a direct effect of nitrifier and denitrifier population on nitrous oxide emission, and the effect was more pronounced under elevated CO2 as compared to the ambient CO2 conditions. It means that in the coming days, where there is anticipated that the atmospheric CO2 will rise, there will be more nitrous oxide emission from the rice fields. And my conclusion is that the increase in gaseous nitrogen as nitrous oxide under elevated CO2 environment was significantly correlated with the increased denitrifier and decreased nitrifier population. The predicted model for N2O emission developed in this study provides evidence that the effect of varying CO2 level was more pronounced at vegetative stage than reproductive stage. And our findings make a base for further research for enhanced understanding of mechanism of soil plant microbe interaction in different rice cultivar under CO2 enriched condition. Thanks to all of you. Hello. Hello. Hello, sir. My presentation is finished now.
Nobody's taking care of it. Yeah. Yeah. Dr. Dr. Kohli, are you there?
I'm there, sir. You can continue. The seminar is open for discussion after this presentation. The presentation is open for discussion. Please.
Okay, yes, sir. I have a question. If you can hear me, actually, I have to shift to my laboratory because there was some repair going in my room. I want to ask a question to the presenter. Can you hear me, Anjali Kumar?
Yes, sir. I can hear you, sir.
Okay, that's fine. So I want to know why do you think the population of denitrifiers increased and the nitrifiers no, lost it.
Sir.
Sorry. The electricity is gone. Sorry, yeah.
Sir, there was some problem in your sound, sir. I was. Hello.
Yes, the light is gone.
Hello. Hello.
Hello, can you hear me now?
Hello. Sir, you are not audible, sir.
Okay. Light. Okay. The light was gone. Sorry. Can you hear me now? Anjali? Mr. Anjali, can you hear me now? Anjali Kumar, can you hear me now?
Yes, sir. Yes, sir.
Light was gone. Sorry, this happened here. Yeah, so light was gone. I lost you. My question is, why do you think the nitrifier population decreased and the denitrifier population increased with the elevated levels of carbon dioxide in the atmosphere?
Yes, sir. Sir, under elevated CO2 condition, what is happening, sir? There is a enhanced, because CO2 is acting as a fertilizer for rice crop, and therefore the vegetative growth is more. And due to this vegetative growth, the nitrogen uptake from the soil is more by the plant. Therefore, there is a scarcity in the of nitrogen in the soil. Therefore, there is a decrease in there is a decrease in nitrifier population, and there is an increase in denitrifier population, and decrease in nitrifier population.
No, but there is deficit of nitrogen in the soil atmosphere.
Yes, sir. Due to enhanced uptake by the rice plant.
Yeah. So more uptake by the rice plant. There's
Yes,
a
sir.
more vegetative growth. So perhaps the plant is also taking more nitrogen from the soil.
Yes,
There's
sir.
deficit in this thing. But why should it alter the denitrifiers one way and the nitrifiers the other way?
Because sir, one is autotrophic and one is heterotrophic, and the heterotrophs are good competitors. Therefore, their population is increased at the cost of the other.
Okay. So in this particular study, do you think there is also a skewness by creation of deficit of nitrogen in the soil?
Yeah, this is the yes, sir.
So do you think, I mean, there are other factors than CO2, elevated CO2, because you're also creating deficit?
Yes, sir. Yes, sir.
Nutrient management, equivalent amount of nitrogen or sufficient amount of nitrogen should be added to the plants if the plant growth is enhanced.
Yes, sir.
Right?
Yes, sir. Yes, sir.
So, okay, so it is because of nitrogen which is decreasing in the plant growth.
Yes, sir. Yes, sir.
That is the only reason.
Hello.
Yeah, so that would be the only reason you think, right?
And that is due to the elevated CO2.
So yeah, no, definitely elevated CO2 has a consequence of increased plant growth. Definitely,
Yeah.
that
Yes,
is.
sir.
So, do you think this problem then bears such a big importance, because we have been saying this for a long time that CO2 will be elevated, then there will be more of vegetative growth, of course, which is countered by deficit of water. That is another problem. But then wouldn't it be obvious that we will have to add more nitrogen to the soil?
Yes, sir. Yeah, sir, it is evident. So many researchers have proved it, sir. Under elevated, sir, CO2 is acting as a fertilizer. That is called as CO2 fertilization for the rice crops. Okay, sir. And under that condition, there is a luxurious growth, vegetative growth. And if the vegetative growth is more, the requirement of nitrogen is more. That nitrogen is taken from the soil. And for keep the nitrogen to an optimum level, we have to give more nitrogen from outside.
My question is last time asking this question, that you're looking at the interaction of elevated levels of CO2 in the atmosphere and its effect on the nitrous oxide emissions, right?
Yes, sir.
And obviously, what the linkage you're getting is that because plant is taking more nitrogen because of more growth, so this deficit of nitrogen in the soil. When there's deficit of nitrogen in the soil, that means you're decreasing the substrate. When the substrate is low, then the N2 emissions are low anyway. But wouldn't it be obvious that in such conditions we will have to add more nitrogen to the atmosphere, then N2 emissions will also increase?
Sir, that is the one part of the story, sir. What is happening, sir? When there is more vegetative growth, there is more rhizo deposition also. That is the root exudates, and root exudates add organic carbon to the soil. Those organic carbon add as a substrate for the microbes, and microbes are many microbes, methanotrophs, nitrifiers, denitrifiers, and due to that, if you are keeping the nitrogen as a constant, if you are only considering the CO2 as a factor and keeping nitrogen as a constant, there will be a more nitrous oxide emission. Why? Because there is more rhizo deposition, and due to that rhizo deposition, there is an increase in the microbial population, which is responsible for nitrous oxide emission, methane emission also.
So you're exploring those angles also, right?
Yes, sir. Yes, sir. I have not shown the methane data here, sir. Only nitrous oxide data is shown here.
Okay, thank you so much. Thanks a lot.
Okay, sir.
Anybody else has a question? He may go ahead, please. Yeah, Dr. Anshuman Kohli, anybody else want to ask it? They can ask. I am done.
If anybody else has a question for Dr. Anjani? Otherwise, it's okay, Dr. Anjani. Thank you very much.
Thank you, sir.
Please unshare your presentation. Next,
okay,
I'll invite Dr. Bishundev Prasad for presenting his work on arsenic mitigation in rice using microbes.
Thank you,
Dr.
sir.
Bishun, you are there?
Yeah, I'm there. Sir, kindly upload my
Okay, I'll do that.
Good afternoon to chairman, co-chairman. Yeah. Yeah, it's there.
Yes, Dr. Bishun, you can start your presentation.
Good afternoon to all chairman, co-chairman, organizers, and participants. Happy World Soil Day to all. As you all know that arsenic is an element widely distributed in earth crust and present in groundwater in many countries. Long-term intake of arsenic contaminated water leads to arsenic poisoning, which cause several diseases, including cancer, skin cancer, and others. In India, more than 7 states like West Bengal, Bihar, and others are reported to contaminated as well. If we our confine that along the river Ganga states, 18 districts of Bihar state is contaminated by soil arsenic contamination as well as soil water contamination, and which cause severe arsenic poisoning. So we are working on rice because rice is a staple food crop for more than half of the world population. And one more important point is it absorbs or it accumulates arsenic up to 10 times more compared to other food grains. So it is very important to work on the rice. And the data shows that approximately 2 mg per kg of arsenic accumulated in the rice grain and 92 mg per kg of the arsenic accumulates in the rice straw. So there are different ways of mitigating, like physical method and chemical method, but they are expensive and they are not eco-friendly. And the theme of this year is to keep the soil healthy or keep the soil alive. So the best way is to work on bioremediation, where we can use the bacteria or some other microorganism for mitigating this arsenic problem, as bio remediation is cheap, eco-friendly, and effective. So I am working on bacteria in order to identify some of the suitable bacteria. We have visited to several districts of Bihar where arsenic is a problem, and we isolated the soil sample, and we isolated the soil sample and we isolated the bacteria from it, and we did the sequencing and 16S rDNA sequencing, and we identified the which species it is. So we found that one of the Pseudomonas species, which is very prominent, and it has the ability to convert arsenite into arsenate, so highly toxic form to the less toxic form. And it also has the capacity to do the phosphate solubilization. So we know that AS3 and AS3 is converted to AS5, that is arsenate, and arsenate has the same chemistry with the phosphate. So if we increase the phosphate level in the soil, then it will compete. So more of the phosphate may enter into the plant, and if phosphate will enter into the plant, it will affect the yield. It will increase the yield as well as it will be good for the plant as well as the soil. So we submitted the sustenance rDNA sequencing to the NCBI, and we also submitted this culture to the NAIMCC, and we got that session number from there. Then first important point is to see that whether this isolate is able to convert the form or not, highly toxic form to the less toxic form or not. So we did this assay where we used the sodium arsenate, and we did the silver nitrate assay, and you can see the brown coloring in the plate, which indicates that arsenite has been converted into the arsenate. And we did the real-time PCR analysis also to identify the expression level of arsenite oxidase gene, and you can see that as time passes 3 hours to 24 hours, the expression of this gene has increased tremendously, up to 2 to 3 fold higher. Then we wanted to check whether our isolate has the phosphate solubilizing activity. The data I have not put here. We performed the phosphate solubilization assay, and we find that yes, it is having the very high power of phosphate solubilization. And we also checked whether our phosphate solubilizing power is decreasing with the added dose of arsenic or not. So we found that up to 5 ppm, 5 millimolar of arsenic content, the phosphate solubilization of this bacteria is not decreasing significantly. So we have the phosphate solubilizer as well as this isolate can able to transform arsenite into arsenate. Then most of our soil is contaminated with the heavy metal. So we wanted to use those isolate which can able to tolerate the heavy metal. So we performed the metal sensitivity test also. You can see in the table, our isolate is resistant to most of the heavy metal, commonly found heavy metal. We also checked our isolate performance in the different doses of salt, because most of our soil is salty also. So our isolate performed very well up to 250 millimolar of NaCN concentration, and our isolate works well at pH 5 to 8. And we also test whether this isolate has any antibiotic resistance gene or not, because there are lots of problem if our bacteria has the antibiotic resistance gene. So we test the antibiotic sensitive test, and we found that it is sensitive to almost all kind of antibiotics. So we can kill this bacteria by applying any of the antibiotics. Then the next very important point is to do the bioassay. So we performed the pot as well as farmers field assay, and we have also done the testing with the TVK mode. And we found that when the left panel is the control, where you can see that if we increase the arsenic content, the phytotoxic effect on rice is quite visible. But when we do the treatment with this isolate, then the toxicity effect is much low. So we can see in the farmers field also, we have seen that when we have when we applied this isolate, the phytotoxic effect was not visible, and we have got very good result. And when we did the arsenic content analysis, we found that approximately 50% of the arsenic content in the seed has been decreased. So the treatment, if we use the treatment, then up to 50% arsenic has not transported to the seed part. Then we wanted to make this formulation because ultimately it should reach to the farmer. So we made this bioarsenic mitigator 1. You can see the picture also. So this mitigator, we have made this formulation, and this has been released by our RCM, Research Council Meet, and now it is available for the farmers. They can use it and they can mitigate their rice by using this mitigator. Thank you very much.
Yes, the presentation is open for discussion.
Hello. May I ask one question?
Yes.
Dr. Bishan, I would just like to ask, and I've been following this work for quite some time. I was in BU, but then I just want to know that you say that it reduces the 50% arsenic in rice seed, and
yes.
I mean, this mitigator is available for use by farmers. You see, is it safe?
Yes.
When we say that, when this brand of a university or a government institution goes with that now it is safe. But still, if the affected field is still having 50% arsenic, how good or how appropriate is it to recommend? Because otherwise, we should actually not be recommending rice cultivation in such areas. Strictly speaking, we shouldn't be recommending rice cultivation.
No, that is.
But then we are going.
Yeah, see, that is the
saying that 50% contamination is okay.
Sir, if sir, this is very nice question. So what happened? This is true. If we not grow rice on those affected area, but the government policies and farmers cannot be, they will grow the rice because they have ample amount of the water. And you see Bihar and Bihar, West Bengal and Bangladesh, they are known for the rice growing. So this is the one way, that is the government policies. They may ask the farmers not to grow rice. So I'm not going into that part. But if we are able to reduce the 50% up to the 50%, then it will be good for the farmers and it will be good for human consumption also. So it's not like if you are eating those grains, it will like 50% will be hazardous to us. At least we are able to minimize the effect by 50%. So that will be the safe that I can say. It will fall in the safe limit.
Okay, thank you. Others may please unmute themselves. Yes, Dr. Upendra Kumar, please.
Dr. Bishan, presented very nicely and very nice work. This arsenic is really a problem.
Thank you, sir. Thank you.
And my query is this formulation, self-life of formulation. That is the most important. So, have you checked self-life of formulations? This is a liquid.
Yeah, I have checked the self-life. Yeah, this is the liquid formulation. So we have checked the self-life. It is working very well up to 6 months, and but with the same activity, and it can grow up to 7 to 8 months, but it has reduced the activity. So we are recommending up to 6 months you can store this product. At room temperature. At room temperature.
Yeah, one at room temperature. This one, which farmer field scenario is somewhat different. If you apply the inoculum, then more than 50% is going to die. For that purpose, what do you think? And under natural condition, many microbial communities are many. Okay, even your pseudomonas belong to which group? You know that one. What group the pseudomonas is going to belong? Understand? What is the group of pseudomonas? In which group it is belonging?
Yeah,
like
go ahead.
bacillus.
Go ahead. Yeah.
What group? Like bacillus.
So
Tarbicuides. So yeah. Tell me.
Yeah, so whenever you apply any column, definitely there will be a competition. But what happened? You are increase the competition. So most of our, because we are at the time of application, we are dipping the rice root in the solution, in the biomitigator solution. So most of your pseudomonas will get adhere to the root. When it goes to the field, most of the microbial, it will able to perform the microbial activity because at that time it has very high dose of this bacteria. So it will able to perform its work. It is working as well as like rhizobium and other culture we are using, and the phosphate solubilizers we are using.
Dr. Bisun, have you checked what concentration of bacteria is there after application of this? If you apply the pseudomonas, okay, so what concentration of bacteria is left? Have you done work that one?
No, that part I have not done.
Because you are saying that high inoculum.
Yeah.
So
no, in the beginning it is high inoculum we are
because
putting.
it is somewhat tricky. People are doing the marker, because that bacteria.
No, we are doing that. We are trying
Yeah.
to do that. We are using the fish and other markers. We are using it.
And rhizosphere bacteria is certain problem is there. Rhizobia, you were saying that rhizobia is going inside and making residue. So there is limited chance of environment, effect is there. But here, the more environment, even arsenic also itself has effect. So this making a formulation is very easy, but pursuing in field, it is very difficult, particularly under natural inhibited.
So we have tested this for 3 years. It's not like we are just recommending. We have tested it for 3 years to the farmers field as well as TVK mode, and we have done our own analysis also, and we have found that it is successful, and it is working very nice in the field also.
Okay. What control you have kept in field?
We have kept the control untreated.
Untreated.
Where we have not given the this formulations.
So this is due to maybe not your microbe, your pseudomonas, maybe due to the whatever the microbe is there, pseudomonas or other microbe due to that.
So it would have the same effect. We did the analysis in RBD. We have kept the different replication at different places also. So if it is the effect, then in all the places it should have come the same thing. In the control also we could have get the 50% reduction.
There is any commercial culture available?
Yeah, it is available. We have no for this purpose no. For this purpose there is no culture. This is the first time we are recommending.
If you are actually promoting something, you must compare with the available one. So far there is no arsenic related formulation is there. This is the first one.
Yeah, that's
you are
true.
saying that one.
Yeah.
Are you sure?
Yeah.
You're sure?
Yeah, sure.
Okay, good.
Thank you.
I have a query.
Sir, please.
Hello.
Yes, sir. I am hearing you.
What is the mechanism? Means what mechanism these microbes are efficient in, say, accumulating in their biomass, or they are only converting the arsenate? What was the mechanism behind it?
Yeah, it has two mechanisms that we have analyzed. First thing is it has the arsenite oxidase activity, which will convert the AS3 to AS5. That is the one thing. And another thing, it has the phosphate solubilizing activity. So simultaneously, it performs two things. It can convert the form, as well as it will increase the phosphate content near the root zone. So, and this phosphate is competing with the arsenate. So if there is a more phosphate, then more phosphate will be get absorbed by the plant, since rice plant has the same transporter for AS5 as well as the phosphate. So more phosphate will enter because it will increase the competition. So more phosphate will enter, and this phosphate AS5, when this bacteria convert AS3 to AS5, then AS5 is less soluble compared to AS3. So it will be less toxic to the plant also, and it will try to settle down in the soil.
But it will remain in the soil itself, no?
Yes, arsenic will be remain in the soil. That is there. But we can minimize its entry into the food chain so that we can get the less harm of this arsenic. But it is not going out from the soil.
What is the source of arsenic in these soils?
So there are when there are in most of the literature, it is mentioning that when we started to deep boring from these areas, then most of this arsenic which is present in deep will come out.
Water is the source, no?
Water is the source, yeah.
But if there is, say, certain amount of arsenic which is toxic to plant, and then again we are not able to say flush it out from the soil, and again we are irrigating the soil, then accumulation will enhance. Okay, so years and years there will be accumulation. For temporary remediation, these organisms can be useful. Do you agree with this?
Sir, I have another story also. Like, see, when you are irrigating with the rainwater, when there is no arsenic, then concentration will reduce because it will percolate and arsenic will go down where rice root cannot able to take it out. And there are another method also. If you have a pond, if you store the irrigated, so it depends upon on which water you are irrigating the crop. If you are irrigating the crop with the arsenic contaminated water, yes, the concentration will increase. But if we can able to harvest the rainwater, and if we can able to irrigate the plant, then concentration will decrease. And if we do that along with we follow this mitigator, then actually this will be quite beneficial to the farmers.
You have means worked on pseudomonas. I think these pseudomonas are mainly aerobic. Okay.
Yes.
Pseudomonas are mainly aerobic, and in rice the aerobic condition rarely comes. Most of the time anaerobic condition exists. So you have to check it whether the organism was only useful in phosphorus assimilation or arsenic conversion was there in the field. Maybe in lab it was performing because of the you have also isolated, you have tested in the lab, and under all the aerobic conditions you have done it. But in the field it is anaerobic condition in the paddy.
That's true.
It is very contradictory, and it is very much required to check it under anaerobic condition in lab as well as field. This
Sir,
was
in
my
lab we
submission.
have, thank you, sir. We are working on it, and we have done the anaerobic test also, and we have checked whether it is able to convert in the lab condition. And for the soil samples, we have collected the soil samples, and we are speaking with some fellow who can do the speciation whether how much AS3 is converting in the soil itself. So that part is we are trying to do that.
Quantification you can do, no problem.
Yeah.
For total arsenic,
That part we have done. That part we have done. Yes.
Easily. But I am saying that aerobic and anaerobic testing both is required, because under anaerobic conditions, pseudomonas may not be active. Firstly, and secondly, large use of pseudomonas is restricted as per the fertilizer control order. You must be knowing that it is very means not commonly used because pseudomonas have the efficiency to enhance phosphorus, zinc, as well as other ions. But we rarely recommend use of pseudomonas. They have some specific pathogenicity, and it needs to be tested. It needs to be maybe to human it is harmful, or some food chain. It can, if it is an endophytic kind of thing, it can also affect. So pseudomonas is basically used where in the environment where we are eliminating other things, and we restrict its entry into other systems. But overall the study was good, but I means suggest that you should go for anaerobic study as well as its testing for any harmful effects on human as well as plant system. Okay.
Okay, thank you, sir.
Dr. Bhardwaj, any query?
Yes, Dr. Bishnu Dev Prasad, I have some questions regarding your study. It's interesting and a very good presentation.
Thank you, sir.
I want to ask you, does plant have any preference for the form of arsenic?
Yes.
So which form are
there are
they?
lots of
Yeah.
There are lots of transporter has been reported in the rice. So it has it has preferences for AS3 also. It has preferences for AS5 also. because
Yes.
both these transporters are present in the rice root system.
Right. So if the plant can take both arsenite and arsenate forms, then the bacteria which is converting the less toxic form to more toxic, or more toxic forms to less toxic forms, that means if the bacteria is converting AS3 to AS5 or AS5 to AS3, does it matter?
Yeah, it matters because it all competition. If we increase the competition, if we increase the AS5 in near the root system, and if we increase the phosphate near to the root system, then these transporters has the affinity for phosphate also. So it will try to absorb more of the phosphate compared to your arsenic.
So ultimately, the game comes to phosphorus because you see, plant has no preference
Yes,
for
yes,
arsenite
that's
and
true.
arsenate. You're saying that if the concentration of arsenate is increasing in that, or arsenite is decreasing, in other ways, then it will take more of
That's true.
arsenate.
That's true.
But plant, but there's
Yes,
another
that's true.
phenomenon that if whichever form is taken up by the plant, the ultimately in the cells, the arsenate form will be converted to arsenite form, right?
Sir, it has lots of hypotheses. Like if we there are lots of QTS has been reported in the rice, which converts this inorganic to the organic form. So if arsenic, if it passed through the bacteria, then it will be converted into the organic form. So organic form is much less toxic compared to the inorganic form. So if we like DMM or MMM, then it has much less effect on the human. So if any arsenic which passed through the bacteria, or if something has been converted by some of the bacteria, then the inorganic form will be increased. So if we consume the if we consume the organic arsenic, then it has much less effect on the human body.
No, but you're saying that actually it is converting the arsenite to arsenate. That is the role of bacteria in the soil, right?
Yes, yes.
So,
No, the question you asked, that's why I said in the plant, how much inorganic form is doing and how much organic form is doing, because lots of QTS is also there, which converts the inorganic to the organic form.
no,
So that also depend upon how much organic form is there in the plant. That will affect the severity of its effect. Yes.
so you mean to say besides arsenite 3 and arsenate 5, arsenite or arsenite, arsenic 3 or arsenic 5, there's another form which is organic form, which would be more
Yes.
taken up by.
Yes. Yes. Yes.
What would be that form? Wouldn't be that in any anic form?
That
Ultimately,
is the
all
yeah.
ionic form, right? Inorganic ionic form. Only ionic forms can be taken up by the plants.
No, plants can be both the forms can be taken up by the plant, inorganic as well as organic. But if we take up the organic form, then it will be beneficial because there is because it will have less effect on the human.
Have you published anything from it?
Which is we are going to publish it, and we have submitted a proposal, big proposal on the DBT. Also, we are trying to uh, work on some QTL, and we have identified the donor also, and we are working on it.
Okay. That's it. I'm done.
Thank you, sir.
Okay.
Thank you, sir. Thank you very much.
Thank you, sir. If there are no more questions, I would like to invite the next presenter, Dr. Namita Das Saha. Dr. Namita, are you there?
Yes, sir, I'm there, sir.
Okay. Ajit, if you are there, please upload her presentation.
Yes, sir. Sure, sir.
Shall I start, sir? Well, very good afternoon to all of you. I am Dr. Namita Das Shaha. Presently, I am working as scientist at Center for Environment Science and Climate Resilient Agriculture at IARI, New Delhi. So today I am here to present one of the experimental finding entitled 4 years of fumigation with elevated ozone and carbon dioxide found to alter soil microbial communities under chickpea crop. So just to introduce about the topic, so we all know about the present climate change related issues, and the atmospheric carbon dioxide has been increasing at an alarming rate. At present, the concentration is 409 ppm, and also again it is increasing at the rate of 2.1 micromole per annum. Apart from this, increase in the atmospheric carbon dioxide, another alarming issue is the atmospheric ozone level, which is presently a very much special concern. It has grabbed special concern of different researchers. Ozone, when present in the troposphere, is really a notorious gas. We all know that it has a dual role, and in terms of it plays in terms of pollutant, and also it also has a great role in terms of greenhouse gas. So mainly as a pollutant, it has direct deleterious or harmful effect on the plants. There are many reports available on that elevated ozone exposure, it actually deleteriously affect plant and also it reduces yield in case of many crops. So while elevated CO2 is already reported to have a positive effect in terms of CO2 fertilization and more enhanced carbon dioxide assimilation, so one can expect that there will be more photosynthesis. And also this enhanced photosynthesis rate, it may alter the root exudation pattern. And this altered root exudation pattern may really alter the rhizospheric community. So what may be the effect when in a combination with the elevated ozone and as well as elevated carbon dioxide, how actually those root exudates will be altering, or maybe it will be stimulating some growth of microbes. So there was a question about this one. So with this context, we hypothesized that elevated CO2 and elevated ozone exposure for relatively a longer period of time may alter the soil microbial community. Our objective in this regard, we have fixed that to study the effect of elevated carbon dioxide and ozone on soil microbial community structure and function under moderately longer period of time. For this, we have a good experimental setup, well built since 2014 onwards. So we have 8 free air ozone enrichment ring present at our SESCRA climate change farm. So wherein we have a facility for enriching the carbon dioxide separately, as well as enriching the ozone, and also the combined effect ring we do also have. So in this regard, all the rings, it were having 8 meters of diameter. We have set 4 different treatments. The OC1, which I have mentioned here, OC1 denotes the elevated ozone plus elevated carbon dioxide in a combined mode. The C1, it represents the elevated CO2. O1 represents elevated ozone alone, and A1 represents the ambient carbon dioxide and ambient ozone. So here we fixed the elevated CO2 for fumigation as 550 ppm plus minus 20 ppm, and the elevated level of ozone we have fixed 70 ppb plus minus 5 ppb. For this regard, I would just like to tell about, at present in the Delhi condition, the ambient ozone is reported 40 ppb, and it actually fluctuates seasonally as well as diurnally also. So with this treatmental condition, so we tried to assess the soil microbial community structure by following different methods. We have assessed the community structure through phospholipid fatty acid analysis for identifying the abundance of specific microbial biomarker based studies. Community level physiological profiling was done through biolog ecoplate, wherein 31 different carbon substrate that mimics actually the root exudates. So we have studied that utilization pattern. Also, we have analyzed 5 carbon cycling, 2 nitrogen cycling, 1 phosphorus cycling enzyme by utilizing a fluorometric analysis, which is highly sensitive one. So coming to the result part. So based on the abundance of the PLFA biomarkers, we could see that the fungi and bacterial ratio decreased under elevated ozone treatment, especially. So this maybe because the altered root exudates and its composition actually might have selectively encouraged the bacteria to flourish over there. Fine. And more bacteria in actually it indicates about less resiliency to the system. And also we all know higher fungi actually indicates about higher resiliency, and one can expect more carbon sequestration in that system. Also, we could found that the GP by GM, that is the ratio of the gram positive and gram negative bacteria, it also declined in case of the elevated ozone treatment. So actually here one alarming point is that the increase in the gram negative group under the elevated ozone treated rhizospheric zone, it in one way may indicate that there may be the pathogenic population, plant pathogenic population that were flourishing in that area. So further, the biolog based assay, as because the I would just say that the most of the plant pathogenic bacteria is actually belong to the gram negative group. So another point is that the biolog based assay where 31 different carbon substrates were used to see the utilization pattern by those microbes, we have further derived the Shannon-Weiner diversity index, which is basically the functional diversity index through this particular data set. So we could also see that 26% more diversity index in case of elevated CO2 treatment condition. So this denote actually the functional diversity was actually rather more under elevated CO2 condition, and while in compared to your the elevated ozone treatment. So altogether, here I have shown a little amount of all data, but we have a good amount of data set, and just to derive in a pinpointed idea about that whether really these all treatments are really affecting the soil microbes in terms of their functions and in terms of their community structure or not, all the data sets have been put into the PCA for analysis, and where we could see that the treatment ambient one and the treatment, the ozone, these two actually were well separated into 2 different quadrants, and which actually indicates about that there was a significant treatment effect on this for these all treatment combinations. So now finally, from the all findings of this particular experiment, I can conclude that the experimental condition what we have followed here, there was actually a shift of the soil microbes that is actually really happening under the rhizospheric zone of the chickpea crop. Chickpea was our the test crop here. So basically it was a experimental condition for moderately longer period of time. We are also focusing for continuing this particular for further many more years to see exactly these changes or alteration or shift, if these are transient or these are permanent or not. So that's all about my findings. Thank you so much. Thank you all. Sir, I have finished my presentation.
Okay, yeah, so I don't know how long. Yes, so my can I start with the question here? I have some questions for. Yes, so Dr. Namita Das Saha, I have some few questions which I wanted to know from your presentation, and that is that I wanted to know about the setup which you're using. So you said that fumigation of soil with the ozone and CO2, right?
Yes, sir.
So were these injectors into the soil or they were in the atmosphere?
No, sir. This is atmospheric fumigation only, and we had encircled area so that that amount of gases, whatever we are fumigating over there, that doesn't escape. What actually it happens in the nature, we mimic that system over there.
That is fine. That's fine. So 550 ppm of CO2 is the elevated level in the atmosphere in that chambers, which where they cannot escape. Number one, and 70 ppb is also in the atmosphere, which it cannot escape. So did you also determine resultant of these increased levels of CO2 and ozone resulted in how much levels of ozone and CO2 in the soil?
Not actually, sir. Soil will not contain this gaseous kind of thing. Actually, it will, by nature, these gases will ultimately escape to the atmosphere only. Basically, we measure the emission of the carbon dioxide from the soil per unit of area, and not ozone is, we do that, sir.
Good part of the study if you also measure, because I'm asking this because the ambient atmosphere has definitely 410 ppm or 412 with this range of CO2 and ozone also, but the atmosphere in the soil has very high amounts of CO2 anyway. So something like 2500 ppm is the level of CO2 in the atmosphere of the soil.
Yes, sir, you are perfectly right.
I'm wondering, 550 ppm enrichment of the atmosphere or ambient CO2 atmosphere of that particular soil would also be making any significant change in the soil atmosphere concentrations of CO2 or ozone. So that will be good to see for you in future.
Yes, sir. We will sure. Yes.
So when we are talking of these concentrations, let's say, starting with the CO2 concentration of 2500 ppm in the soil atmosphere, and let's say there is some change in the concentration of soil atmosphere CO2 due to your elevated ambient CO2, how much would you expect would change? Just a guess.
So first of all, there would not be such kind of thing, because if we enrich the atmospheric carbon dioxide in a reverse way, that carbon dioxide is not anyhow going to enter into the soil. That is the first of thing. So, and also, sir, ozone can only penetrate up to a level of 3 centimeter from the top soil, and again that has tendency to go out from the soil. So whatever effect,
that is fine, yeah, you're saying right. So my question is this: if there is no change in the soil atmosphere concentration of CO2 and ozone, then how would it change the microbial communities?
Yes, sir. So I just presented, sir, the basics behind that. We hypothesize that whatever effect will be there, that will be through indirect way from the effect on the plant, and root exudate through modification of those all native microbes, sir. So basically, yes, sir, these are all indirectly expected.
So that is, you're correlating the activity of the plant and maybe root exudates and substances which are going to the soil and they're altering it. Did you measure that also? The changes in the root exudation and the plant responses from the roots. Did you measure any? Did you take any parameter which quantifies that change?
Yes, sir. So we have analyzed the root exudates pattern from all the treatment combinations, and also we could found that the easily utilizable microbial, carbon substrates are quite more in case of your ozone treated plants. So they are actually secreting some of the carbon substrates which are actually fast utilizable, and that's why they are actually encouraging the bacteria to grow faster, in a faster way. Okay. Otherwise, bacteria is also expected to grow faster than the fungi.
Okay, I have another question, and the last one, perhaps then somebody else will ask. From the graph which you're presenting, I see that there is a increase due to the elevated levels of ozone, and I also see there is increase due to the elevated level of CO2, right, in the fungal and bacteria populations, right? Yes, but the fourth bar shows kind of beneficial effect of CO2, that if you have increase in the population due to ozone, but when you add CO2 increased levels, then perhaps it is negating the sole CO2 effects. Why is it?
Sir, I guess the fourth bar, it was a combined treatment. So it was actually combination of elevated CO2 and as well ozone. So what we, behind this particular response, we could expect that there may be the whatever positive effect it was there through the elevated CO2 that was actually in a way suppressed by the deleterious effect by ozone. That may be one, because there were many visible spots on the plants itself that might have reduced the photosynthesis and all. So those kind of things, because of the elevated ozone, it was suppressing the positive effects of your carbon dioxide, elevated carbon dioxide. There may be, that's why that there may be a mixed response in that particular treatment.
Okay, I'm done. Thank you so much.
Thank you, sir.
Namita,
Yes, sir.
very glad to see you after a long time.
Yeah, so nice of you. we had been classmates.
Yeah, my intention is not to ask question, just say interaction. Okay.
Yeah, sure.
Very nice study you did.
Mm.
One thing is there, you focus these root exudates. Okay, and many times you repeated also. Root exudates might be a couch to alter the microbial community and all those things. Correct or not? Once elevated carbon dioxide is there, definitely biomass increase and root biomass also increase, and they will secrete more root exudates. So, what root exudates are there? Have you identified or estimated any root exudates? What compound is there and there?
We have estimated 6 carbon compounds like glucose contents also we have seen, malic acid also we have seen, and
Okay, Navinda. What organic compounds really actually, proliferating the shift in microbial community? Any pinpoint I think you did? Like malic acid, tartaric acid, some organic acid may be there, no, in root exudates, which is actually pinpointing that one, is shifting that altering the microbial community from this ozone or elevated carbon dioxide or combination of that. Something is there or indicator, because that is the most important thing once you go for publication.
Exactly. Yeah.
Okay, and second thing is this community structure. Shift is, very vague term. We will not see till it is going to shift. Shift is not in one day or even 4 years also.
Yes, exactly.
It is taking time.
Exactly.
Okay, for long-term experiment, we can say, oh, slightly shift is there. But what kind of microbe it is going to shift? Okay,
exactly.
For that one, analyze the Illumina mycic or some is there. What structural changes is there? PLFE is there, of course, but we cannot see what kind of, whether proteobacter is there, firmicutes are there, nitrospira or cyanobacteria, whatever it is.
Exactly, you are perfectly right. I know that these are all outsourcing kind of analysis, and it requires huge amount of money as well. So that's why
IRI, huge amount of money is there. Okay. We'll go for latest molecular tool. And the second thing is for quantifying the exact thing. HPLC based you might have done for root exudation, but qPCR, real-time PCR, once you know this one is there, okay, rhizobls is more, which is actually slightly shifting that one. So we have to create some primer, and then target based you have to. So this two is there, very, very good thing to publish your article. Otherwise, people are asking, continue asking.
Exactly. Dr. Upendra, we very nicely you have suggested, and also we have planned in that way, in our in-house project also we have that target, so that whatever microbial community structure we have to need to correlate with the GHG emission based related kinds of, yes. So that's why we have, exactly. That's why we have planned for that target gene based real-time PCR and then Illumina sequencing based structural analysis and all. So we are in that way process, and also this system has already been today it is a sixth year, so we will be running that for the longer term studies. Thank you so much.
Try to go for community structure. Okay.
Thank you so much.
Correlate the yield, because if whatever it is there, a diversity that should be linked with the yield. What it is there, because yield parameter here it is missing, but you might have that one yield parameter.
We
So
do
in
have.
some treatment, it is, if diversity is going to change, how it is influencing the above ground part, because the below ground is actually attached with or linked with permanently. Dong paper is there. You must follow that Dong paper.
Yes,
He is
yes.
the Australian scientist, and very nicely he correlated the below ground microbial community, how it is changing, and then above part. Okay, so these are the some queries. Okay, not
Thank
okay?
you so much. Thank you. Thank you. Thank you. Sir, is there any question? Kohli sir, I am done. If suppose any question is not there.
I think if there are no more questions, then we can thank Dr. Namita Saha.
Thank you, sir.
And I would like to invite Dr. Ranjan Paul for the next presentation. Dr. Ranjan Paul, are you there? Please unmute yourself. And Ajit, please share his presentation.
Yes, sir. Sure, sir.
Mute yourself, Ajit. Let Dr. Ranjan Paul unmute himself. Dr. Ranjan Paul, are you there?
Yes, sir.
Yeah, please unmute. Yeah, please start.
Yeah. Sir, you cannot start skiing. This is please the schemes. I couldn't. Please allow. Hello. Am I audible, sir?
Yes, you are audible.
I couldn't share my screen. I am not.
Your poster is already on the screen.
Am I audible, sir?
Yes, Ranjanji, you are audible and your poster is already on the screen.
Okay. Thank you. So, good all. Myself, soil science division of Induce planning. Today, I will be presenter on the identify amount of in existing soil. Actually, PAC is a and the severely and they are well quality. And Hello? Am I, sir? Hello? Hello?
Yes, you are audible, Dr. Mohapatra. There's some issue with your network.
It is decided my that is the one who is offer, and that's properly identified. So, and the problem is that it is not often eliminated during PT for particle size and like for removal of oxides and organic matter H2 and treatment, that type of treatment. Also, in routine XRD scanning, which are performing at 2 degree 2 theta per minute scanning speed, is often not hello, for which we have selected the two vertisols from the Chhattisgarh. Then separated the water dispersible clay fractions of the soils and captured the XRD scans at both rapid, that is the 2 degree 2 theta per minute, and slow scans, that is the 1 degree 2 theta per minute. Very poorly saturated hydroelectric conductivity values of less than 0.2 centimeter per hour in the 0 to 100 centimeter depth. This is the weighted mean. And also the base saturity of the soil is more than 100%. So this is the presence of there is one indication of presence of peligoscite. Also, the poor hydraulic conductivity values are compatible with the exchangeable calcium magnesium ratio, since peligoscite releases the magnesium to the soil solution more faster than the other sodium spectrite and the other mineral, and that decreases sharply with the depth. That is the calcium magnesium ratio decreases sharply with the depth, and that also indicates the presence of peligoscite, and which halt the free entry of rain into the soil profile. That's why hamper the soil development for long run. Peligoscite was resolved more clearly when the clays were scanned at 0.5 degree 2 theta per minute rather than the 2 degree or 1 degree 2 theta per minute, along with applications of the deconvolution techniques of method of profile fitting as observed in the picture, that we have applied the some deconvolution method of fitting the XRD scan. So this is a kind of modeling approach to precise properly the X-ray diffractogram. So finally, we concluded that the unique depth distribution of the saturated hydraulic conductivity, base saturation, and the exchangeable calcium magnesium ratio, that would be enough to ensure the presence of peligoscite in such vertisols in absence of the XRD facilities. So from that we can conclude this that if sometimes we don't approach some XRD, or if we don't have some XRD kind of things, so from the seeing the soil properties and the other properties like calcium magnesium ratio, base saturation, we can also say that these are due to the presence of peligoscite in soil, and so we can identify also them. So that is my presentation. Thank you, sir. Hello, sir, my presentation is over, sir. Hello, am I audible, sir?
Yes.
Yes, the presentation is now open for discussion.
Yes. Dr. Ranjpal, Dr. Anshuman Kohli, can I start with a question for
Yes,
you?
please.
Yes.
Please.
Yes. Dr. Ranjpal, we had discussion yesterday also on your poster, peligoscite.
Yes, yes.
Actually, when I see your poster once again, then my curiosity increases again, being soil scientist, and I want to ask you, because you're presenting, that is fine, that you standardize the XRD conditions for identifying the peligoscite, But your conclusion is something which takes me further down into it, that reduced hydraulic conductivity, BSN exchangeable calcium magnesium ratio would be enough to ensure the presence of paligoskite in the vertisols. But you see, if a mineral is only 1% quantity in the soil, along with the other minerals which are in larger quantities, let's say for example 2:1 clays of montmorillonite or smectite, which have the similar effects of reducing the hydraulic conductivity, being swelling type of clays, right? Then reduced hydraulic conductivity will be less evidenced by presence of paligoskite than smectites or montmorillonite. That's what people usually do, right? Or suspect.
Yeah.
So this conclusion doesn't. I'm not taking it a huge conclusion coming out of what you said, right? Is it?
Yeah. Actually, let me try. Let me say something that yesterday I also said the same thing. Here I mentioned that these are the conditions that is suitable only for the non-saline and was the non-sodic soils, where there is no effect of sodicity. That is the no, if there is exchangeable sodium buildup is very low. This is not. That's why you can't call it as a sodic soils. So in that case, as well as if the soils are the non-saline, so in that case also and also these soils have the highly which is dominated by high amount of smectite mineral. So in that soils, that small amount of paligoskite can have the very detrimental effect by affecting the hydraulic properties of the soils, and this is possible by releasing the magnesium to the soil solution. Since paligoskite, it is the laboratory study that and also the by incubation studies with soil that paligoskite can release around 3 to 4 times more magnesium to the soil solution as compared to the sodium dominated smectite or the sodium smectite soils. So that's why it releases the magnesium. So that's why the calcium magnesium ratio of the soil, that is the normally calcium will be 4 to 5 times more than that total magnesium. So that that ratio is lowered down. That is the magnesium preponderance is increases. And also some scientists, they are the Alperovich et al. In the 1981 and so they termed that magnesium has the similar effect like that of the sodium in developing the soil sodicity. So this type of and this is the wide scale study from the last 10 to 15 or 25 years. The indices LUP is doing such kind of study. So first report was from the Hiller and Faraday in the some irrigated vertisols of India, and after that the Bhattacharya and co-workers and Pal and co-workers, Jade et al. And co-workers. So they have documented a lot of research in there, and they have observed that that XRD, the paligoskite peak, and also the soil properties, that is the calcium magnesium ratio is very poor, and they have severely affecting the soil hydraulic properties. So from that, by seeing that studies and on the basis of that long-term studies, the basis of, we can say that this type of conclusions.
You don't have to tell all the history of paligoskite and what in this. So we had discussion earlier also, and still we agreed that sometimes magnesium has the similar effects like sodium. Concentrations, right? Okay. Let me ask you this another question regarding this thing. So when we have sodic conditions, are you saying this is not regarding the sodic or saline conditions? You're not talking about the sodic saline conditions. So the question is, when we have sodic conditions, salinity counters that sodicity effect. So in a way, we have reduced hydraulic conductivity when we have sodic conditions, but saline water to some extent counters the reduction in the hydraulic conductivity when it does. Would it be applicable to also the conditions where the minerals, phyllosilicates like paligoskite is present, that if we add saline waters, there will be some improvement in the hydraulic conductivity?
It is not the question of adding the saline water to that, because by adding saline water, we couldn't change the exact properties of the soil mineral, because these are very long-term effect. And also we can say that the soil is sodic or like something. So we can say that due to some presence of some mineral or due to the soil chemical conditions, the sodicity or the salinity have developed. These are things. So mineral is another that soil presence of soil minerals that's inherited from the soils, beginning of the formation of the soils.
Absolutely. So sodium disperses the soil and salinity actually flocculates it. That is the
flocculates it. That is
in case of minerals, it should not happen, right? So what do you think palygoscite would be such a big importance if the concentration is only 1% compared to other minerals which are in very, very high concentrations?
In our swelling soils, that is posing the 60% or more amount of clay. So we found that the around some smectite content is also very high. In the total clay fractions, that smectite content is also 50%, sometimes more than 50% is obtained also. But in that soil also we get some 2 to 4% or 5% palygoscite. So that palygoscite may have some detrimental effect, because this is observed in the socio-radioactive conductivity. Because we laboratory in the where we are measuring that water couldn't pass. This is very
that is fine, but we will find anything like 5% in the normal soil.
For that there is need of extensive laboratory. That is the various different kind of soils that have the palygoscite content. We have to put in the laboratory, then we have to incubate, then we have to do the all this these studies. Then after that also we can say strong conclusion like that. But this is the may possible that may have the effect on the possible reduction in the crop yield, or may have some possible reduction in the soil property.
Our studies indicate that there is also interactions of ions. For example, if it is the clays, there is more of calcium, then the interactions are beneficial than if it is sodium. Does it also happen? That is the case with the montmorillonites and smectites. Does it also happen with the palygoscite? I mean, there are interactions of palygoscite concentration with the sodium and calcium also.
No, the palygoscite concentration is directly linked to the magnesium content, because it is rich in magnesium.
Because it's rich in magnesium, of course. But if the concentrations of calcium and sodium,
that is not so much of importance, because calcium is dominant cation in the soil, and it is inherently present in the soil in very high amount, as you all know that. But sometimes in the arid regions or in our semi-tropical regions, that calcium is present at the nodular form or as the powdery form of calcium carbonate. So this is due to more aridity and from the temperature and the soil. So also, so this calcium inherent calcium was but for in terms of as far as the palygoscite is concerned, literature says, or we can say that the magnesium is the most important as we link with the palygoscite in presence of either presence or absence of the sodicity. That may have the effect may have differed.
Okay, yeah, I'm done. Thank you so much. Good presentation.
Yeah, thank you, sir.
Yeah, Dr. Kohli, I'm done. If anybody else has question, they may ask. Dr. Kohli, Dr. Arora, Dr. Kohli, somebody is there. Somebody else has question. He may ask if Dr. Paul. Yes.
If anybody else has a question, he can please. Otherwise, Ajit, please unshare Dr. Ranjan Paul's presentation. And next we have Dr. Shubham Singh. Is he there?
Yes, sir, I'm here.
Dr. Shubham Singh?
Yes, sir.
Thank you, Dr.
So I will share my screen. So can I start? Hello. Hello.
Yes, you can start.
Hello.
Everybody is hearing you. Yes.
Okay, sir. Okay. Good afternoon to one and all present here. My name is Shubham Singh, and I am from Central Agriculture University, and I'm going to present my poster on managing soil acidity and phosphorus availability by Ajola Incorporation. So, to take this topic, we have some key issues, especially in Northeast region. 95% of soils are acidic, and especially in Meghalaya, the amount of area which is under acidic soil is 2.24 million hectares. So in this acidic soil, the main problem is phosphorus deficiency, and it is due to the high native P fixing capacity. And also to manage this, the farmers of this region generally use liming method for soil acidity management. But there's a problem in over liming. When the farmer use over than the recommended dose, then it can lead to the P fixation by calcium and also the eutrophication. And in this region, the main staple crop is rice with the production of 1.12 ton per hectare. So we chosen the rice as a crop. So in material methods, we design our experimental treatments with first treatment control, second with sole application of Ajola, 3 and 4 is the sole application of urea with different doses, and 5 is the integrated treatment of organic and inorganic in RBD. So we formulated is with two objectives. First is to estimate the soil acidity indices by Ajola Incorporation, and second to estimate the temporal soil phosphorus availability by this integration. So we find out the results on the different soil parameters. In the graph one, we can see that the effect of different treatments on 4 parameters: pH, uh, exchangeable calcium magnesium, CC, and the saturation. So here the probable reason for increasing the pH in Ajola treatment, which is treatment T2, T5, and T6, is the decrease while applying the Ajola at the time of transplanting. The decrease in the aluminum and the release of basic cations due to decompositions of manures will be the probable reason for this, and also the low pH status in the soil application of inorganic application by using T3, T4 is the residual acidity of the fertilizer, because of the residual acidity of the fertilizer. And here the acidity is also increasing because of the higher biomass of the Ajola which is incorporating at the time of transplanting. So second graph, in graph 2, here we study about the effect on the acidity indices parameters. So here we see that the Ajola treatments were showing uh, a bit about the soil acidity are low than this soil application of the urea. This is because of the proton consuming availability of the humic materials might reduce the acidity in this case, and also the slow reduction of the acidity might be explained by the steady formation of the organic materials with functional groups such as carboxylic and phenolic groups during the decomposition of the Ajola, and also here the low solubility of the calcium carbonate. And the third graph, which is graph 3, here we study on the temporal phosphorus availability on the like how phosphorus is performing on the basis of time with different crop periods starting from 30 days to till maturity. So here we see that the treatment 6 showing the better result as compared to other treatments. It's because of the higher content of organic carbon helping to immobilize helping in immobilization of the phosphorus into labile form, and also the organic matter which enhance the labile phosphorus through complex sections of calcium and magnesium when applied by inorganic matter. So overall, we concluded with the 3 broad conclusions. First is the soil acidity indices parameters significantly improved by Incorporation of Ajola and it also helps in soil fertility. And second, the temporal availability of the phosphorus decreases with increasing crop growing periods in respective treatments. We also analyze for the N available K and organic carbon, and in that three same results were coming. And the third conclusion is finally the treatment 6, which is the incorporation, which is the combination of the azolla incorporation and urea at the rate of 60 kg nitrogen per hectare is the best treatment for maximize the production. And in the right side, these are the crop at the different stages. So thank you all. With this, I conclude my presentation.
Thank you, Dr. Shubham Singh. I think you are logged in from two devices. That's why there's some noise. If you could log out from one of them.
Actually, sir, I am sharing from desktop and connecting from phone. That's why. And in desktop, this
Okay.
audio
If there
problem
are
is
any
there.
questions, yeah. If there are any questions, okay. Shubham, I just want to ask you one thing. If this technology of incorporating Azolla is implemented, how do you think this much demand of Azolla is going to be met?
Sir,
if you want to manage soil acidity using Azolla.
Yes, sir. Yes. For that, we can use the technique of dual cropping. We can grow the azolla with the rice crop, and we can incorporate in that soil only. Sir.
Okay. Yeah, I want to. Sure. Yeah, Dr. Kohli, can I ask a question?
You're welcome. Welcome, doctor. Please.
Okay, yes. So, Dr. Shubham Singh, nice presentation. Thank you so much. I want to ask you one thing which I'm noticing now. Yesterday also we had some discussion about it, but today I see. So you're talking about acidity, which is the levels I see here is 1.5 to 3. It is highly acid soil, is it?
So the initial pH was 5.1.
But you know, I look at the graph too. Then I have no idea where is the indications of levels of acidity, because on the left side we have a scale which is 0 to 3.5, but it doesn't specify what is it. And to the right, it specifies 70 to 84. It doesn't say what is it, right? And perhaps there is no scale for acidity. So my suggestion, future suggestion, is that, yeah, these graphs should have.
Yes, sir. Yes, sir. Yes, sir.
There should be statistics,
yes,
and there should be proper labeling of the axis, right?
Yes, sir.
Yeah, it's a suggestion for future, right? Yeah. So, this,
okay, sir.
this being suggestion, my question here is this, and this is what I asked yesterday also. Question is that, you know, you're talking about phosphorus levels of azolla to be something like how much? 4%? 5%? What?
So 6%
6%.
in azolla.
6%. So did you calculate?
So 0.6. 0.6.
So you must have also calculated that according to that content, the phosphorus levels which are being added through treatment 3 and 4 as recommended phosphorus levels, and
Yes, sir.
treatment 2, 5 and 6 are the equivalent.
Sir, the phosphorus application for all the treatment is same, sir, as per the recommended dose, that is 60 kg per hectare.
And how much? How much of phosphorus did you add through the azolla?
So the initial is 0.6, and the later, sir, we didn't analyze after because it's already incorporated in the soil.
Yeah, so of 16 tons per hectare of azolla, If it is 6%, how much would it make?
So it's 0.6%.
Oh, it is 0.6% only?
Yes, sir. Yes, sir. 0.6% only.
For Azola?
Yes, sir. Ajola on dry.
On dry weight basis, na?
Yes, sir.
So you're just adding uh, how much kgs of you? So you're adding something like if it is 0.6%, then 16 tons per hectare. That means you are adding how much of it you are adding? 0.096 tons, right?
Yes, sir.
Or 96 kgs of phosphorus you're adding. So there is discrepancy in any way because you're adding 60 kgs of phosphorus through the fertilizer and you're adding 96 kgs of phosphorus to the azola. Could it be because of you are adding different amounts of phosphorus?
And so maybe from the initial application of phosphorus by that recommended dose, maybe for that reason this graph is coming like that because it's adding extra phosphorus in that Ajola treatment with the application of Ajola.
What if you compensate that from the inorganic fertilizer? That means instead of 60 kgs of inorganic fertilizer, if you add 96 kg of inorganic fertilizer P, maybe they all will come to same level. Is it? Do you think so?
Yes, sir.
So if you add another 36 kgs of inorganic fertilizer in treatment 3 and 4, do you think all these treatments will come to the same level in terms of phosphorus availability?
Yeah.
So that's why the presentation I'm done. So anybody else can ask.
Actually, this 16 ton per hectare of azola is on fresh weight basis. Okay, and azola.
Yes, sir. Fresh one.
Azola contains more than 80% water. Okay. Do you agree?
Yes, sir.
So means and your phosphorus content in azola is based on dry weight basis.
Yes, sir.
So you are giving very means less amount of we cannot calculate on direct basis how much quantity of phosphorus was supplied through azola.
Yes, sir.
So in your treatment T2, what is the amount of phosphorus through azola and through chemical fertilizer you have applied? 60 kg per hectare in T2 also?
Yes, sir.
Plus
for phosphorus in all treatments, 60 kg.
So means the total quantity of applied phosphorus was much more in case of T2 and T5.
Yes, sir. But sir, the Ajola application is same in T3, so the effect will be same in the T2, but the difference will come in T5 because in that the urea is there. So this difference will differentiate between the sole application of Ajola and the combined application.
No, it will affect the yield. Na? Nitrogen by urea will be affecting the yield, not the phosphorus availability or your pH.
Yes, sir.
There's also this thing. Can I in between? Yeah, that is right discussion actually. Kind of, I'm finding some kind of discrepancy here, and this is that even if it is 20% moisture content in this thing, that would make it 19 kgs, roughly 20 kgs of phosphorus is being added through azola. Now instead of 96, now we're coming down to 20 kgs. Now the question, there is a different question which arises. If you have in treatment 2, you have only 20 kgs of phosphorus being added, but in the treatment 5 and 6, you have recommended doses of phosphorus, which is 60 kgs per hectare, plus 20 kgs through azola, which makes it 80 kgs per hectare, and the availability is same in treatment 2, 20 kgs, as well as treatment 5, 80 kgs, as well as treatment 6, which is also 80 kgs, right?
Okay, sir.
Another ambiguity, Dr. Shubham Singh. You said that azola would ideally be grown in the same rice fields. So it's basically competing for phosphorus at that time only, and then when are you applying it?
No, sir. I applied it before the so
Before
that it can incorporate it completely.
You are in one season you will be cultivating azola, and that azola you will be harvesting and applying in the subsequent year, or what?
No, sir. This application, this incorporation is 1 day before the transplanting.
How do you bring it? When I asked you how are you going to source so much of azola, you said that we'll be growing it along with rice.
Oh yes, sir. That dual cropping.
So there is
For
some
that, sir,
some ambiguity. When you
so
are
for
growing
that,
it
like
along with rice, how are you going to apply it to the rice? For
No, means
experiment it is okay, but how do you do it practically?
actually
so
azola.
for this,
Please
Azola is
listen.
usually grown with rice, yes, but then how will you apply it to the rice crop?
Actually, what was happening, I believe, that azola was used. There is no use of azola, especially for phosphorus. It is for nitrogen. Okay, and the biomass total carbon content enhances, and during uh, these paddy season, when azola is being cultivated along with means introduced along with the paddy, the enhancement of nitrogen is main, and then microbial activity also get enhanced, and sometimes temperature as well as pH interference can be reduced. But here the study claims that managing acidity and phosphorus availability. Phosphorus availability is very rarely affected by azola. You
We
are
can move
not
to the next
incorporating.
presentation.
You are not incorporating the biomass of azola into the soil. Here you have to means you have inoculated the azola media, and you are cultivating it throughout the growing season to enhance nitrogen availability, or your target was only phosphorus, because your treatment also suggests that there is urea, there is nitrogen, means your emphasis was mainly on nitrogen. And but
yes, sir.
for preparing this poster, you have only mentioned about
Okay.
phosphorus and
Phosphorus.
soil
Yes, sir.
acidity. So actually, this study doesn't clearly depict the total study or the main objective
Yes,
of the
sir.
study. The main objective was nitrogen, I think.
Yes, sir. Main objective was nitrogen, but for the I incorporated phosphorus.
That's why this is a partial presentation, so we cannot conclude anything significant from this, because number of discrepancies और जो प्रॉब्लम दिख रही है लोगों को, वो इसी कारण दिख रही है क्योंकि जो मेन काम के लिए आपने वो डाला था वो तो बताना नहीं चाह रहे इस प्रेजेंटेशन में। आप फास्फोरस में लेना चाह रहे हैं। फास्फोरस के लिए एजोला का कोई बहुत बड़ा रोल नहीं है। मेन बात तो फास्फोरस तो वैसे भी आपकी सॉइल में अच्छा है या कम है? डेफिशिएंट है सॉइल फास्फोरस में?
नहीं सर, नॉर्मल।
तो नॉर्मल है, बताओ ना कितना है? ऐसे थोड़ी ना सॉइल साइंस का आदमी बोलेगा नॉर्मल। बताओ कितने केजी पर हेक्टेयर?
सर 17.23 इनिशियल।
हाँ तो अच्छी क्वांटिटी है। कि इसकी क्रिटिकल लिमिट क्या होती है फास्फोरस की?
So from 15 to 21.
चलो कोई बात नहीं, हम लोग 12.5 से 25 मानते, चलो कोई बात नहीं। आपका ये वाली रेंज तो सफिशिएंट रेंज में आया ना, डेफिशिएंट रेंज में तो है नहीं। तो इसका और फास्फोरस की जो भी आप केमिकल से दोगे उसकी भी जो है यूज एफिशिएंसी इतनी लो होती है कि और खासतौर पे एसिडिक कंडीशन में तो वो क्या कहते हैं, फिक्सेशन बहुत है, एल्युमिनियम का हो गया चाहे कैसे भी ऑक्साइड बन गए। तो यहाँ पर एजोला दूसरा काम कर रहा था कि अगर उसकी अवेलेबिलिटी को एनहेंस कर सकता है जो आपका नेटिव फास्फोरस है, ना कि जो आप इसके थ्रू डाल रहे हैं। इसके थ्रू डालने का कोई इसमें ऑब्जेक्टिव भी नहीं था और ना ही इसका आउटपुट आ रहा है इस रिस्पेक्ट में कि आपने एजोला डाल दिया तो जो है वो फास्फोरस ऐड हो गया सॉइल में, ऐसा नहीं है। फास्फोरस बहुत ही माइन्यूट क्वांटिटीज में ऐड होगा, वो ऑर्गेनिक मैटर के रूप में जब आपने डिकम्पोज हो जाएगा तो उसके बाद वो थोड़ा बहुत क्वांटिटी में होगा, वरना मेन तो आपका एम्फेसिस नाइट्रोजन पे था, इसको नाइट्रोजन के हिसाब से प्रेजेंट करना चाहिए था। चलिए कोई बात नहीं। अच्छा ये बताइए, ये एसिडिक सॉइल में जो फास्फोरस को डिटरमाइन करने का मेथड कौन सा यूज किया था?
Bray and Kurz 1.
ब्रे में ओल्सन क्यों नहीं किया था?
Sir, pH below 7 m, below neutral है सर, इसलिए 5.1 pH है। सर ऑल्सन तो अल्कलीन के लिए।
अच्छा, और जो ये पीएच सॉइल का आपका 5 पॉइंट कुछ है, ये 1:2 है या 1:2.5 है या और कोई मेथड यूज किया?
सर 1:2.5 है।
चलिए ठीक है। अभी पीएचडी कर रहे हो या कर चुके हो?
नहीं सर, पीएचडी फर्स्ट ईयर सर।
ये एमएससी की स्टडी है?
यस सर।
एमएससी में आपके एडवाइजर कौन थे?
सर डॉक्टर संजय स्वामी सर।
हाँ, उनका फास्फोरस पे बहुत काम है, तो ठीक है, अच्छा है। पर इसको जिस काम के लिए जो चीज यूज हो रही है, पीएच पे एम्फेसिस देना चाहिए, और ऑर्गेनिक कार्बन कंटेंट कितना बढ़ गया एजोला डालने से, या और जो अवेलेबिलिटी का जो इशू है ना, फास्फोरस की जो अवेलेबिलिटी विद रिस्पेक्ट टू आपने टाइम दिखाया है, उसका जो मेन रीजन आपको फाइंड आउट करना पड़ेगा कि कैसे उसने अवेलेबल कराया। अगर बढ़ गया किसी ट्रीटमेंट में एजोला डालने से तो उसका क्या मैकेनिज्म रहा होगा, है कुछ आइडिया?
सर एक रीजन तो सर उसमें ये है कि जब अजोला हम लोग इंकॉर्पोरेट करते हैं, जब वो डिकम्पोज होता है तो वो ऑर्गेनिक एसिड रिलीज करता है, और वो ऑर्गेनिक एसिड सर बाद में
मेरी बात सुनो, एजोला तो 60 डेज तक चलेगा या 10-15 दिन में ही खत्म हो जाता है वो।
सर वो तो इनकॉर्पोरेट हो जाएगा जब सॉइल में चली जाएगा, तो सर जो उसका इफेक्ट है,
पैडी क्रॉप खड़ी है, पानी भरा हुआ है, उसके अंदर एजोला डाला हुआ है, ठीक है?
यस सर।
आपके साथ डिकम्पोज होके मिलता जाएगा ऐसी मिट्टी में?
नहीं सर, वो टाइम लगेगा सर, विद इन दी क्रॉप पीरियड वो रहेगा
जब तक हार्वेस्टिंग का टाइम आ जाएगा।
जी सर।
तो वही इशू है ना।
उस केस में ये ऑर्गेनिक एसिड्स वाला लॉजिक तो काम नहीं करेगा।
वो तो बाद में करेगा, अगली क्रॉप में उसका इफेक्ट आ सकता है, या उसके में आता है जबकि जब फास्फोरस की अवेलेबिलिटी की जरूरत ही नहीं है। आफ्टर 25 डेज ट्रांसप्लांटिंग के बाद आपको फास्फोरस की अवेलेबिलिटी की जरूरत ही नहीं है। कोई ऐसा फेज बता दीजिए जिसमें फास्फोरस की रिक्वायरमेंट हो रही हो प्लांट के लिए, जरूरत ही नहीं है।
यस।
थोड़ा ये इसको देखो, एमएससी की स्टडी है, कोई बात नहीं, अच्छी स्टडी है, बहुत ही मतलब एक आइडिया अच्छा है, पर आपको सब चीजों के इसके जवाब चाहिए और आपको इसको थॉरोली देखना होगा कि कौन कौन से जो हैं मैकेनिज्म काम कर रहे थे और इसको मैं जस्टिफाई कैसे करूं, ठीक है?
यस सर।
ठीक, थैंक यू।
थैंक यू सर।
डॉक्टर नीले, आर यू देयर? डॉक्टर नीले?
Yes, sir.
हाँ, आप तो एसिडिक वाले एरिया के हैं, कुछ
जी,
बताइए।
Yes, whatever observations you have mentioned, these are true. Normally, we use azolla to replace mineral nitrogen fertilizer.
Yes, yes.
Their effort was good, but as you have said, they may include some additional treatments, just like that nitrogen
तो इनकी स्टडी कंप्लीट हो गई ना, तो अब ये है कि इसको जो भी चीज प्रेजेंट करें उसको जस्टिफाई प्रॉपर करने की कोशिश करें। और जब अवेलेबिलिटी फास्फोरस की अगर बढ़ रही है तो इसका एक बहुत दूसरा खतरनाक पहलू ये होता है डॉक्टर साहब कि जिंक की अवेलेबिलिटी रिड्यूस हो जाती है।
यस यस।
अगर जिंक की अवेलेबिलिटी रिड्यूस हो रही है तो फिर तो प्लांट के लिए हो सकता है कि वहां पर डेफिशिएंसी हो, और खास तौर पे एसिडिक कंडीशन में रहती भी है डेफिशिएंसी
रहती।
हाँ, तो फिर कहीं ऐसा ना हो कि एक काम किया और दूसरा बिगड़ गया। ये दो तीन चीजें देखनी चाहिए।
बिकॉज एजोला के ऊपर काफी काम हुआ है हमारे यहाँ पे भी।
अरे आपने भी काफी किया है, तो इसीलिए तो हमने कहा आपके इनपुट जरूरी है भाई।
कोई नहीं, ये एमएससी का काम है बस, इसे
नहीं ठीक है, अच्छी बात है। ठीक है सर, ठीक है, थैंक यू। तो ओके शुभम, Dr. Kohli, we can move to next presentation.
Yes, we can move to the next presentation. Ajit, please load the presentation of Dr. Anand Kumar Nyaram. Dr. Anand Kumar Nyaram, please unmute yourself and.
Yes, sir.
You're invited for your presentation, please.
Thank you, sir. Can I start, sir?
Yes, please start.
Okay, thank you so much, sir. A very good afternoon to all the respected members present today in the moment. My name is Anand Kumar Nauram, belonging to ICAR Kajri Regional Station of Bhuj. Today, I would like to share one very important yet very untouched area of research. Therefore, my topic is how India lost its vast bunny grassland to Prosopis juliflora. To start with, bunny is Asia's largest grassland, located in arid Kutch, Gujarat, extending an area of about 2,617 square kilometer. This is one of the unique and complex ecosystem you will ever see in India, very rich in biodiversity. So in Bunny, there is a pastoral community called Maldaris. Mal means cattle, who lives in Bunny and depends on Bunny for their own livelihood. But in 1962, when India was celebrating its 13th Independence Day, a major event took place in India's largest district, Kutch. So what happened in Bunny? Bunny shares its northern border with Rann of Kutch. So to stop the ingress of soil salinity from Rann of Kutch, seeds of an exotic plant called Prosopis juliflora was scattered over an area of about 31,500 hectares using helicopters. This changed the ecology of Bunny forever. Today, it's not a research article which I am going to present to all of you, but it's a kind of a highlight that how Bunny is losing to this Prosopis juliflora. This bushy and thorny plant propagates very quickly and extract lots of water from the soil, thus not allowing the other native plants to grow. Instead of saving the bunny, it replaces the native grassland. This started affecting the kettles of bunny as the native grasses were disappearing slowly. Even the Maldaris need to buy grasses from outside, even though they are from the bunny grassland. The cows cannot digest the juliflora leaves and causes a lot of harm. Therefore, in 1965 to 1966, there were about 66,000 Kankrech breed of cows. Now we have only bunny buffaloes. In 1979, only 5% of Bunny was covered by Juliflora, but now more than 54% of Bunny is covered by this invasive plant. It was once thought to be a solution of soil salinity, but increases the problem manifold. Soil salinity is increasing at the rate of 80 km per year in Bunny. So the question is, today I would like to highlight the mechanisms how this Juliflora can survive like a king in this arid Gujarat. The first thing is the tree has low branching and high coppicing abilities. It can even flower even after 3 to 4 months of germination. It has two distinct root system, as you can see in the left side of the figure, that it has two distinct root system, one lateral surface root system and another deep tap root system. They may have a role in hydraulic redistribution. Most important is the allelopathic effects. It can produce a significant amount of allelochemicals from everywhere. You say either from the leaves or either from the roots or pods or even the dry leaves. These chemicals are water soluble and can reach the ground before the leaves fall on the ground. So some of the examples of allelochemicals which I have, you can see on the right side of the poster, they are juliflorine, julifloridin, julifloricin, juliprocin, and mesquitol. Numbers are there. The tree is adapted to low phosphorus levels, but still able to fix the nitrogen also. It's also tolerant to soil salinity. It is easily distributed through animals. But there are so many questions to be answered. What are the major soil changes due to the Joliflora invasion? Some have reported a little bit of change in pH, some have reported null changes. So why is there variability in the results? And there are very, very limited scientific evidences in this matter too. Any differences below the canopy or outside the canopy? Are these chemicals affecting the soil microbiota? Or is the soil microbiota, the composition and the structure of the soil microbiota changing according to the yellow chemicals it's producing? How much this Juliflora is contributing to bunny desertification? Several things are unanswered till now. It's not a new problem, but still very new in research till now. Being said all these problems, it is also a very good source of charcoal and biochar. The pots can be used as a flour, but there are certain exceptions regarding this. It is used as a foil for subsistence because of its high calorific value and low ash content. It is also explored as an animal feed, but precaution must be taken because if the animal feed contains more Juliflora, it is not safe for the animals because of its neurotoxic effects, as it contains high amount of piperidine alkaloids. So there is a lot of research gap in this field, and we need to act quickly to fill this gap. The Green Revolution has brought Juliflora to bunny, but it significantly affected the White Revolution in bunny. Therefore, I take this platform to address to all the young researchers today to work on this area, not only to save bunny, but also to save the first pastoralist community called Maldaris. Thank you so much.
Thank you, Mr. Anand Kumar. Dr. Kohli. Mr. Are you listening?
Yes, sir. Yes, sir. Your voice is audible.
Your presentation, you want to, first of all, what is your designation at Hazri?
I just joined as a scientist, sir, in Booth Station. Yes, sir.
Which year you joined?
Yes, sir. Please repeat again. Your voice was not audible.
In which year you joined?
Okay, sir. I joined last year, September, sir.
Okay. And last September you joined, and during the lockdown you thought that bunny is creating problem, and you come out with a number of questions on bunny system and advising the young researchers to work on that. Okay.
Yes, so we also have a project under this where we are doing a special assessment of it. We are also trying to restore this place also.
You have to work and soil science or agronomy?
Yes, I'm from soil science.
Soil science.
Yes, sir.
So, do you think that introduction to flora during 1970s was a mistake?
Yes, sir. Seeing the invasive nature of this species, seeing the current situation, I think that was a mistake now, because it has
the policy was wrong to introduce vegetation into the very saline areas to stop ingress of saline water, and nowadays You are seeing that large number of projects are being run by government to stop ingress of seawater, use of halophytes, and as well as that means plants are being cultivated in the coastal region. So these are all mistakes being done.
No, sir, it's not a mistake if we have to just stop the ingress of soil salinity or erosion. But when it comes to its invasive nature, when it turns from a plant which was thought of restoring the bunny and turns out to be an invasive which cannot be controlled and making the grasses of the grassland disappearing, so that might be a little bit more priority problem for that. That's why I would like to address that.
Lantana Lantana was also introduced in hilly areas to stop erosion during the 1950s. Before that, 1940s, during the British period, and nowadays it has become the largest weed.
Yes,
Okay, but,
yes, sir.
but at that time was to conserve the soil, to minimize the landslides, and similarly here also the motive was to uh, restrict erosion from those areas, because there
Correct.
is less water availability,
Yes, sir.
stop salinity. So this was the main motto, and in a one go you can have these vegetations which can not only beans, these are hardy, they require less water, less management, and they can cultivate easily. Self propagation is possible. So these were things behind that. And now there, if there are some issues related to, otherwise prosopis has multifarious uses, not
Yes,
only
sir.
for fuel, not only for uh, if you have been to CSSRI Soil Salinity Institute at Karnal,
Mm-hmm.
you can have tasted biscuits as well as
Mm-hmm.
many
Oats. Yes, sir.
things by from Prosopis, and Dr. Gurbachan Singh, our ex-director, uh,
Yes, sir. Yes, sir.
CSSRI, as well as renowned scientists, have explored these things, that there are multiple uses of Prosopis. So we
Yes.
cannot say that it was a mistake. It was the requirement of that time, and it was very well planned to. But nowadays, as the population is growing, as the need for land is there, multiple works are there, and the scientists are searching for new ideas, new things. So you come out with some options that, um, if the juliflora is not palatable for the cattle, how mm, you can introduce along with some other fodder grasses to mix it with them and then use it. As a scientist or an agronomist, you have to see whether it is benefiting the soil. If
Yes,
it
sir.
was not there, then what? And if it is there, then what?
Yes, sir.
Soil
We are
is
also
there.
checking the legacy effects also. Even
Yes.
after the removal of the surface, it should not cause erosion to that grassland. And how much that the chemicals which is releasing, are they affecting the soil for how many years? We are trying to see the legacy effects also, because as a scientist we should not directly say that okay, we should remove the plant.
That's why I
Yes, sir.
pointed out.
Yes, yes, sir. Thank you so much.
In Lucknow, there is a Salinity Research Institute, CSSRI Research Center is there, and there also Prosopis has been extensively cultivated and have benefited in amelioration of soil salinity and sodicity.
Yes, sir.
Okay, so if they, and having a good carbon content, and
Charcoal.
yeah, you are saying it yourself that biochar can be one of the options.
Yes, sir.
So multiple uses of that, and keeping this in view, the requirement of the cattle and all that, there are other options. It doesn't mean that eliminate uh, completely the vegetation and go for the grasses you require.
Yes, yes, I agree to that, sir.
The problem in the Bani area, I have also visited Kutch, and it is that you cannot grow common fodder grass.
Yes, sir.
It
We
is
have to go for native.
You agree with me?
Yes, sir. I also completely agree with you.
There be some problem, but the solution can be searched by the scientists, and you must have be having your Kutch station some of
Yes,
the
sir.
animals also, or no?
Presently, sir, I'm the only scientist left here. Nobody is there in scientific staff. So last time also we have done the restoration program in which we try to have a seed production plot in our station for the native grasses, and we have distributed around 200 to 300 kgs of native seeds to the farmers of Pani. So we have started a little bit small step we have done.
Carbon, soil carbon, as the environmental issues were there at that time when the bunny was engulfed with this.
Yes.
Uh, so the number were addressed with this policy, and now if the problem is there in respect to fodder and all that, so you should means the your organization as well as the other allied organization should take this challenge without criticizing the earlier works. Okay, that will be a healthy way forward.
Sure, sure.
Dr. Bhardwaj.
Yeah, so yeah, thank you so much. You already had a vivid discussion about the precipice and bunnies.
Yes, sir.
We had some discussion yesterday also. Yeah, that is
Yes.
right.
Yes, sir. Yes, sir. We did. Thank you so much.
I have new curiosity now, and that curiosity is, how do you know that the movement or expanse which has come into the bunny grasslands, the precipice expanse of precipice which has moved into the bunny grasslands, is only driven by the seeding or invasion. Could it be that the native grasses could not tolerate the climatic changes in the region, and precipice, which is a hardy plant, actually very hardy plant, tolerates salinity, tolerates low water content, just moved in? You think it's maybe it is the conditions which are created in the region, climatic conditions, that made the grasses vanish, and the precipice is just moving because it can survive the hard conditions.
Yes, yes, that
Could it
is
be
one of the
possible?
options. Yes, sir, that is one of the options also. But the thing, the one of the hypothesis made by the all the researchers which have done work here is that even though when we visit the bunny grassland, when we see a very big canopy of Brassophylla flora, we can see very clearly that the grasses won't be able to grow around the vicinity of the plant because of the chemicals, chemicals.
But
So
yeah, and that is the case with many plants, many trees actually. So when
yes,
you
sir.
have tree population, the grasses rarely grow under them until unless it is scarce population, right?
Yes, sir. Yes, sir.
And if you add adverse conditions to it, for example, there is no availability of water in that particular region, then the survival of the grasses is harder than the trees, just like
Yes, sir. Yes, sir.
precipice and all that. Condition.
Certain,
One suggestion is that you should look at the climatic conditions also,
sure,
what
sure, sure.
changes which has come in.
Certainly, sir.
Doctor,
Sir, there will be a
Doctor,
contribution of the climate change
actually,
on the. Yes, sir.
you cannot say that under the canopy the grasses vanish or have some adverse effect due to precipice, because during last 15 years we have seen that in Sodic areas precipice there is no problem of growing grasses um, between the lines of precipice. Maybe some of the species, it is maybe species specific, may have been having some problem.
Yes.
Otherwise, there are number of grasses which can easily grow in and around
Yes, sir.
the
Yes, sir.
zone of precipice. I think Dr. Bhardwaj, you have witnessed at Shivri Farm uh, the experiment where the silvipastoral system was followed,
Yes.
and for 5 years different grasses were cultivated
Yeah, yeah.
in the
Yes, sir. That's correct, sir. Even in Delhi NCR also we have seen growing of
yes.
the processes with the other. Yeah.
So we cannot vaguely say that the grass cannot grow with
Yes, sir.
precipice. Grasses grow, but
Yes, sir.
maybe the
native species, sir. The native species of the bunny.
Native species are most susceptible sometimes.
Yes, yes. So, and certainly, as Ajit sir have also stated, there's a part of climate change also. That's why I really wanted. We are also working, but I really wanted to highlight that there are a lot of things that we can do in this area. I'm not trying to give a negative this thing, but I'm trying to encourage
Yeah.
the other researchers also that this is a very big scope that we can work.
Already that you being the lone scientist and having the
yes,
of a very premier institute of India working on arid zone, and you should take this. And if you required some guidance and say inputs from Dr. Bajwa, expert
Sure,
in
sir.
soil
Very happy. Yes, sir. That will be very delighted. Yeah.
and I will say, Dr. Swami, if you have any query, Dr. Swami, are you there? Okay.
Maybe out for lunch.
Okay. Thank you.
Thank you so much, sir. Thank you to everybody. Thank you so much.
Hello, Doctor. Hello. Hello.
Dr. Swami.
Let's move to the next poster, maybe.
Okay. Dr. Kohli, next one.
Okay. Next, next we have Dr. U. Bhagwati Ammal.
Yes, sir. I'm here, sir. Good afternoon, sir.
Good afternoon. Ajit, please share her presentation.
Sir, can you able to share my presentation, sir?
You want to share your own? Okay.
Yes,
Please do
sir.
that.
Actually, I'm in outside.
Dr. Kohli, you share it.
Okay.
Dr. Ammal is very active scientist, and she has been actively participating in our activities.
Yes, sir. Yes, sir. I'm continuously attending the webinar and other things, sir.
Get good research from your side. You are working with Dr. Pradeep Day also, no?
Yes, sir. He is our project coordinator, sir.
Okay.
Okay.
Please.
Good afternoon, sir. Respected chairman and co-chairman, thank you for the opportunity given to me, sir. Actually, my work was STCR IPNS based fertilizer prescription for chili crop in insecticide. Let me introduce myself. I am Dr. Bhagwati Ammal, professor from UT of Pondicherry. Last 10 years, actually, we are working in the scheme, that is STCR scheme. As a project in charge, I am now presenting the fertilizer prescription equation for chili crop. The important thing is the entire experiment was raised in farmer's field only. Okay, so with the knowledge of him only, all the things are carried out. So all the fertilizers and menus, whatever the thing we are doing in the field. So after the getting concurrence from him, we conducted the experiment, sir. Actually, this experiment was based on the targeted yield approach given by Ramamurthy et al. 1967. Here the important thing is we are going to derive the the nutrient contribution from soil, then nutrient contribution from fertilizer, then nutrient contribution from organic manures. So based on this, we can have some idea. Then we can apply or we can give the recommendation to the crop. Okay, so the main objective of this experiment is to optimize the fertilizer, nitrogen, phosphorus, and potassium requirement for optimum yield. for the specified yield target. That is a very important thing. That is whatever the fertilizer requirement or schedule, if you are given to the farmers, so the farmers are much concerned about the yield, economic yield and the targeted yield. So that parameters we are considering here, that is we are taking the soil test value and we are also taking the, that is target yield for the particular crop. So the target yield was fixed based on the result of the experiments. Then coming to the materials and methods, how the experiment was carried out means, so minimum half acre is required for carrying out this experiment. So initially the exhaust crop, especially maize crop, was raised in 3 strips. Then in one field itself we are creating the fertility gradient, low, medium and high. So it was called as strip 1, strip 2 and strip 3. So we are applying the fertilizer. So the nitrogen requirement, that is requirement based on maize, then phosphorus and potassium based on the fixing capacity of the soil. Then after the harvest of the exhaust crop, the test crop may be raised. Here the chili crop was raised in the farmer's field. So the totally the field is divided into 72 plots. Then it was carried out with 4 levels of nitrogen, then 4 levels of phosphorus and 4 levels of potassium. With farmyard manure, 3 levels, 0, 12.5 and 25 tons per hectare. So the outcome of the research is, so due to want of place, actually I have not given the fertilizer prescription equation, but we have developed the fertilizer prescription equation. So everything in its computer program. So when we feed the strip 1, strip 2 and strip 3 data, it will give the, that is prescription for the particular chili crop. Then based on it should be, after this it should be verified to the farmer's field. Here also I have posted one poster, that is a lady farmer, in her field only I have raised this chili crop. Then in the table I have given only the part of the project work, that is nutrient requirement, which was actually expressed in kg per quintal. That is to produce 1 quintal of chili, how much kg of nutrient should be applied. So the formula for to calculate the nutrient requirement is total uptake of nitrogen by yield. This is for nitrogen, same as phosphorus and potassium. The second one is percent contribution from the soil. It was denoted as CS. So how it is calculated means total uptake of nutrients in control plot divided by soil test value for that plot into 100. Likewise, for nitrogen and phosphorus and potassium, it is calculated. Then percent contribution from fertilizer, it is denoted as CF. It is calculated based on the, that is total uptake in a treated plot minus soil test value into average CS divided by how much fertilizer we are applying into 100. Same time we are also calculating the how much contribution from organic manure. It is denoted as CFIM. So based on this we have calculated and it was presented in the table. So coming to the percent contribution, the contribution for phosphorus is more when compared to nitrogen and potassium. But in the case of fertilizer, the contribution more in the case of potassium than nitrogen and phosphorus. Similarly, the contribution from farmyard manure also more in the case of potassium. Then after having this that is calculation, we have developed the fertilizer prescription equation. Suppose if the farmer wants to raise the crop in the same soil series, we cannot extrapolate to other soil series. So if he is having the available soil test value, nitrogen, phosphorus and potassium, then we can give the fertilizer recommendation. That is one part. Another part is, suppose if we want to, that is if the yield target may be, for example, in chili we got up to 25 tons per hectare. Suppose if the farmer wants to get the yield only 20 tons means we can give the fertilizer recommendation. So accordingly, we can give the recommendation. In TNA also, they have developed one, that is software. It is called as Decipher. In the Decipher, what we are going to do is we can incorporate all the reading, nitrogen, phosphorus, potassium. Then we can ask the farmer also. That is, for example, for getting 25 tons, if he want to spend 6,000, suppose if the farmer is telling I'm having only 4,000 means, so we can give the value as 4,000, then it will give the value. So what is the difference between blanket recommendation and this recommendation means it will give the balanced supply of nitrogen, phosphorus and potassium. So what is the major difference between the blanket recommendation and STCR recommendation is, for example, if you take phosphorus, okay, the Olsen phosphorus, the medium level is 11 to 22 kg per hectare. So if the soil test value is 10 means, in case of blanket recommendation, it is considered to be low, then we have to apply more amount of fertilizer. Suppose if the value is more, that is 12. So what is the difference in 2 kg? So for each and every soil test value, we can give the fertilizer recommendation to the farmer, provided we should know the available soil test value for N, P and K. Then apart from this, we should also taken care of whether the soil is acid soil or alkali soil, or it is having any micronutrient deficiency means, first we should take care of all the deficiency, then we can go for the fertilizer recommendation. Then only the validation will be, that is plus or minus according to Velayutham et al. What he told us, that is plus or minus 10% yield obtained is actually the good fertilizer prescription equation. So by applying this organic manure, we can also reduce the fertilizer dose. For example, if we applied, that is 12.5 tons per hectare, we can reduce 49 kg of nitrogen, 26 kg of phosphorus and 37 kg of K2O. Okay, so we can taken care of both soil health and adding proper organic manure also. So balanced application of fertilizer with target yield concept is nowadays it is being a World Soil Day. This project actually will that is address the problem of soil health and other things. Thank you, sir. Thank you, sir. That is the end of my presentation, sir.
Yeah, thank you, Dr. Bhagwati Amal.
Yes, sir. Yes.
The presentation is open for discussion. Anybody has any questions, please? You're welcome.
Dr. Amal.
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
What was the pH of the soil in which the experiment was conducted?
So here the pH was neutral, sir, 7.2. Hello, sir.
About 7.2 or 7.5?
7.2, sir. 7.2.
Okay, 7.2. What was the NPK status?
In my experiment, sir. So actually it is low in nitrogen, sir. Low in nitrogen, sir.
Mm-hmm.
Actually, actually I have projected for fertilizer prescription equation, sir. But when we are giving the recommendation, we have to know the available soil test value for the particular field, sir. We can extrapolate to low to high level. Whatever may be the value, we can extrapolate, sir.
Means STCR is basically targeting the nutrient to be in high category?
No, sir. No, sir. Not at all, sir. Not at all, sir. We can, so here, basic exhaust crop raised was with the 3 levels of fertility gradients, sir, low, medium, and high. So based on the response only, the prescription equation was developed, sir.
Actually, going up to 180 kg nitrogen and 120 kg potash.
Okay, sir.
As your levels are saying that you have tried for 0, 30,
Yes, sir.
60, 120
Yes, sir.
for potassium,
Yes, sir.
and 120 and 180 kgs for nitrogen.
Okay.
I think it is on very much higher side. If your soils may be containing it.
Actually, I have tried with the 4 levels, sir, but maximum yield was up because want of space I have not given the value, but the maximum yield was obtained with that is 120, then 80, then 80.
This is my query.
Okay.
If you are applying 180 kg of nitrogen,
Okay, sir.
so was there any negative effect on yield, and if that, what was that and why?
So actually, I won't find there is no negative effect, sir, actually.
But you are saying that highest quantity of highest yield was obtained in 120 kg.
Yes, sir. Actually,
in 180 it got reduced.
No, sir. Actually, first it was tried with the 4 levels of NPK, sir. Then we have to know in which that is level we are going to achieve the maximum yield. That is yield in particular crops, sir, in test crop experiment.
Maximum yield in quantity, or you have some other parameters also, like means economics, or you say you have considered economics as one of the components, or optimum yield we say, which involves say the yield was not economically sound at 180 kg, or the quantity was also means say if you get the yield in 120 kg.
Yeah, sir. Actually, the same. There won't be any statistical difference between 120 and 180, sir, actually.
Okay.
But both are
The
actually
numerical values were higher, na?
Yes, sir. Sir?
The numerical value was higher, but there was not statistical difference.
Yeah, sir. Yeah, sir. Yeah, sir.
And the similar was found in case of potassium also.
Yes, sir. Actually, one higher level means, suppose if the soil is having any problem means, it may give response up to the highest level, sir. For that purpose only one higher level is fixed in the experiment, sir.
Means one higher level, how we can say 180 is a higher level? We can
No,
say...
sir. Actually, normally for chili, this is a chili hybrid, sir. So normal recommendation for normal crop production, they have given as 125, then 60, then 60, sir. Actually, that is the recommendation for hybrid in our place, sir. Actually, so we should know that is the blanket recommendation followed here. So I have fixed one lower dose and one higher dose. So based on this one, sir, that the dose was fixed, sir.
Okay.
Okay, sir.
Economics was also considered or not?
But in the test crop, the economics was not considered, sir. But it should be verified. Again, it should be verified in 4 or 5 trials in farmers field, sir. There we have to go for BC ratio and economics, sir. So
and
it is a base. It is the base for next thing, sir. Next one.
Yes. And it should also be replicated in different areas, also not a single farmer or single chunk of land. Otherwise, your prescription will remain confined only for that farmer or that field only. Otherwise, if you have replicated it in say nearby areas having similar insectisodes, then I think more.
Yes, sir. Yes, sir.
Yes.
Actually, it should be verified in 4 or 5 places. Actually,
Yes,
we are
yes.
developing equation in one field, sir. Actually, it should be verified in some other field with the same soil series, sir.
Yes, before recommending something, multi-locational trials are
Yes, sir. Yes, sir.
essentially needed. Yes, yes.
Multi-location trial,
Okay.
then FLD we have to conduct. Then only it should be given to the department, sir. Department.
Does this nitrogen, higher dose of nitrogen also, as I have seen in the literature many times, the quality of the produce also gets deteriorated. Maybe the quantity enhanced, but
Yes,
the
sir.
pungency of the chili or say some quality aspect can be harmed with higher nitrogen application.
Yes, sir.
So
Actually, we have
that
also noticed that is capsaicin content is actually not much actually with a higher level of nitrogen, the pungency capsaicin content is actually decreased. I agree your point, sir.
Yes. Okay, Dr. Bhardwaj.
Yeah, sorry. Yes. So we have, yeah, thank you so much. Nice presentation, nice poster, Dr. Amal. I have this question in my mind. This is a very old approach, time-tested approach, soil test crop response. Trials are going on throughout the country for a long, long time.
Yes, sir.
Do you think in the present context, this approach or in other words, the approach which is based on removal of nutrients by the plants,
yes, sir,
approach based on that is better than soil test crop response approach?
But actually, even though it is conducted for 40 to 50 years, sir, still in most of the places we are following only the blanket recommendation, sir.
That's true.
Yeah, actually, we are actually in Pondicherry. We are conducting all the things in front of the farmers. We are conducting all the things, sir. But still, they are having some hesitation to follow this fertilizer prescription, sir. Still, they are following the blanket recommendation only. Actually, if you are telling that is we have to reduce the dose to 50 kg means the farmers are not willing to reduce the fertilizer. They thought that they are going to get the poor yield. Likewise, their interpretation is like this, sir, actually. So we have to change that is their behavior, sir. Then actually we are having lot of things in the paper and abstract and all other things. But when we are going to the field, actually most of our findings are in the paper only. That is my thought, sir.
Yeah, I'm asking this in context to something like, we are following new approaches in nitrogen management. For example, recently IFCO has nano nitrogen release.
Yes, sir.
Which is foliar application of nutrients, so that when we apply the granules, there's a lot of losses.
Yes,
and
sir.
To the extent of 60%, we lose the nitrogen fertilizers to the groundwaters or runoff, right?
Yes, sir.
So if we are moving towards foliar fertilizer applications, country, do you think we should also change this approach of soil test crop response based to a hybrid approach where we look at the crop removals and maybe lesser of soil test based, because ultimately we will have to apply the foliar fertilizers to the plants directly, right?
Yes, sir.
The role of soil will be becoming less and less.
Yes, less and less.
Do you
Yes,
think
sir.
it will happen?
But actually, the farmers are actually, I think they are not at all accepting our foliar spray and other things, sir. That is the main thing, because they are our clients. So whatever we are actually any findings. So if it is in the field only, our findings is 100% success. But we are having more ideas, sir. But actually, even with application of this fertilizer and all other things, no. So suppose we are telling you can go for split application and other things. That alone is actually not at all accepted by some of the farmers, sir. Actually.
That's true. That is true. Farmers will not accept easily until they see a big change.
Yes, sir. Yes, sir.
Yes. So all the best. It's a good work. Yes. Soil test crop response is a good approach.
Yes, sir.
I guess there would be more hybrid.
Amal, in which season is chili actually grown there?
Sir?
In which season do you grow chili in Puducherry?
Which? In which?
In which season?
Season. Actually, it is in. It is raised in the month of January, sir. January. So here, November, December, we are getting the northeast monsoon, sir. Actually. Sir. Hello, sir.
Yeah. What other crop? What other crops are the farmers growing in the rainy season or the kharif season?
Rainy season, entire thing is paddy, sir. Paddy here. Then after that only they are going for either bendi.
Actually, this should be customized to the cropping system also.
So here we are following rice pulse actually. Rice pulse, sir.
No, that is okay. But with chili, what rotation is there? Whatever farmers are growing chilies, what is their crop rotation? What is their cropping system?
Their crop rotation, sir. First cotton crop, sir. Cotton, then chili, or they are following some flower crops, then chili, sir. Likewise, they are following. Hello, sir.
So this has got cotton crops, so it should be synchronized with that system also. Because
so according to the release of
residual
water,
nutrients for that crop will also be used. And if you just
no, sir. Actually, this is a test crop only, sir. Test crop only, sir. So after their cropping system is entirely based on the release of water, sir. Actually, release of water.
Release
of water actually. So release of Kaveri water, sir. Kaveri water. So based on the irrigation facilities, they decide.
Rabi season, depending on the release of Kaveri water.
Yes, sir. Based on the release of water, they decide.
They decide the cropping system.
Yes, sir. Yes, sir.
If not chili, then what?
Bendi.
If not chili, then what?
Sir, bendi crop. They will raise.
That does not need water?
Sir?
No, but that does not need water. Vegetables, they need water.
No, not that is actually rice requires more water, sir. So first crop, if they receive water.
No, but you are talking of the Rabi season.
So here
you are talking of the winter season.
Sir, actually, here we are receiving the northeast monsoon October, November, December, sir. Actually, at that time it is very difficult to raise the vegetable crops,
yes.
Sir. Then during January, after January 15th, after Pongal only.
So at that time, what is the crop in the field? Cotton. So what is the crop in the field in December, January?
Cotton, pulse,
November, December.
Vegetables. Cotton, pulse, vegetables.
Yeah, okay. So that is done. Now what I'm saying is that if you have a cotton crop in rotation with chilies, your system should take into account whatever residual nutrients are there after the cotton crop.
Yes, sir. Now we are immediately we are not at all raising the chili crop, sir. After the crop, first we are raising the maize crop as an exhaust crop. Then only we are going to raise the chili crop, sir. So immediately after the crop, we won't raise. First the exhaust crop, then chili. The exhaust crop maize followed by chili. Likewise, the cropping pattern is followed here, sir.
If not chili, then what? If not chili, then what?
Sir, actually, here we are following rice pulse. Actually, Hello, sir.
Okay then.
Okay, sir.
Are there anybody else has any questions? Okay, so now let's move to the second last presentation of this session by Ayushi Jha.
Thank you, sir. Thank you.
Ajit, can you please load her presentation? Thank you, Dr. Bhagwati Amman.
Yes, sir.
Ajit, please load her presentation.
Very good afternoon to all. I am here presenting my MTech research work that is on adaptation of subsurface drip irrigation on wheat crop. Subsurface drip irrigation for efficient utilization of fertilizer and water in wheat crop under Punjab condition. So we all are very well aware that wheat is a worldwide staple food, and its consumption is increasing with the increase in population, but its production is not increasing to that level. So for increasing the production or the productivity, we can adopt the efficient system, which is micro irrigation system, which will provide an efficient utilization of water as well as fertilizer. And under micro irrigation system, I have adopted subsurface drip irrigation system for my work, which is one of the most efficient system in terms of concise application of water as well as fertilizer. The objectives of my work are to study the fertilizer uptake efficiency and to develop an fertigation and irrigation optimum schedule. So it was a 2-year trial with 4 levels of irrigation and 3 level of fertigation, including the control one. Now, under 4 level of irrigation, first one was at 100% ETC, second at 80% ETC, and third at 60% ETC. These 3 were the subsurface levels irrigation treatments, and the fourth one was at 80% ETC, and that was the surface irrigation treatment. Then fertigation levels, first one was at 100% RDF, that is recommended dose of fertilizer. Second one was at 80% RDF. Third one was at 60% RDF, and fourth was the control treatment for comparison purpose. And then many parameters like plant height, then tiller counts, and the 1,000 grain weight, straw grain yield, all those were calculated. At the end, the straw yield was found maximum under the I1F1 treatment, that is at 100% ETC and at 100% RDF. But there was no significant difference between the I2 and I4 level of irrigation. Then the grain yield was maximum under the surface level, that is at 80% ETC in comparison to the subsurface one. But there was a non-significant difference between I1, I2 and F1, F2 level of irrigation as well as fertigation, means either of the combination can be adopted. Then coming on to the concentration of nitrogen, the nitrogen concentration was more in grain as compared to straw. It was about 1.5 times more, and the concentration was decreasing with declination in the level of irrigation as well as fertigation. The concentration in grain compared to straw was 1.5 times higher, and there was a non-significant difference in I1, I2 and I3 level, but they were significantly higher than the I4, that is the surface treatment. Then under the straw, the concentration was higher under the F1 level of fertigation. But there was non-significant difference between F1 and F2 level as well as I1 and I2 level. Again, means in most of the parameters that I've observed, there was non-significant difference between I1, I2 and F1, F2 level. So according to the condition, according to Punjab condition, it could be recommended that 80% ETC, 80% ETC, that is irrigation level at 80%, and 80% RDF, that is fertigation level at 80%, could be adopted for subsurface drip irrigated wheat. Thank you.
Hello. Hello. Hello.
Yes, sir.
Ayushi, I'm Dr. D.K. Chaudhary speaking from Arare Sagwanpur.
Yes, sir. Yes, sir.
I want to know simple things. So what does it mean 80% irrigation level and 80% fertigation level? That means 20% saving of water.
Yes, sir. 20%.
Hello.
Yes, sir. Yes, sir.
So that means your system saves 20% precious water.
Yes, sir.
From the traditional one.
Yes, sir.
Okay. Thank you.
Thank you, sir.
Actually, your objective doesn't reflect that you are calculating water efficiency or something like that. Just to study the fertilizer uptake efficiency. Uptake efficiency means quantity of fertilizer being taken up by the plant. Na?
Yes, sir.
So, which treatment you find is economically viable and saving water? Both the things were common. Which treatment?
The subsurface treatment.
The cost involved you have calculated for
No, sir.
subsurface?
No, sir. Actually, I've calculated individually the uptake in kg per hectare for NPK. All three of them. Also, I've calculated the efficiencies, the recovery efficiency, the physiological efficiencies. But I have not shown here everything. Means I have a huge data collection, but
Physiological, agronomical efficiency may increase, but what about the economics in case of subsurface? Lot of infrastructure is required. Input cost is very high, and so that should also be considered while saying that this treatment was better. Maybe the observation in respect to nutrient efficiency was okay, but when we say that this treatment was very much effective and I recommend it for adoption, that needs economical analysis. Do you
yes, sir.
agree with?
I'll include that, sir. This crop was in rotation with DSR, sir. So I'll include that part, the cost index.
CPCS1 software was used. CPCS1.
Yes, sir. Statistical analysis.
What was the full form of this software application, and who was the developer of this software? Do you know?
Developer. No, sir. Developer, I don't know exactly.
Developer may, if you are from PAU, and whenever you open this software, the first screen itself shows who is the developer and what was the
Yes.
version, and in which year it was developed.
Yes, sir.
This my question was especially to know whether you yourself have used this or you have taken help of some statistician.
No, sir. I've used it myself only, but I means there is Dr. Javed in statistic department. He guided me how to use it, but I used it myself.
It was developed by Dr. Chima. Dr. Chima from statistic department, very old software.
Yes, sir.
And also it needs your vision also while using this software, as you have to develop some parametric line also. No.
Yes, sir. Parametric line was there.
What was the parametric line you have used for your analysis?
So 36220. As I have worked for the combined data for all for the 2 years. That's why it's 36220.
2202 and 2, the last
Sorry,
two.
sir. 440. Sorry, sir. So for 4 level of irrigation and 4 level of fertigation and 3 replication.
And last 0 represent?
So that was I think null.
Null hypothesis.
That is for conversion.
For
Conversion
environment.
from kg per plot to kg per hectare.
Yes, sir.
That is for some unit conversions. That is for some unit conversions.
So you should check it with your statistician, and you should whenever you write something in material method, you should be very much aware of what software you have used, how it worked, and when you are handling yourself, then you should be also clear about that. But as a technical point of view, the straw and grain yield you have mentioned in kg per hectare. Okay.
Yes, sir.
Is this the standard unit for, say, for means these crops, for wheat especially, in kg per hectare?
No, sir, it's in quintal per acre.
Quintal per acre is for farmers, but for say, for if you are in hectare, then these days megagram and all that unit should be considered. Otherwise the numerical values goes very high. Say in your x-axis of this graph, from 2000 to 12,000
Yes,
kgs. So it must be okay. Just application of nitrogen and phosphorus as well as potash. What was the basic dose of these you have applied as a basal?
so for nitrogen, it was applied in split doses and about 123 kg per hectare, and potassium and phosphorus were applied as basal dose. Phosphorus was about 25 kg per acre, and 25 kg per hectare, and potassium was 12 kg per hectare.
12 kg potassium application is not a recommended dose. Huh? Can you hear me?
Hello, yes, sir.
12 kg per hectare is the recommended dose for wheat in your area?
Yes, sir. For surface irrigation, it was recommended. That only I opted.
Through fertigation?
Yes, sir.
What was the means fertilizer used for fertigation in respect to potassium?
Sir, phosphorus pentaoxide, sir, P2O5.
For phosphorus. And for
Yes, sir.
potassium?
Sir, it was murate of potash.
Murate of potash is available?
Yes, sir.
And no application of zinc or other micronutrient?
No, sir.
It was a MSc research or a PhD?
Sir, it's MSc.
Being a master student of College of Agriculture Engineering, most of the things mentioned in this poster relates to either soil or agronomical. Okay.
Yes,
What means your input as an agriculture engineer do you think in this presentation? Modification in surface, subsurface system, or is needed or not?
Yes, sir. Modification, I suppose it's needed, but many things I need to add to conclude to a point like cost index and all.
What is about adaptation that you have worked here? What adaptations have you worked for here? Apart from modifying the levels of irrigation or nitrogen application, what adaptations have you worked on for this subsurface drip irrigation? Ayushi, can you hear me? Could be some engineering adaptations or something. Have you worked on any adaptations? Ayushi, are you there? Ayushi, can you hear me?
Competition.
Sir, am I audible?
Ayushi?
Yes, sir. Sorry, sir.
Hello,
Sir, am I audible?
Ayushi, are you audible?
Yes, sir.
Are you there?
Yes, sir. Yes, sir. Sorry, sir.
I
There
was
was a
asking,
network problem.
yeah, you
Yes,
are
sir.
audible. I was asking what adaptations have you?
So what adaptation, sir? I can't hear.
What adaptations have you tried? Were there any engineering adaptations? What adaptations with respect to laying down of that subsurface? Respect to laying down of your subsurface drip, adaptations in subsurface drip irrigation, modifying or altering the dose. You get what I'm saying?
Sir, I'm not getting the question.
Your title is adaptations in subsurface drip irrigation. I'm asking what adaptations have you made in the irrigation system?
Sir, what all I have adapted for installing the system, sir?
Yeah.
So like spacing and all.
What are specifically?
Sir, laterals were laid at 20 centimeter depth.
Okay, I think you have some network issue. That's okay. Let's move on to the last presentation for this session. The last presentation for this Olympiad by Megha Kaviraj.
Yes, sir, I am here.
Megha, are you there?
Yes, sir.
Okay, Ajit, please load her presentation and you can start.
Okay, sir. To the full screen, please. Okay, thank you so much. Good afternoon to one and all presenting here.
I am Megha Kaviraj, SRF and PhD scholar from ICR National Rice Research Institute, Cuttack, Odisha. And I am very thankful to judging and organizing committee for selecting me for the final round of Olympiad. Now coming to my poster. So my today's topic is coupling effects of vitamin C and Azotobacter coccocultum on plant growth promotions in rice under water deficit stress. Sir, as we all know that rice cultivations requires higher amount of water and in fertilizers. So drought is one of the major factors which decline the grain yields. Basically, droughts creates an oxidative environment and which produces the reactive oxygen species, that is ROS, and damages finally the cells. And another aspect is excessive use of chemical fertilizers not only hamper but it deteriorates the soil health. Thereby, biofertilizer is the better options. But the most of the use biofertilizers not working properly because they are exposed with several environmental factors which ultimately causing the diet of the organisms. So before I coming to my hypothesis and objective, I would like to briefly explain the previous work behind this present study. So I have taken that Azotobacter coccocultum AVI2 as it is endophytic diazotrophic in nature, and endophytic diazotrophic capability is already established by the fluorescence resonance emissions techniques in planta. We published that one, and it had the multiple PGP traits like indole acetic acid productions, ammonia productions, HCN, siderophore, and so on. So we have taken that organisms as a model organisms for our study, and we given different doses of H2O2 to creating the oxidative stress from 0 to 5.5 millimolar of doses, and we found that more than 2.5 millimolar of H2O2 killed that bacteria. And once we used that 1 ppm of ascorbic acids, or commonly called as vitamin C, not only the growth occurred, but under that higher concentrations of H2O2, but also enhances its plant growth promotions and end fixing capability. So keeping those things in our mind, and we already that data are published just previously in Plant Physiology and Biochemistry journal, and keeping those things in our mind, and after reviewing so much extensive literature, we are not unable to find any single literature that combinatorial effect of that bacteria and the vitamin C, that too under water deficit stress conditions, can enhance the rice growth promotions. We found plenty of research on solo applications of bacteria or solo applications of ascorbic acid for the plant growth promotions. So we hypothesize and we planned an experiment to check the combinatorial effect of the bacteria and ascorbic acid on the rice growth promotions. So we planned treatments like 1 flooded control and 4 moisture stress conditions. In 4 moisture stress conditions, and we kept moisture stress conditions alone, and moisture stress conditions alone with bacteria, and moisture stress alone with ascorbic acid, because we have to check also does ascorbic acids alone have any effect or not, and then finally that combinatorial effect of bacteria and ascorbic acid under stress conditions. And we have taken the 4 rice cultivars, 2 drought susceptible cultivars, that is IR64 and Naveen, and 2 drought tolerant cultivars, that is Ankit and Satyabhama. And we conducted an pot experiment where we estimate their oxidative stress enzymes, their antioxidant stress enzymes, and we check that photosynthetic ability, their photosystem performance, their NIFE gene quantifications. And I coming to my result one by one. As we can see that in our first table, in my first table, you can, as we all know, that is proline is the oxidative stress indicators. When stress is much more, that proline surge is very high. But once we use that combinatorial applications, that proline content is normalized, and that antioxidant enzymes, they are also normalizes under the combinatorial applications compared to the moisture stress conditions. And if we coming to my figure 1 here, I show you that the photosystem efficiency, as we did the new technique, chlorophyll fluorescence imaging technique. Actually, photosynthetic efficiency are directly related to the plant growth, and for base and the particularly the photosystem 2 are very sensitive to the oxidative or any kind of stress. So from that photosynthetic efficiency, we can tell that yes, that plant is either imposed with any oxidative any stress or not. So this the figure 1 graph showing that fluorescence chlorophyll imaging data of photosystem 2. Here you can see the green curve line showing the upper positions, which show that electron transfer rate from PS2 plus 2.1 to B, how the electron is smoothly shifting from plus 2.1 A to B, and finally the reaction center is closed and fluorescence coming in very high quantity. So and the red line which are in lower in positions showing in moisture stress conditions, showing that photosystem efficiency are not good in the stress conditions. So from that result we can depict it that photosystem is photosystem 2 and electron transport rate, photochemical quenching, non-photochemical quenching, all the parameters related with the chlorophyll are in very normalized or good conditions under the combinatorial effect of that applications of that bacteria and ascorbic acid. And here I also did the NIFE gene quantifications. Either we have checked that that nitrogen fixing gene copy number, how much it is altered or how much is there in that root of the plant. So we did the gene quantifications through RT-PCR, and in figure 2 I have shown that agronomical data, that is the plant height and the grain yield. And grain yield, from grain yield we can got that 34 and 44% increased in IR64 and Nabin variety under that combinatorial effect compared to the moisture stress alone, and 39% and 36% increased grain yield under the combinatorial effect compared to the moisture stress conditions. And now finally from that my whole data measurement, I did the PCA analysis, where you can see very nicely that the green circles or eclipses, you can see that clubbed in a single group, those are from belonging from the drought susceptible cultivars, and the pink one, that two circles are there which is belong grouped in a single group, and they are belong from the tolerant cultivars based on the following variables, based on the measured variables. So from the overall study, from the data of chlorophyll fluorescence imaging, the chlorophyll content, antioxidant enzyme assays, and oxidative stress enzyme, and agronomical characteristic grain yields, from overall data, and then obviously that nitrogen fixing gene copy number inside the root, it is very much important for the nitrogen. So we can depict it that AVI2 and vitamin C formulations in future may be used as novel formulations to alleviate drought stress as well as enhances nutrient use efficiency in rice. So as we are quite familiar with that cutting edge research like gene editing, genetic engineering, gene mining, mutations of targeted gene to enhances the any targeted gene functions, and by CRISPR. And recently we are familiar with CRISPR-Cas9 technique, TALENT technique to enhances the genetical functions. But those things are very expensive, and maybe many ethical issues maybe arises. But why not, if we are going to like this simple and much easier technique, if we getting that promising such our targets, so why not we are going for that. In my poster you can see an animated picture where my bacteria Azotobacter phococcum is still smiling, as although it is exposed with that oxidative stress condition, because as it is drinking the ascorbic acid juices. So I recommend that AVI2 and vitamin C formulations may be the novel formulations to alleviate the drought stress. Thank you, sir. I'm done. Hello. Sir, I have finished my presentations.
Thank you, Megha.
Thank you so much, sir.
The presentation is open for discussion. The presentation is now open for discussion. Yes, Dr. Aura, are you there? Dr. Nilan?
Hello, hello, ma'am. I have a question.
Yes, please.
Ma'am, you can explain the tolerant and resistant variety difference actually, and how can this adapt the plant?
Yes, thank you for your questions. Actually, I have taken two rice cultivars from drought tolerant as they are cope up the stress, and thereby I am choosing and with drought tolerant with contrasting cultivars like susceptible. We have IR 64 and Naveen variety, and to check that sensitive varieties are actually hypothesis is sensitive variety is much prone to that oxidative stress, so we can easily compare between two, thereby we choosing the contrasting cultivars. So yes, the mechanism you want to know actually, and the mechanism is when we use that ascorbic acid or vitamin C, it transfer it when it oxidizes it form the dehydroascorbate or DHA, then it is transported to the from the root to the mitochondria or endoplasmic reticulum through the transporter channel of GLUT10 or GLUT11. Actually, I have recently read that GLUT10 or GLUT11, those are the glucose transporter channel, and those are very sensitive to uptake that ascorbate, dehydroascorbate inside the mitochondria or endoplasmic reticulum. So once that form of ascorbic acid DHA is uptake inside the mitochondria, actually mitochondria is the more prone to where the most of the reactive oxygen species are formed. So mitochondria is very prone to be damaged first by their genome and membrane. So we have to trigger the mitochondria first. So that dehydrogenase ascorbate DHA when enters into the mitochondria, it then reduce back to ascorbic acid, as today's my guide also presented the same. He have very nicely presented. So the reductions back to ascorbic acid and through thiols and glutathiones. By this way, it protects that mitochondrial genome and membrane. So you can say by this way that scavenging mechanism is going on.
Yes, ma'am.
Got
Ma'am,
it?
one more question, ma'am.
Yes.
Yes, you ask.
Ma'am, what is the source of these type genes?
What? Hello?
What is the source of this gene?
Actually, I have targeted the NEFA gene. I quantified the NEFA gene because NEFA gene is responsible for the nitrogen fixations. Thereby, my objective was to quantify the NEFA gene inside the root, how gene copy number is altered or not under the stress, or may ascorbic acid give some cofactors or coenzyme to alleviate or accentuating their gene copy number and so on. Thereby, I'm telling that genetic engineering and gene editing is very popular nowadays. But if we given such some kind of simple techniques, then we got the similar kind of result. Then why we can't go? Why we don't looking for that one also?
Okay.
What is the
Okay.
What is the particular source? ठीक है. Okay, sir. Thank you.
Megha,
Yes, sir.
your introduction says that rise endophytic species have potential to save chemical fertilizer.
Yes, sir.
Is Azotobacter a endophyte?
Yes, sir. Azotobacter crococum, the strain AVI2 is the endophytic in nature. We said established that it colonized in the root cortex, and we published also in Planta Journal through sir freight analysis, fluorescence resonance emission technique.
Yes, yes, they can move to the as endophyte, but they are basically the soil organism.
Yes, sir.
And you have to apply in soil. Either are you going to apply, if you recommend it to use, are you going to apply through foliar application?
Sir, we are not thinking about that. If you are suggesting that, we also think
No,
like this way also.
if it is an endophyte, then
Yes, sir.
then its spray will be beneficial to
Yes.
immediately
Yes,
go
sir.
into
Yes, sir.
the
Yes,
stomata.
sir.
But
Yeah.
if you are, and the second point is,
yes, sir.
why you are designating ascorbic acid as vitamin C? We
Sir,
know that both
yes,
are
sir.
same thing, but we
Yes, sir.
know
Yes. Yes,
it
because
should be
vitamin C
ascorbic
is much
acid.
familiar.
No,
That's
no,
why I am telling.
no, not familiar. It has become very much familiar during this
Yes.
COVID. Then you have designated.
Yes, sir. Yes, sir.
Otherwise,
Very
it
much
is
true.
not. But ascorbic acid is a chemical. Okay.
Yes, sir. Yes, sir.
And when
Chemical.
it is used by human to get immunity, or say some
Yes, sir.
as a
Anti-aging.
as a
Yes, sir.
vitamin. Okay.
Anti-aging factor also.
Yes, yes. Anti-aging is vitamin E basically, but okay, vitamin C also have some potential.
Yes,
But
sir.
but here, whenever you are going for some study related to a plant,
Yes,
and
sir.
it is not a vitamin for plant. Okay.
Yes.
Is it? Is it a vitamin for plant?
Sir, as it is, sir, we can not vitamin, but some cofactors which can accelerating the
But my
efficiency.
concern is
Yes, sir.
we should avoid using the
Okay, sir.
means these nutritional terms are basically changed with respect to the particular use.
Yes, sir.
Otherwise, you must be knowing that salicylic acid
Yes, sir.
is basically a growth enhancer.
Yes, sir.
But on the other hand, salicylic acid is very much useful as a drug for means for fever. Okay.
Yes.
So that doesn't mean ki if we are using salicylic acid, then we write paracetamol, we have applied 2
Yes, sir.
grams per plant. It is not feasible. Na?
Yes,
So,
sir. In our previous paper, we write that ascorbic acid. It is might be not visible our paper, but the title of our project was.
otherwise, if you have written vitamin C,
Yes,
they
sir.
might have rejected it on that ground.
Yes, sir.
Yes, please see, these things are very much critical in respect.
Yes, sir.
Because vitamin C is very popular these days. So designating ascorbic acid as vitamin C is not means. This is my idea. You talk it with Dr. Nayak. He's our senior colleague, and he's a soil chemist basically. So he can give you a good idea. The other things which I have seen is, if this is a drought tolerance elevator,
Yes, sir.
okay, then it must be beneficial in salinity also, so elevating salinity levels.
Sir, maybe we are not actually thinking about that. We are also, we will, sir, think about salinity, about that acidity. It may be work or not.
Acidity
We should, sir,
is
check
a
that one
different thing.
also.
Acidity has different mechanism, but salinity stress is related directly with the moisture stress. Okay.
Okay. Okay, sir. Very good
So,
suggestions you have given, sir.
basically, you are a soil scientist or a genetic
Microbiology.
microbiologist. Okay.
Yes, sir.
So, you have done the isolation AVI2, or you have procured it from somewhere?
No, sir. No, sir. Actually, previously we have isolated more, sir, about 300 isolates from the different parts of the rice plants, like leaf, root, stem, and finally we come up with three based isolates. And among 30, we the Azotobacter coccoculum is the most potent bacteria, and we further going with that bacteria also. Actually, this isolates is ours.
What was the media used for isolating
Sir,
these?
Azotobacter for Jensen, and to check its sir, one character is very important. You can easily check by naked eye that it produces the brown color pigmentations on SB mannitol agar. So we can use both SB mannitol agar entry media and also the Jensen agar.
Okay, okay. So temperature was?
Temperature was 25 degree for
Okay.
incubations.
For incubation, 25 degree.
Yes, sir.
And have you find out the thermal death point also for these temperature tolerance?
Temperature tolerance, no, sir, I have
Because
not.
when you are claiming for drought stress is very much related with the temperature stress.
Yes,
Okay.
sir, but its nature is endophytic, na, so sir, that much that bacteria is exposed with that much
Okay.
environment
So,
also.
okay, okay. Direct, it is not
Yes,
getting
yes,
with
sir.
the direct.
Directly it is not getting, na.
Okay, thank you.
Thank you,
Any
sir.
query from Dr. Nile? Dr. Nile, are you there? Okay, thank you.
Thank
We
you
will
so
move
much
on
for
to
your
the next.
valuable suggestions. I am also looking for your suggestions like NaCl also.
Okay.
Thank you
Thank
so
you.
much.
Thank you. Dr. Kohli, next.
Thank you, Megha.
Thank you, sir.
So this was the last presentation of this session.
Okay, then
This brings us to the
made
end
this
of this
meeting.
Olympiad. Maybe the judges can take some time to collect the results, and then we can start preparing for a short valedictory session. All good things have to end.
So what is the point?
Also.
So the presenters should wait for the valedictory session in which the judges will give their feedback on this finale,
Yeah.
and then we will announce the results of both the soil resource awareness quiz as well as the presentations of e-posters. Okay. So is Dr. Suraj Bhan available? Dr. Kohli, please check.
Yes, he is there, sir.
Hello.
Good evening, sir.
Good evening.
Good evening, sir.
अभी शुरू करते हैं सर वैलिडिक्टरी बस ये प्रोग्राम.
अरे डॉक्टर सुन नहीं पा रहा हूँ बोलो. हाँ
अभी करते हैं वैलिडिक्टरी.
क्या कह रहे हैं डॉक्टर?
मैं कह रहा हूँ सर कि अभी वैलिडिक्टरी शुरू करने जा रहे हैं.
हाँ करिए, मैं तैयार बैठा हूँ.
हाँ हाँ ठीक है सर, ठीक है.
हम तैयार बैठे हैं. हम भी.
आप ही चीफ गेस्ट हैं वैलिडिक्टरी के.
तो कितने मिनट मिलेगा टाइम हमको?
फ्री है सर, फ्री टाइम है आपके पास.
अच्छा वेरी गुड. वो निकाल ले सुबह बोल.
So good evening, everybody. We are at the end of this program. The formal valedictory function, where we will be getting a brief report of what was deliberated over the last 2 days, and then the way forward, advice, blessings of our president, and obviously we'll be getting the results of various quiz, various poster presentations in various sessions. So without wasting much time, I would just like to invite the chairman for this session, Dr. Suraj Khan, who is the president of our society, the Soil Conservation Society of India. Co-chairing the session would be Dr. Sanjay Arora. The conveners for this session will be Dr. Pankaj Singh and Dr. YK Singh. Are you there, both of you? Dr. Vipin Kumar and Dr. Geeta Kumari will be the co-conveners.
Yes, sir, I'm here.
Welcome to all of you. Yes, please. I just like to invite one of the organizing secretaries to be there so that the brief report of the program can also be shared. But first of all, I would like to hand over the mic to Dr. Sanjay Arora to Dr. Arora, are you there?
Yes, I am very much here.
So can we start the session?
Yes, you means in this session you will be giving the who will be giving the report, organizing secretary.
Brief report will be given by one of the organizing secretaries, Dr. Shweta Shambhavi. Can I invite her?
Okay. No, I first.
Shweta, please present a brief. Yeah.
Okay. On behalf of Soil Conservation Society and on behalf of the participants, I welcome Dr. Suraj Bhan, President of Soil Conservation Society, and I also hereby express my gratitude to Dr. Kohli, Dr. Vikas Sharma, Dr. Nile Bora, and many of colleagues, the organizing secretary as well as co-organizing secretaries who have actively participated and arranged the things to conduct the e-poster sessions as well as the lecture sessions for celebrating the World Soil Day. So, and Dr. Suraj Bhan was very much means cooperative during these 2 days, irrespective of his some health issues sometimes, but he has managed things very well. And today we have in the morning one program from Soil Conservation Society, lecture by Dr. A.K. Sikka. That was also to commemorate the World Soil Day. So with this 2 days program of National E-Poster Olympiad is complete. So I request the concerned official to present the detailed report of the 2 days sessions. Shweta, Dr. Shweta.
Yes, sir.
Dr. Shweta Shambhavi, please.
Yes, sir. A very warm good afternoon to the chairman of this session, Dr. Suraj Bhan, and co-chairman Dr. Sanjay Arora, along with organizers and participants. Myself, Dr. Shweta Shambhavi, one of the co-organizing secretaries, is here to present the brief highlights of the different sessions. National E-Posters Olympiad, organized by Soil Conservation Society of India to celebrate World Soil Day 2020 on the theme Soils, Biomes, and Resilience to Climate Change, was inaugurated on 4th December 2020. Dr. A.K. Singh, former TDD NRM, was chief guest on this occasion. While the session was chaired by Dr. Suraj Khan, President of Society, and co-chaired by Dr. Sanjay Arora, Principal Scientist, CSSRI Regional Research Station, Lucknow. Dr. Anshuman Kohli welcomed the dignitaries and participants and briefed them about the Olympiad. Dr. Suraj Bhan, President, briefed about the origin, role, and functions of the society, and also highlighted the achievements of society, especially about the various national and international events organized during this pandemic situation, thereby making society being recognized at the global platform. Dr. Sanjay Arora gave an insight about the history and importance of World Soil Day being celebrated across the globe on 5th December every year. A detailed and informative presentation on scientific poster presentations, the art of making posters interesting and effective, was made by Dr. Vikas Sharma, Professor and Head, Division of Soil Science and Agriculture, Chemistry, SCOS, Jammu. The presentation would serve to be beneficial to both scientists as well as students, as poster making requires art as well as skill. Dr. A.K. Singh, chief guest of the event, talked about the role of microbes and various other soil properties, which plays an important role in maintaining soil quality and soil health. Apart from various other factors, he emphasized on the role of soil microbes in making soil resilient to climate change. He also elaborated the importance of celebrating World Soil Day and why 5th December was chosen by FAO as World Soil Day every year. At last, vote of thanks for this session was proposed by Dr. Manar Ghosh, organizing secretary of this event. On the first day, there were 3 poster sessions, while 1 e-poster session and 1 plenary session was organized on second day, that is today, along with Olympiad finale and soil resource awareness quiz. The Olympiad finale was consisted of different presentations which were selected from the other 4 sessions. Now, regarding session 1, the session 1 was chaired by Dr. Jagdish Prasad and co-chaired by Dr. Sushama Sudhishri. There were total 14 presentations made in this session. Almost all the presentations were based on the themes such as climate change, carbon sequestration, and soil health. The main highlight of this session was on the role of microbes in mitigating climate change. Session 2, this session was chaired by Dr. Ajay Bhardwaj and co-chaired by Dr. R.S. Yadav. Total 13 presentations were made on different topics under the theme agricultural biomes, diversity, practices, and conservation agriculture. Major highlight was on conservation agriculture, and from most of the presentations, it was inferred that conservation agriculture system appears to be tempering option to accomplish sustainable and intensive crop production. It is helpful in carbon sequestration and reduces cost of production. The third session was chaired by Dr. Pradeep K and co-chaired by Dr. G.J. Patel. The session focused on the presentations on soil management, functions, and organic agriculture. There were 8 presentations on different aspects of organic amendments along with crop diversification. The major highlight of this session was first, microbial consortium need to be explored for enhancing productivity and soil health. Second, integrated nutrient management offers sustainable option to mitigate soil pollution. Third, organic farming need to be redefined based on inputs and production of crops. And last, crop and input diversification will be the key to improve soil biodiversity. In session 4, total 9 presentations were made by different speakers. Presentations made were on different themes like INM on carbon sequestration, livestock, water productivity enhancement, coupling effect of vitamin C, and azotobacter on growth promotion in rice, analysis of groundwater, impact of wastewater irrigation, water use efficiency in cherry production, delineation of hydrological response, adoption of drip irrigation in wheat in plains of Punjab. All the presentations were finally delivered by speakers. Chairman Dr. Hadda, in his final remarks, emphasizes to adopt soil and water conservation methods for reducing soil erosion. He also said that nutrient loss in soil is also a huge problem nowadays, and efforts should be made to develop certain resource conservation modules, especially for hilly areas. Dr. Arya, convener, also highlighted the importance of natural resource management. He said soil loss up to the tune of 70 tons per hectare is posing a real threat to soil health as soil is eroding at an alarming rate. A plenary session to commemorate World Soil Day and its theme on soil biomes was conducted today, and Dr. M.J. Singh, Director PAU Ludhiana, chaired the session, and Dr. N.K. Parikh co-chaired it. 2 learned speakers presented the thematic lectures in this session. One of them was Dr. Dilip Kumar Swain, Professor, Agriculture Systems and Management, IIT Kharagpur, who elaborated his talk on climate change and its role in achieving food security. Through his concepts, he enlightened the participants on how his interventions is working to improve productivity and income of Indian farmers. The other speaker was Dr. Upendra Kumar, scientist, soil science, microbiology at ICAR NRRI Cuttack, who delivered his lecture on microbial diversity in rice-based system and its influence on crop and soil health. In his lecture, Dr. Kumar described about the role of microbes in soil, their function, and importance in improving agricultural productivity. He emphasized on microbial formulations, biofertilizers products, and novel methodologies for enhancing and sustaining production. Apart from different poster sessions, a soil awareness, resource awareness quiz was conducted, which was taken by 55 participants, the results of which will be declared by our co-chairman of this session, Dr. Sanjay Arora. This is a brief report about different sessions which were conducted yesterday and today. Thank you.
Thank you, Dr. Shweta. Now I request the co-chair of this session, Dr. Sanjay Arora, to please share the results of various competitive events of the quiz, the results of various sessions, and the Olympiad finale. I hope you are ready with all the compilations and collations of the last session.
Thank you, Dr. Kohli. Yes, now the moment is for declaring the results of the different sessions, from especially the finale, and as per the results received from different Dr. S.B. Singh, please hello. Hello, am I audible?
You are audible, sir.
Okay, so we have received the results from different chairman, co-chairman of the session, as well as from the jury. We have designated 3 scientists to review the Olympiad finale and compile the results, and on that basis I will be presenting the results. So the best poster for this 2 days event is by Bishun Dev Prasad for arsenic mitigation in rice using microbes, a very significant as well as relevant study and nicely presented. The first runner-up for this Olympiad was Anjani Kumar and his team, and he was having a tie with Ranjan Paul. Ranjan Paul is the first runner-up, and the second runner-up in the poster session was Megha Kaviraj. So we have these 4 as the winners of the Olympiad in finale. However, in different sessions, first, second, and third winners will be given the certificates for their respective sessions, and overall these 4 participants have excellently presented the posters and participated. So I congratulate all the winners, and actually all were the winners because they have very much followed the style and format of presentation and with having good posters. So I congratulate everybody, especially Bhagwati Ammal has presented very nicely, but we have to promote the young participants as well as per the advice of the jury. So I congratulate all of the winners. So we will be announcing the result of soil resource awareness quiz. For the sake of chairman of the session, sir, we have organized a e-quiz. Dr. Kohli was instrumental in designing the quiz, and we have received 55 entries in the quiz program, which was for 10 minutes. It was open for the participants to quickly respond to the questions, and we have got good attempt by 55 participants, and the results were the first winner. We have two winners for the first place, Tajamul Aziz and Sumit So. And Tajamul Aziz got the first position with maximum right answers. The second one is Megha Badhani, the second winner in the quiz, and Surendra Paul, the third winner in the quiz competition. So I congratulate these quiz winners as well as the e-poster winners, and hope we will be organizing such event in future and you will be actively participating, as we want to promote young participation relevant to soil and water management. So this was all from my side to the participants, and congratulations to all the winners. Thank you.
Thank you, Dr. Arora, for announcing the results. The results of the poster sessions, apart from those who had participated in the Olympiad finale, will be declared in due course through emails. And now it's time that we should hear. So I would like to invite Dr. Suraj Bhan on the dais for his concluding remarks and to say some words of advice for the whole organizing team and the young researchers, early career scientists who have participated in this Olympiad for the last 2 days and celebrated the World Soil Day in a befitting manner. Dr. Suraj Bhan, sir, please.
Good evening, all. Respected Dr. A.K. Singh, Secretary NAS, former Vice Chancellor RBS KBA Gwalior, and former DDG NRM ICR, and chief guest of the today's program, Dr. Sanjay Arora, Dr. Anshuman Kohli, co-chairman, conveners, and co-conveners, scientists, students, participants, ladies and gentlemen, to extend my warm wishes on behalf of the Soil Conservation Society of India, New Delhi, I want to convey my heartfelt gratitude to Dr. A.K. Singh for accepting the invitation and grace this occasion. My special thanks are due to Dr. Sanjay Arora, Dr. Anshuman Kohli, who organized this event and made a great success. It is their best efforts who has organized the event in a very short notice. I express my thanks to all members of the society participating or coordinating to organize the event. I would also like to congratulate the winners of ex-poster session and soil resources awareness quiz. World Soil Day is an occasion for both reflection and introspection. In the present situation, where the population is increasing day by day, cities are expanding, requirement of food is increasing, and climate is changing. We urgently need healthy soil. Preserve and protect it over the long term. I urge all of you to double your efforts to support the theme of the World Soil Day 2020. Keep soil alive, protect soil biodiversity, and sustaining healthy ecosystem and human well-being. To improve soil health and soil resources, it is urgently required raising the awareness to conserve soil biodiversity for a food secure future. It is possible by soil conservation and management, soil and water conservation, watershed management, soil health management, land degradation and control, scientific use of water for per drop more crop, grassland management for soil and water conservation, forestry and agroforestry vis-a-vis soil and water conservation, crop and cropping systems for efficient soil and water conservation. Soil is a key natural resource, and soil quality is the integrated effect of management of most of soil properties that determine crop productivity and sustainability. Good soil quality not only produces the good crop yield, but also maintains environmental quality and consequently plant, animals, and human health. A quantitative assessment of soil quality can provide much needed information on the adequacy of the world's soil resource base in relation to food and fiber needs of growing population, continuous cropping without use of any fertilizer, and imbalanced application of fertilizer leads to reduction in the level of soil organic matter in semi-arid region of central India. Unless soil management practices are improved, yield reduction continues and long-term production will be under threat. The inadequate and imbalanced chemical fertilizer use, decline in soil organic matter, and inefficient production. For sustainable production, utilization of farmyard manure and fertilizer is an efficient management to maintain the soil quality. For maintaining soil organic matter, it is necessary to maintain soil organic carbon under intensive cropping system. Soil quality can be assessed by measuring soil attributes or properties that serve as a soil quality indicators. The changes in these indicators signal the changes in soil quality. Declining soil fertility and soil loss are the predominant causes of soil degradation in rainfed region. Undoubtedly, due to water erosion. In fact, the process of water erosion sweeps away the topsoil along with the organic matter and exposes the subsurface horizons, the second major indirect cause of degradation in loss of organic matter by virtue of temperature, mediated fast decomposition owing to high temperature prevailing in these regions. Several other farming practices, such as reckless tillage methods, harvest of every small component of biological produce, and virtually no return of any plant residues back to the soil, burning of the existing residues in the field itself for preparation of the clean seed bag, open grazing, etc., aggravate the process of soil degradation. As a result of several above mentioned regions, soil encounter diversity of constants broadly on account of the physical, chemical, and biological soil quality, and ultimately end up with the poor functional capacity. In order to restore the quality of the degraded soil and to prevent them from further degradation, it is of paramount importance to focus on restorative practices and conservation agriculture practices on long-term basis. If we do not act up soon, the fertility of soil will continue adversely affected at an alarming rate, threatening food supplies and food safety. Thank you very much for patience hearing, and in the end, on my personal behalf, I congratulate organizers and its team members, and as a state, Bihar state chapter of SHI, for doing excellent, successful event. Thank you very much.
Thank you very much, sir, for your words of wisdom and advice and guiding us as you have been doing this forever. I have no words to express gratitude towards you. It's with your blessings and support that we are always able to do things like this. And like all good things, this also has to come to an end. So probably we'll be failing in our duty if we don't just propose a vote of thanks and then convey who has contributed to make this event a very successful one. And probably, you see, this is the first event which I have heard of, which I have seen, in which emphasis was on young presenters through a mode which is often neglected in our country and always considered a second-rate presentation time. we highlighted those sort of presentations, and our all proceedings will be drawn from whatever was deliberated and discussed in the poster presentations. But for time being, I would like to invite Dr. Sanjay Arora to propose a formal vote of thanks, and then we can say goodbye. Thank you. Dr. Arora, please.
थैंक यू डॉक्टर कोहली. अब ये काम भी मुझे ही करना है. डॉक्टर श्री जगतवीर सिंह is not available due to some reason, so I will be proposing vote of thanks on behalf of Soil Conservation Society. So at the outset, I want to thank Dr. Suraj Bhan for his support, cooperation, and guiding us for the organizing this program. Dr. Suraj Bhan has been always eager to initiate activities, organize things, although he's having number of assignments like President of Soil Conservation Society of India as well as President of the International Soil Conservation Organization. So taking both into his hands and moving forward is a very difficult task, but he is capable of doing and taking these challenges. So I am thankful to Dr. Suraj Bhan. And for this 2 days program, I have no words to express my thanks to Dr. Kohli and his team, especially the organizing secretary, Dr. Maina Ghosh, Shweta, and many more, Dr. YK Singh, and many other who are involved from BAU chapter, who actively behind the curtain, they were very much present, and they were doing good things so that the event goes successfully. And I believe that participants should realize what efforts everyone has to put in to organize such events. It takes a lot of time, lot of energy, and so the efforts made by all the team members of the organizers is appreciated, highly appreciated. And also I want to thank Dr. AK Singh, who has formally inaugurated the 2 days program and guided us for taking up these activities. He's also very much busy person. He is secretary of NAS and he's ex-vice chancellor of RVS KVB and DDG NRM. So we are thankful to him for accepting our invitation very shortly, and his close association with the society. Dr. Vikas Sharma, who presented in the inaugural session, and he is also one of the convener of the program and giving tips how to improve the things which we have planned. I am also thankful to chairman and co-chairman of different sessions who have accepted our invitation in a very short notice, and they agreed to convene the session to take the proceedings. Dr. Jagdish Prasad from NBSS LUP, Dr. Ajay Bhardwaj from CSSRI, Dr. Pradeep Dey, Dr. MS Hadda, Dr. Manmohanji Singh, they have accepted to chair the session and very meticulously conducted the proceedings of the session. Dr. Sushma Sudhishri, Dr. RS Yadav, Dr. GJ Patel, Dr. PK Rai, Dr. NK Parikh, Dr. Sanjay Swami, they all were the co-chairmen of the sessions in different sessions. So I am thankful to them also for being part of this event. Conveners have a very important role, although we say that conveners can be anybody be a convener, but no, conveners have the responsibility to put down the proceedings, to arrange the report and send it to the organizers so that a report can be prepared for the overall program. So coordinating different sessions, different presenters in the session, conveners Dr. RP Sharma from NBSS, Dr. Sumita Chandel from PAU, Dr. Mukesh Kumar from IGNOU, Dr. Mahinder Singh, Dr. Ajay Kumar Mishra, Dr. K.K. Yadav, Dr. V.M. Arya, Dr. Amit Mishra, Dr. Vishnu Dev Prasad, Mr. Rajiv Padma Bhushan, Dr. Priyanka Suryavanshi, Dr. Swaraj Kumar Datta, Dr. Pankaj Singh and Dr. Y.K. Singh are with us here in the valedictory session. So their efforts during the session were very crucial, and I am thankful to them also. And the co-conveners having a specified role also gets appreciation and our respect in respect to organizing the program. Dr. Anita Kumari, Dr. Sunil Kumar, Dr. Mr. Binod Kumar Bimal, Dr. Ragni Kumari, Dr. S.C. Paul, Dr. Dev Kumar, Dr. S.K. Gupta, Dr. Kasturi Kasin Bera, Dr. Raj Kishore Kumar, Vipin Kumar and Dr. Geeta Kumari are with us in the valedictory session. So I am thankful to all of them for conducting the proceedings of the session. Also, I want to thank the Dr. Please mute yourself, Rohit Sharma. Please mute. I want to thank SCSI executive members, vice presidents, councillors, secretary, joint secretary, treasurer. They have been instrumental in helping us and organizing the event. Dr. Dilip Kumar Swain, Dr. Upendra Kumar, who were the lead speakers in the plenary session, gets our appreciation because they were the gems during the two sessions to enlighten us about the different aspects on climate change as well as microbial diversity, the major theme of this event. So I am thankful to them also. Dr. Kohli organized the quiz, awareness quiz, and his team has designed the questions. 40 questions were designed, and these things needs also appreciation as to engage different participants and to give them insight about the questions and working on them. So it was very nice that the event was having a poster session, which is very important, and then a quiz program, which was very much liked by the participants. And last but not the least, I want to thank Mr. Bittu, who was there in the SCSI headquarters and working on computer and communicating the things to the president. So thank you all, and hope you will be having a 2020 till 2021. We have to be alert, and as Dr. Suraj Bhan has told, we should double our energy and efforts to protect our soils. Thank you. And I also want to congratulate the winners of the participants who are the winners of the quiz as well as the e-posters. So thank you, Dr. Kohli. So we have to, if I have left somebody, you can add it, but all needs.
Okay, we can say that anybody who has directly or indirectly contributed to the success of this program, to the organization of this program, I, from the core of my heart, thank him or her, and with the wishes of God, I would like to end this program. All of you, and I wish all of you to be safe in these times of pandemic, and hope we'll meet again on such virtual platform and deliberate on issues of scientific interest of the soils and whatnot. Thank you very much, everybody, especially Sir Dr. Suraj Bhan, sir. Thank you very much, and all my organizing team members. I'm very much thankful to you. I'm very much thankful to the Soil Conservation Society of India. Thank you, everybody. Goodbye. So I can end the meeting now.
Okay, thank you.
Thank you.
Goodbye.
Goodbye.
Chalo.